Stay manually.
Good morning, one and all. So it's that I welcome all. Stop that, not let's speak. It's a good. Good morning, everybody. I welcome all the delegates and our respected speaker, as well as the chairperson, to the day 2 of our 4th International e-conference, that is BMSeCON 2023. And today's session, it will be moderated by Dr. Gautam Sarkar, Professor, Department of Biochemistry from AVMC. So over to you, sir.
Good morning, India, and hello, world. The topic for the forthcoming presentation is heavy metal-induced lung injury. The session will be chaired by Dr. Mathan Raj. Dr. Mathan Raj is a professor and HOD, Department of Pulmonary Medicine at AVMC. He is an MD Pulmonary Medicine from JIPMER, Puducherry. And he is also a DNB in respiratory diseases. He is an excellent clinician with vast teaching and clinical experience. He has published many articles in national and international journals. His research topics were always of great clinical importance, like MRSA carriers. He's involved as chief investigator and co-investigator in many clinical trials related to the field of pulmonary medicine, markers of COPD, novel diagnostic methods for lung cancer, early diagnostic and preventive measures of hypertension, etc. So the chair is yours, sir.
Good morning, sir. Good morning to everyone. Thank you for the opportunity, sir. The topic for today, heavy metal-induced lung injury. With the advent of COVID from 2019 December or 2020 March, I think there has been much talk about the respiratory system. If you see, factually or historically also, we always call the top 10 killers or the top 10 cause of death. 6 out of 10 are from the respiratory system, and of which COPD, which was the 4th, has now become 3rd. So respiratory system was having a major impact on health over a period of long time. Now with the advent of industrialization and commercialization, there are more impact of pollution is causing a lot of impact on the lungs, and it is more pertinent than we have some topics specifically related to environmental pollution or industrial pollution, like the heavy metal induced lung injury. And to talk about this topic, we have an able person who is working on the specific field, Dr. Pooja Singh, who is an instructor in the University of Alabama, Birmingham, in the Department of Pulmonology, Allergy and Critical Care. She's a postgraduate from HNBG University, and she holds a PhD from University of Delhi in the Department of Medical Microbiology. More importantly, she worked as a research fellow and research assistant in VP Chest Institute. It's one of the premier institute in that particular department in India. She holds many honors and awards, like the best papers in the MicroCon conference and Indian Association of Medical Microbiology conference and ICMR papers, all those things. It's a very big list. Let's just make it short and sweet. She is also a member of many associations like IMM, ATS, and other things. And most importantly, being a research assistant, she is showing much research in the department, in the field of pulmonary medicines, mainly in the variables governing mucus accumulations in airway disease, and specifically brought on by the environmental stresses and the factors which is regulating repair and remodeling. Her research mainly focuses on matrix proteins, chloroflux channels, and the topic of today, heavy metal induced epigenetic regulation of mRNA. Over to you, Dr. Pooja. The stage is yours.
Thank you for such an elaborate introduction. Thank you, and good morning, everyone. I'll quickly share my screen. So as mentioned, I'll be taking you through the heavy metal induced lung injury work, and this is under the umbrella of Superfund Research Center that studies the impact of airborne heavy metals on the lung disease and the environment. So I would like to start by just giving an overview of my academic profile. So I completed my PhD in 2017 and I moved for my postdoctoral training at Rutgers University. And in my PhD duration, as well as my initial postdoctoral training, I looked at the implications of host pathogen interactions on lung health. And here I gained expertise in mouse as well as rabbit models of tuberculosis infection. And moving on, then I proceeded to broaden my area of research by looking at the environmental factors associated with lung injury. And for this, I did an initial postdoctoral training at Superfund Research Center, followed by my research faculty position, which is ongoing right now under the supervision of Dr. Veena Anthony, who's a pulmonologist, and she's the director of the Superfund Research Center. So the slides I'll be taking you through in this presentation will focus on cadmium as our focus of the heavy metal, then Birmingham, which is listed as the Superfund site. Birmingham is a city of the state Alabama, and the UAB SRP and the work that is ongoing here, and the legacy of Birmingham. And then the respiratory disease I'm focusing today is COPD. And we'll be looking at the airway remodeling that is happening because of this heavy metal, that is cadmium, and some scientific achievements from our Superfund Center. So cadmium, as we know, is the 65th most abundant element in the Earth's crust, and the exposures to cadmium could be through food, cigarette smoke, drinking water, air, and the occupational exposures could be through battery production, alloy production, the plastics, meltings and refining and whatnot. The point to consider here is that the half-life of cadmium in our body is more than 25 years. That means there is a lack of cadmium elimination mechanism in human, which contributes greatly to the cadmium-induced stress and accumulation in the lung epithelial cells. So as we know that heavy metals we can have the exposure of these heavy metals through inhalation, the ingestion or absorption. So inhalation through air, then ingestion through our food or water, and absorption through even the contact of anything. So mainly cadmium has been studied to be exposed through cigarette smoke, as well as the other previously mentioned sources of smelting and refining, etc. So since cadmium accumulates for a very long period of time in our organs, so it increases the oxidative stress, and which leads to the alteration into the immune system or increased susceptibility to infections, chronic inflammation, fibrosis, ultimately emphysema, and that's why there is an increased likelihood of chronic lung diseases. Mucus cell hyperplasia, that is increased mucus secretion or production, is the hallmark of inflammatory airway diseases, and it also leads to the airway obstruction, as we see in case of COPD. So this is a representation of lumen of airway, where we see that the airway epithelial cells have mucus and there is air liquid homeostasis that is maintained in healthy state. But in case of COPD, we see a disruption in or dysfunction in that, and we see increased accumulation of thick mucus in the airway lumen because of the increased cell debris as well, and thereby restricting or narrowing of the airway lumen. Now, why Birmingham, Alabama? So Birmingham is a city of Alabama, which was declared Superfund site. A Superfund site means it's the name given to the environmental program that is established to address abandoned hazardous waste sites. And in the Superfund site, we observed that there was high prevalence of COPD or chronic obstructive pulmonary disease as compared to a non-affected area, even though we took in account the ethnicity, that is African American population, as well as prevalence of smoking and the socioeconomic status. And we observed that the soil cadmium levels were really high in this Superfund site. Also, looking at the blood cadmium levels in the never smokers as well as smokers from the Superfund site was really high, significantly high as compared to the non-affected areas. So why COPD? So UAB Superfund Research Center was established in 2020, and EPA, that is the Environmental Protection Agency, had proposed 35th Avenue of North Birmingham as the Superfund site. And this was because of the high prevalence of heavy metals like cadmium, arsenic, manganese, and there was high prevalence of chronic lung diseases like COPD, asthma, lower respiratory tract infections, which are studied by other project leaders from SRP. And our project works on the COPD, the prevalence of COPD in this area. So looking at the legacy of the past of Birmingham, Alabama, that it's being called the Magic City of Alabama, because of the establishment of ABC coke, the bluestone coke, and all throughout the 20th century, Birmingham was a hub for the iron and steel production, which required coke to fuel the smelters. And you can see the condition of the residents of how they were treated here in Birmingham. So there was a very clear discrimination amongst the Black and the white population here. And Martin Luther King even stated that Birmingham is the most segregated city in the US, where you can see that these blue dots represent the Black population and the red dots represent the white population. And this is our Superfund site. And here is the UAB campus, the university campus. And we observed that the life expectancy was also very low here, around 65 to 70 years in this region. At present, although Birmingham has come far way in improvement of the air pollution, as you can see from 1970 towards to 2020, there has been a major change, but still there is prevalence of lung disease, and COPD is one of the most prevalent ones here. And even the kids are being diagnosed with asthma at a very early stage, as that of kindergarten to the middle school. So this lung disease has been across the lifespan. So what is COPD? So COPD is marked by the airflow obstruction. As I mentioned before, the mucus hypersecretion is one of the reasons here. And we basically see that the lumen of the airway is blocked for any airflow to pass. This is a CT scan image where A panel shows a healthy subject CT scan compared to a stage 2 COPD showing the blockage or narrowing of the distal airways, the lumen of the airway. And here is a cross-section image of the a lung section that is stained for different markers presented in green and red for the mucus or mucin protein. And you can see that this is a classic representation of airflow obstruction due to cell debris as well as the mucus hypersecretion in smokers with COPD. Moving on, we looked at the heavy metal content. So we used hyperspectral microscopy here to visualize how the heavy metal distribution was present in these lung sections. And this is a lung section from a resident from our Superfund site. And as you can see, this brightened area, this is all the metal content that you can see in the epithelial cells, and this is the lumen. And even in the macrophages, we see this, there is great heavy metal content. So moving forward, we are trying to look into what type of what content it is available here, so that we can know that whether it's a mixed heavy metal pollutant or a single type. So cadmium may be regulating mucus production, and cadmium has been shown to be highly significantly increased in case of the smokers with or without COPD when compared to non-smokers in n number of studies. And this is mainly because of the lack of an elimination mechanism that is present in human for cadmium. So there are different ways of studying the in vitro studies that are performed in our Superfund Research Center, where we use the 2D cultures to 3D cultures. We use whole lung perfusions. We even use lung on chip things, and our lab specializes on the 2D and 3D culturing techniques. So I'll be taking you through a very short study where we looked at the chloride efflux channels in the airway epithelial cells. So CFTR is one of the most or highly studied calcium chloride efflux channel that is present in the airway epithelial cells. But with recent studies, ANO1 is another chloride channel that has come into picture, where it is mentioned that this is involved in maintaining the air liquid homeostasis. So smokers, this is a representation of ANO1, where it was co-stained with the mucus. So this is a generalized pass staining for mucus. And then the brown stain here represents the ANO1. And we wanted to see whether ANO1 was present in and around our mucus secreting cells. So this is the inset of this lung tissue section image from the smokers with COPD, and you can see that the airway is blocked with the mucus content, and there is a high intensity stain for brown as well as pink compared to the control. So air liquid interface culture is what we started with, where we try to mimic the environment of in vivo environment, where there is an air liquid interface, and we can have these pseudo-stratified or differentiated airway epithelial cells with the cells that have cilia, so ciliated cells, goblet cells, or different differentiated cells of airway. And we looked at the ANO1 expression pattern when these cells were exposed to cadmium, and we observed ANO1 expression pattern was highly increased and upon low dose of cadmium exposure. And this we demonstrated with microscopy as well as through Western blot analysis. So we wanted to know what could be the interacting partners for ANO1, and at what stage can we be inhibiting or activating ANO1 to look at what was the effect of this cadmium-induced ANO1 expression. So we try to look into the microRNA and mRNA expression or interaction here. We looked into different databases for microRNA. If we found four of these listed microRNAs to be interacting with ANO1, and we proceeded our study with microRNA 381 specifically because it interacted with all the known sequences of ANO1 mRNA, and secondly because it was not inhibiting or interacting with EZH2, having no impact towards EZH2. However, it's been reported that EZH2 downregulates MIR381, and we tried to see that if it was for the proof of concept, if it was happening in the presence of cadmium. And we observed through the expression analysis that EZH2 expression was increasing with time upon exposure to cadmium, and MIR381 expression was going down. So we used an inhibitor for EZH2 and observed that MIR381 expression went up in the presence of inhibitor for EZH2. Now, EZH2 is an epigenetic regulator, and heavy metals like cadmium are known to induce bring epigenetic changes. So H2 is one of the epigenetic marker that is upregulated in presence of cadmium. And so we observed that MIR381 was being inhibited here. Now we wanted to see how in our airway epithelial cells, MIR381 and ANO1 expression pattern was present. So we used a MIR381 inhibitor and observed decrease in the MIR381 expression, and therefore we use this inhibitor to analyze the expression pattern of ANO1 mRNA, and we observed a significant upregulation upon inhibition of MIR381. So we used cadmium in the form of cadmium chloride, and we observed that ANO1 expression was increasing in the presence of cadmium, and MIR381 expression was decreasing in the presence of cadmium with time. This is for 24 hours and 48 hours. So with that, we prepared a MIR381 mimic in these airway epithelial cells, and this is again an air liquid interface culture, but we are looking at from the upper side, so from the apical region, so as to be able to see the ANO1 expression pattern. And here you can see that upon inhibition of MIR381, ANO1 expression was increased, but decreased upon in the cells which had overexpression of microRNA 381, and we have shown it with the Western blot and gene expression analysis. So we used this MIR381 inhibitor, and we also exposed the MIR through the mimic the airway epithelial cells which had the microRNA 381 overexpression, and then expose them to cadmium chloride together. And we observed that the mimic was able to kind of inhibit the expression of ANO1 even in the presence of cadmium chloride. And so we did the Western blot analysis for that, and we observed the same here. So finally, we looked at the effect of this MIR381 expression on mucin protein, because that was ultimately what we wanted to see, if it had any effect on the mucus production. And we looked at one of the critical marker of mucin protein, that is MUC5AC, and we observed that even in the presence of cadmium, MIR381 overexpressing cells were able to inhibit the mucin protein levels in these cells. So we summarized that microRNA interaction with ANO1 could be regulating the effect of cadmium exposure here, and that cadmium from cigarette smoke may cause the mucus hypersecretion in patients with COPD. So this is one part of our work that we defined in the 2D culturing or air liquid interface culturing. Moving forward, we are trying to look into the 3D cultures, that is organoids and airway organoids. As recently, it's been well defined now with the apical and basal cell polarity, and the 3D models would give us an environment where we could contain the mucus secretion within the organoids and look at the mucus hypersecretion there. So this is the airway organoid cultures that we have been preparing using the brushings from the endobronchial brushing from the patients. And this is a short video showing how our 3D models are. And this is the cilia is present outside, and that's why you see this kind of movement of the cells here. And we are also developing these mouse model, the whole lung organoids from mouse lung. And here is just to show a mouse lung tissue that is stained for the surfactant protein here for the 82 type cells, and the extracellular matrix protein that is collagen 1, and that can be seen here in the mouse lung organoid as well. So it's kind of mimicking the in vivo structure here. So organoids provide that kind of cellular arrangement by self-arrangement, which helps us, which would help us in detailing the toxicity or use it as a precision tool here. So moving on, I would like to also mention here quickly about another study that we have done using a coagulation factor, that is fibrinogen, and it has been discussed in a number of papers that fibrogen level significantly rises in COPD patients compared to control, and this has been seen throughout the different stages, as the severity increases for COPD, that the fibrinogen levels increase. in plasma as well as the valve. So just an overview that fibrinogen and fibrin play together. These level needs to be maintained as they are important factors in the coagulation pathway, and they form the blood clots. So whenever there is a wound, for healing, there is a signaling to the liver which releases fibrinogen, and it contributes in the formation of the clot. So we observed that the cadmium levels were higher in COPD patients, the smokers with COPD, and we analyzed it in the non-smokers with COPD, in ever smokers with COPD, the ex-smokers with COPD, and ever smokers of COPD, that the cadmium levels were really high. And then within these, we looked at the fibrinogen levels, and we observed that in case of COPD, the ever smokers had really high levels of fibrinogen in the lung macrophages. And yeah, and here we show just the density plot for that. And here we correlated in the right panel the cadmium levels with the lung function test. And we see that the lung function test decreases with increase in the cadmium levels in the COPD lung. This is for the same set of patients that were analyzed here. So we then compared the fibrinogen levels and the COPD with the increasing COPD concentration, and you can see this linear correlation that it is positively correlated with the increasing concentrations of cadmium. And so we compared it with the lung function test as well, and we saw that as lung function tests were decreasing, there was increase in the fibrinogen level in the lung tissue. And we used our Murine model for COPD, which we established in 21 days, and we are showing through this IHC of the lung sections that how the lung parenchyma or the lung structure changes with because of the cadmium exposure in mice. And we see that the collagen, that is an extracellular matrix protein, that it increases upon cadmium exposure. And we also see there is increased airway resistance in this. The mean alveolar A spaces, like these pieces that you see, these also increase upon the exposure to cadmium. So this is just a proof of concept. And we see that the fibrinogen level in the bronchoalveolar lavage fluid as well as plasma is highly increased upon exposure to cadmium. And this was positively correlated with the hydroxyproline test that is done for the extracellular matrix protein, that is collagen. So the fibrinogen levels were in direct correlation with that. And we also observed that upon with time of cadmium exposure, this fibrinogen levels were increasing. And we also observed that when these mice were exposed to just cadmium, just fibrinogen, or the combination, there was increase in the release of TGF-beta cytokine, which is a profibrotic cytokine. So thus we concluded that the cadmium exposure disrupts the normal balance among coagulation and fibrinolysis, leading to fibrin deposition or formation, and that the overproduction of fibrinogen promotes cadmium-induced collagen accumulation and profibrotic cytokine production. So I would like to finally kind of summarize our scientific achievements through the Superfund Center, where we are collectively working with the biomedical or the engineering, material engineering department, and other biological and departments, where we are collectively working towards like a development of these 3D lung pulmospheres, which we use as a predictive tool for the predictive toxicology with exposure to different kind of heavy metals. And then we are also working towards the acute lung injury in the presence of heavy metals and the infections because of the Streptococcus pneumoniae. And then we are also looking at the biomarkers that we can have through the blood test. For example, we had published in the scientific translation medicine about vimentin, that citrulated vimentin levels are really high in lung fibrosis cases. And moving on with the material engineering side, we are also collaborating with them for production of biomass so that we could work towards the remediation of this heavy metal pollution in our community. So this is just a list of publications, and we are also connecting with our community increasing our community connection and bringing awareness of these raised bed gardenings so that there is less exposure to consumption of the of food at least, and also collaborating with our community and bringing awareness by reaching out to school kids and involving them in our newsletter publications. Like this is a picture drawn by one of the kids from the school of this community, and handing out flyers about bringing awareness about asthma, COPD, and also providing them reusable N95 masks that were produced and formulated within our engineering department under the Superfund banner. So I would like to conclude by stating a few lines that we should never doubt that a small group of thoughtful, committed citizens can change the world. Indeed, it is the only thing that ever has. So finally, I would like to extend my acknowledgements to my mentor, Dr. Veena Antony, who provided me with the opportunity to work in her department, and my lab mates and our division director, Dr. Dwensfield, and our SRP community. And thank you, Dr. Manju, for providing me the opportunity to speak in front of the panel here. Thank you.
Thank you, Dr. Pooja. I think that was an extensive and detailed analysis about the role of cadmium and the exposure cadmium causing in COPD. We all know that smoking causes COPD, but there has been always a small part of cadmium in smokers, in the cigarettes, which is going to cause. I'll just add on another point which you elaborated. Even if you stop smoking, from the day of stopping smoking, even after that, till 10 years, the inflammation still persists. The reason probably is because cadmium has a much long half-life, close to 25 years. So even after stopping smoking, there will be ongoing inflammation, which can cause lung damage further. So that is another point I just wanted to add, which we see clinically, because patients come and ask, no, we stop smoking, but still we have symptoms. So that is another thing. I just want to ask you a simple question. So there is always a duration as well as dose relationship for any heavy metal. So do you think that is there a role for this amount of cadmium causing just mucus regulation, this amount of damage will lead to fibrosis, or this dose of cadmium is going to cause more of fibrosis? Is there any dose relation leading to just mucus hyperregulation, or it's just extending to fibrosis part, something like that? Because the moment you say MAR and the other gene, no, you end up in fibrosis. The ultimate thing is it's ending up in fibrosis. So is there a dose relationship which you found in your studies?
There has been dose relationship that we have previously published from our lab. I have not in particular worked on that because this was something that was already done and published from our lab members previously. But yes, you are right, there is dose relationship, and that's why we have defined and come down to a particular dose where within 20 days we achieve the kind of structure that we are looking for there, and we stop there. We don't go for the survival studies, but we stop there to see what is happening in that much duration. So that's why we have different kinds of model where we go up till 8 weeks also. We have gone to 6 months also. So there is definitely, as longer we study, more we are going towards emphysema. But for mucus hypersecretion, we came down to 21 days.
Yeah, fine. The other thing is that there have been a lot of biologicals which has been developed for asthma, like mepolizumab, but whereas we have very few biologicals for COPD. I think the way you are working on your research, I think you're already starting working on the role of biologicals in COPD, on the MAR expressions. I think that will be a good source for the future drugs. I think that will be a part of the trials, which I think your study started working on specific drugs, right? On the effect of the drugs on these MAR expressions.
So my work is targeting ANO1 expression pattern in this. So I am using different drugs, for example, niclosamide, I won't go under for the clinical implications of that, but with the mouse model as well as with the organoid cultures, we are right now using niclosamide. We are also using different like other markers here where we are targeting oxidative stress studies. So for that also, like targeting lipid draws, the inhibitors for that, that's the most that we have gone as of yet.
Yeah, fine. And for the last question, we know that cadmium is a part of cigarette smoking. Is there a role of cadmium even in e-cigarettes? Have you differentiated between e-cigarettes and the normal cigarettes? Is there any studies? I mean, you have a plan, something like that?
With East, no, we don't have. Yes, one of our collaborators have done e-cigarette studies, but they have not actually looked into the effect of
Because
cadmium.
that's what. So if that is another reason, like I'm not
This
promoting
is a great
e-cigarettes.
suggestion for us to have a comparison.
Because the role of cadmium is more in cigarettes, and again the role of filters come when you go to e-cigarettes, right? Like they say cadmium is comparatively lesser in e-cigarettes. Of course it has its own implications, but maybe when you have a wider comparisons over a period of 5 to 10 years, you can see whether the expression of ANA1 is different in patients who have e-cigarette smoking compared to non-cigarettes with non-smokers. So something like that. I mean, because you have the technical expertise as well as the facility to do it. So just a small idea.
Yeah, this is a great suggestion. Yes, thank you so much.
And I think we all had a wonderful session. So over to you, sir, Dr. Saka, sir. Thank you, sir. Sir, you need to switch on your mic.
Please unmute, sir.
Yeah. As far as cadmium and COPD is concerned, I'm a layman of the highest order. So, but I have a few kiddish questions. Please forgive me. I think both of you talked about the half-life of cadmium, which is around 25 years. And my question is that, for heavy metals, for our poisoning purposes, we use heavy metal chelating agents such as dimercaprol and others. Do you have any role of such drugs in the removal of cadmium from the body? Am I through? Could you hear me? Yeah.
Yes, sir.
So cadmium elimination has been a critical thing here. One, for the reason of high penetration value. So heavy metal absorption is not something, so heavy metals are already absorbed on the surfaces of these particulate matters, which are of very small size, like 2.5 microns. So they kind of penetrate really, really deep to the distal airways. And that's what we see, the narrowing or the obstruction of airways, as I showed in the CT scan as well. And that's one of the reason to reach out to these for binding or chelation or like taking them out from the cells. So that's something that has been a difficulty there. But I think that's the most I would be able to.
Just an observation before I go to the next question, that Birmingham in UK and Birmingham, Alabama seems to have a similar industrial background. Just an observation. Okay, my next question is, other than cadmium, of course, I'm not, Dr. Matan Raj is a better person to comment on it. In our country, in India, aluminum is a big problem also, which is also, though not by definition heavy metal, but is considered as a heavy metal because of its toxicity. So what is the role of aluminum in causing COPD?
Sir, yeah, sir. I'll just
area of expertise,
so the
but I
main
think-
okay, I'll just add on to it. The main aluminum per se, like all heavy metals, will have two types of diseases, sir. In an acute exposure on a high dose, they cause simple metal fume fever with like normal symptoms of cough, cold, breathlessness, and there'll be some tracheitis and bronchitis. And when it is exposed to smaller doses over a period of long time, you get something called pneumoconiosis, and it will eventually end up with emphysema, fibrosis, and other things. Like most of heavy metals will have similar symptoms. But why specifically there is much talk about cadmium is a part of the cigarette, which even a non-industrial, non-worker or a common man uses on a day-to-day basis. So there is a large population which is being affected. But when it comes to aluminum, it is only a small set of industrial workers, or maybe you can extrapolate it to their spouses or their family members. But still there are a lot of studies on aluminum, as well as if you want to export, like we can go to beryllium in the glass industries, and silica in the sand industries and the glass industries. So all of them will cause exposure. It will lead to some initially in the metal fume fever, or it's just simple cough cold, and over a period of time it will end up with bronchitis, airway inflammation, and the long-term emphysema. And there are even now studies indicating that this chronic deposition of metals can, because of the chronic inflammation, can eventually lead to bronchogenic carcinoma, or even sometimes the tracheal carcinomas in future. But these are all, we don't have enough data, because the heavy metals are, these are all very small studies, and it's in pockets. So we need large studies and multi-centric trials to prove the correlation. But yes, aluminum is still a part of the heavy metal induced lung injuries in us, and we don't see it on a day-to-day basis, sir. It's relatively very less compared to the other disease which we see on a day-to-day basis.
Thank you, Dr. Pooja, and thank you, Dr. Madan Raj.
Just
I mean,
1 minute, sir. Sir,
yeah.
1 minute. Just want to add, because you asked about the aluminum thing. We have a speaker coming up today or tomorrow, I suppose. It's the same topic, aluminum toxicity and its effect on lung diseases is included in this same conference. Okay, so continue, sir. Over to you, sir.
Though virtual, as a token gesture, we have a virtual memento for both of you. And first, the memento and certificate goes to Dr. Pooja. And Dr. Matan Raj, for you too, we have a memento and a certificate.
Thank you, sir.
Thank you so very much for the.
Thank you, sir.
Thank you very much, Dr. Pooja. Thank you, Dr. Pooja. I know that you are out of your station and you are attending a conference, but still thanks a lot, and for coming over at this time of the hour from US and enlightening. It was really very interesting, and a lot of eye-opening things were shared with us. And hats off to the not only involved in research. I was so impressed to see your social work you are doing, also your institution or your foundation, along with doing research. Hats off to you and your team. And Dr. Madan Raj, you, to chair this and making this, this 40 minutes talk really interactive, and sharing with us a lot of information other than the resource person. Both of you, I really thank you so much from the depth of my heart. Thank you for joining us today. Thank you.
Thank you, ma'am. Thank you, ma'am.
Good morning, everyone. I welcome you all to this session. I feel extremely happy to introduce the chairperson of this session, Dr. Jamuna Aswathnarayan. Dr. Jamuna Aswathnarayan is an accomplished assistant professor in the Department of Microbiology at the School of Life Sciences, JSS Academy of Higher Education and Research. She obtained her MSc and PhD from the University of Mysore. Followed by her PhD, Dr. Jamuna pursued postdoctoral research under the UGC sponsorship at the University of Mysore itself. Her postdoctoral research was marked by a focus on functionalizing nanoparticles and peptides, which found applications as antimicrobials, biofilm inhibitors, and anti-cancer agents. She also gained valuable experience during a brief stint at Cardiff University, UK, specializing in metagenomics. Dr. Jamuna has significantly contributed to her field with 21 research articles and 18 book chapters, and co-editing 6 books and 1 for Springer Nature Group. She has around 1,300 citations with H index of 11 and ITEX index of 12. She has been recognized with several grants and awards, including the Interstellar Initiative Earlier Career Investigator Award in 2021 for her research on microbiome dysbiosis in lung cancer and its implications in CAR therapy. Dr. Aswathnarayan actively engages in research and mentorship, having guided more than 20 PG students and currently supervising 2 PhD students. I am happy to welcome you, ma'am. Now I kindly request you to introduce today's speaker of this session. Thank you. Over to Dr. Jamuna.
Good morning, sir, and thank you for the kind introduction. I would like to thank the organizers of this conference and AVMC for giving me an opportunity to chair the session. Honored to our next speaker, Dr. Ramasamy Damodaran. Sir is an assistant professor at Mahatma Gandhi Medical Advanced Research Institute. Sir's focus areas have been microbiome research, antimicrobial resistance, taxogenomics, and comparative genomics. Sir has previously worked as a consultant at the ICMR National Institute of Epidemiology, and he was there investigating the infectious disease transmission. Sir has also had the experience of being a principal scientist at Avans Bio Private Limited, and he focused his research there on infectious diseases, gut microbiome, environmental microbiome, and probiotics. And he has a vast amount of international experience, wherein he carried out his postdoctoral research at Seoul National University, and then in Marseille University in France, and Busan National University in South Korea. So sir is going to today talk about an interesting topic, that is functional diversity of human gut microbiome in health and disease. And we are all excited to hear sir's talk about it. And over to you, sir.
Thank you, ma'am, for a nice introduction. Thank you for the organizing team for giving me that opportunity. Just please let me share my screen. Can you see my screen, ma'am?
Yes, sir, we can see.
Yes, sir, it is visible. It is visible.
Yeah.
You can start.
Yeah. Good morning, all. So I'm Ramasamy Damodaran. I'm working as a faculty in MGM IRI Pondicherry. Actually, today I'll be talking on functional diversity of human gut microbiome in health and disease. So mainly my talk will be focused on how microbiome is playing a major role in human health and disease. And I will also highlight with various examples how the functional diversity of microbiome is associated with health and disease. We all know that microbiome and microbial environment differ from one another, starting from like a hot spring, cold environment, birds, animals, human. Everywhere you can find microbes, but their abundance and diversity varies from one location to other location. So before going to my and see that microbiome functional diversity, how it works, just a few terms we should know. Actually, one is the genome and proteome. and that is entire gene set is and the entire protein set that is involved, that is mainly associated with the functional diversity, because that genome and proteome have direct association with the functional diversity. So here I'll show that how the concept, the microbiome and microbiota works in different way actually. So microbiota, generally they use a microbiota if is to study only the microbial composition and diversity. When it comes to microbiome, it's related to both the microbial diversity, the abundance diversity, and also the functional potential of the particular environmental microbes. So human contain different kind of microbes actually. So it depends on like oral microbiome, urogenital microbiome, skin microbiome, and digestive microbiome. These are the major microbiome actually. Apart from that, many other microbiome, almost many body parts have microbial content. And sometimes even infectious stage, blood also have some microbes actually. So the major one, the composition of microbes include mainly the bacteria, virus, and fungi, and archaea parasites or microparasites also contribute in the composition of microbes. When it comes to abundance, the bacterial, like here I have given that what is that the diversity and the abundance actually. So bacterial the virus composition, viral diversity is highest among human microbes. They reported that 140,000 viral species were present in human gut alone. To understand the microbiota and microbiome and functional diversity, the Human Microbiome Project was started in 2007 by NIH. Actually, their main aim is to characterize microbial community using culture-dependent methods by using 16S amplicon sequencing or shotgun metagenomics, that is whole genome sequencing. With 16S amplicon sequencing, we can analyze only the microbial composition and diversity. But when you go for a whole genome sequencing, that is shotgun sequencing, we can also study the functional genes and the antimicrobial resistant genes. Various functional genes can be analyzed from the results of metagenomic sequencing. From a Human Microbiome Project, thousands of samples were submitted till now, but among the samples that microbiome samples among different body sites collected, so gut microbiome is on the top, and the second is oral, and the third one is vaginal microbiome samples were submitted. So how microbiome is playing a role in health and disease? Like what is the healthy microbiome? What is the pathogenic microbiome? How they are playing major role? What that is the significant issue. For understanding the role and functional diversity of microbes, that is the human microbiome, just a little bit about chemistry, we should recall that here I have mentioned that the life is like chemistry: friend to concentrate, enemy to dilute, joy is to precipitate, and sorrow to evaporate. In general reactions, like any substrate plus reagent, it is catalyzed to product and byproduct, which is like the many factors involved in catalysis of any chemical reaction, which include enzyme, coenzyme, cofactor, allosteric factor. But when it comes to gut microbiome, so what kind of chemical reactions are happening in human microbiome? And how many catalysts are present in human microbial environment, and what are the byproducts are released into the human microbial ecosystem, and how it is influencing the human health and disease. Like here, friends to concentrate means, like, for example, commensal microbiome to be balanced, and the enemies means the pathogenic bacteria to be diluted, and joys to precipitate means the beneficial metabolites to be precipitated, and the pathogenic and toxin to be evaporated. Like that we can compare. So this is a simple example, like how the simple biochemical reaction, like for example, any bacteria or fungi or virus, it will take the nutrient and it will catalyze and it will release byproducts and metabolites. So that byproduct as a metabolite, product and byproduct, sorry. So these metabolites will be used by other bacteria or virus or any other eukaryote present in the human body. Sometimes human also use the bacterial metabolites. So this is a classical example how what is the important role of microbes, that particularly human gut microbes. These are the 5 different roles actually, like whether they help in digestion, they produce vitamins, they boost our immune system, and they protect from our pathogens. This is actually the answer is all 4 are correct actually. And Another example for understanding the significance of gut microbiome in health and research, there is a Indian practice of eating the drinking fermented rice drink. Actually, the leftover rice will be soaked in the night, and then next morning they will drink that rice. So this include a lot of beneficial bacterial species present in this fermented rice drink, and also a lot of vitamins and minerals actually. So based on this, actually, recently Stanley Medical College have conducted a project with the inflammatory bowel disease patients, the 2.7 crore project. They included Crohn's disease and ulcerative colitis by a conventional therapy along with the drink of the fermented rice drink. They started observing uh, the patients were responding well and cured better than conventional therapy. Along with conventional therapy, the group who has taken a fermented rice drink, they felt better actually. So recently in Japan and Canada have come a commercial drink based on fermented rice. That is, they are called as a magic drink. So here gut microbiome, actually there are beneficial bacteria and pathogenic bacteria. For example, some of that like Clostridium difficile jejuni, these and all considered as pathogenic bacteria. In general, like each bacteria code for approximately 3,000 genes actually. So because each bacteria code for 3,000 genes, like for example, how many bacterial species are there actually? So each bacteria code for 3,000 protein. This is higher than the number of genes coded by a bacterial alone is higher than the total number of human genes coded. So the bacterial diversity also varies due to a pH gradient in human people's the digestive system. And majority of the highest diversity is present in small intestine and large intestine, where the more diverse bacteria live, and wherever more diverse bacteria is there, that is equal to more functional diversity. That is more diverse genes are present. But the composition of gut microbiota includes firmicutes, bacteroids, proteobacteria, and actinobacteria. These are the major phyla, and other minor phyla include Fusobacterium, Verrucomicroca, Spirochaete, and some kind of archaea also present in human gut. Actually, these all together as a synergistic way, they play an important role in harvesting nutrient, assisting immune system, and protecting the host against pathogens. So based on the diversity and of human microbiome, the human we can group a blood grouping of human based on gut microbiome. Also, we can group people. That is based on fecal microbiota. They have grouped the whole world. All people in the whole world can be divided into three groups. Either they belongs to bacteroids, or Prevotella, or Ruminococcus. So we Indians mainly belong to a Prevotella group, which is mostly related to vegetarian diet. Actually, here I have mentioned that what kind of diet is associated with different uh, group actually. So proteins and animal fat diet was linked to the bacteria genus, and carbohydrate lacking meat and vegetarian diet was linked to Prevotella disease, and alcohol diet was linked with the Ruminococcus. So first, we should know before going for functional diversity, how we are getting the bacteria into human body actually, starting from the birth, how they are getting bacteria, birth, the lifestyle and the diet, and whether vegetarian or non-veg, or a child or the lifestyle, and whether that's urban or rural environment the babies are growing, whether the childhood to adult, how much drug or antibiotic they are consuming, whether they are alcoholic or smokers. These all factors will decide what kind of microbes should colonize in your gut. In 2013, they have reported then from Canadian group actually that choice of birth, feeding method changes baby's gut microbiome. That is, they reported that babies born by C-section have less diverse microbiome compared to babies born by vaginal delivery. And the same way, the babies consume breast milk more beneficial bacteria than the formula milk babies. So this is well-known concept. Recently, they have proved that the diet play a major role in abundance and diversity of the bacteria. So wherever abundance and diversity is changing, where the more diversity means more functional genes you can get. And this is another example that earlier they were thinking that breast milk is sterile actually, until this study came in 2013 that they identified that nearly 700 different species of bacteria are present in human milk. breast milk, and most of the bacteria are beneficial, and it is consumed by baby as a probiotic. Like babies are taking probiotic bacteria from mother's milk. So they proved how mother's milk, like breast milk, contain bacteria, and they proved that the gut bacteria is moving to the breast milk actually, and some says immune system help actually. That still the research is going on how the gut bacteria is moving to breast milk, and then from breast milk babies are getting actually. So they identified that bifidobacterium, parabacteria, and 24 members of Cholesteria species were present in maternal feces, that is mother's feces, breast milk, and the baby's neonatal feces actually. So even probiotic therapy like treatment of infectious mastitis, when mothers consume lactobacillus fermentum, L. fermentum, and L. salivarius, two species, the lactating woman who ingests the same salivary species exists in their breast milk, and that means that same bacteria, whatever bacteria their mothers is consuming, is transferred to baby through milk. So family and close friends share mouth and gut bacteria, like through aerosol contamination also, like microbiome colonization happen to neighbors and friends. And the microbiome composition and diversity varies geographical based on the geographic location. And microbiome, the microbial species also can be used as a marker for identifying any pathogenic species. For example, this Methanobrevibacter species, Smith is an archaea species. Whoever is having this species, they can be easily identified by measuring their methane composition by breath test actually. So gut bacteria also associated with some kind of autism. Not all kind of autism is associated, and some patient can raise the alteration of mitochondrial function and fat metabolism. For example, the propionic acid produced by bacteria have the ability to induce a brain symptom, brain behavioral changes actually. The elected level of propionic changes immune function and which affect metabolism and energy absorption. This is directly related to autism kind of symptoms in children. So gut microbiome also affect the response of drug actually, whether the efficacy of the drug or the drug can be altered to some other product, that also can be possible by microbiome actually. And this is a simple example how the different antibiotic drugs relate to abundance and gut microbiome. Like these are the drugs used for diabetes, actually metformin and various other drugs. And but based on the different microbiome level, like Akkermansia level increases, proteobacteria decreases. If we take this second, if we take metformin, like Intensivibacter, Lachinobacter, and these and all will decrease actually. So these various examples are there. I'm not going in detail actually. So how the microbes are associated with the disease actually, whether the disease is associated with microbiota or microbiota is associated with disease actually. So the both way it is happening actually. Even non-communicable disease are also reported to have associated with imbalance or imbalance in microbiome diversity. In general, microbiome diversity is associated with functional potential, and this functional potential will be directly linked with difference in biochemical pathways. And different biochemical pathways means related to metabolites, and that is directly related to health and disease things. So here I have described, I have mentioned here that the various diseases are associated with imbalance of microbes. For example, the gut microbiome is imbalanced, or various disease can cause imbalance gut microbiome. So the imbalance in gut microbiome is associated with a strong association with the health and disease. So what will happen if imbalances there or pathogenic bacteria is very high? We have to cure means we have to consume antibiotics. So antibiotics consumption, if it is proper, no issues. But without any suggestion, unwanted use of antibiotics may lead to antibiotic resistance, and that is due to antimicrobial resistant genes. So if you are without antibiotic, we cannot kill the pathogen. So the reason method the other method to kill the pathogen is using the probiotic, prebiotic, and symbiotics. So probiotics are live bacteria directly consumed. Prebiotics are any food substances that can be used to for the development of for the growth of beneficial bacteria. Symbiotic is combination of both probiotic and symbiotics. So like in the recently, like even dead bacteria, like postbiotic is the compounds produced by bacteria That also can be used as postbiotics to kill the pathogen or to improve the balanced microbiota. These probiotic, prebiotics, and biotics are used. So many people know that what is the difference between the curd and yogurt. The curd is made at home actually, which contain more number of beneficial bacteria. But in yogurt, only the selective bacteria which is used in the industry. For example, lactobacillus yogurt or bifidobacterium yogurt. This is contains selective bacteria. This is contain more diverse bacteria. So these are probiotics also can be used to imbalance and to balance the gut barrier. But if there is no beneficial role by antibiotic, like for example, Clostridium difficile is not working. So there is no other way that we have to go for transplantation, none other than this. Already we know that many transplantations are existing, but the recent one in the field of gut microbiome is fecal microbiota transplantation. That is called the FMT procedure. This is mainly they started when the person was not responding to the Clostridium difficile pathogen with antibiotic. If we have to save the patient, so they started transferring the entire fecal sample from healthy subject, from for example, from some of his family member, brother or sister, they can donate the fecal sample. The entire fecal sample, initially the fecal transplantation was happened through nozzle tube, and directly they put the fecal sample into the stomach of the infected person. But fortunately, it was working fine, and more than 90% success the rate was there that they could successfully kill the Clostridium difficile and save the patient. But people are hesitate to take this kind of treatment directly, putting the fecal sample into the stomach actually. So they were looking for alternative. And even for conducting fecal transplantation in Cambridge, they started a stool bank, like blood bank. They started collecting fecal sample with good beneficial bacteria. That means person with high diversity of beneficial bacteria, if they feel that their microbiome is low, and they can donate the sample. Like they store the sample after knowing the microbiome diversity, and that one person's fecal sample can be used to treat 4 persons infected with the Clostridium difficile. So in USA also they started, there is a company called Open Biome, and they started fecal bank in many different states and a number of states. So in India, only few research institutes they started doing fecal microbiome transfer. So the alternative to fecal microbiome is poo pills actually. The custom designed poo pills also can be used to treat microbiome diversity like Clostridium difficile or any other gut related infection actually. So here what they will do, they take the fecal sample, they filter all debris, and only the bacterial capsule will be powdered and make it made as a capsule. This is custom design, and this was shown that 100% success rate in killing that Clostridium difficile infection. The Spanish group also came with a sausage from baby's poop sample actually, because the baby's poop mainly come from, most of the bacteria come from mother's milk, that is mainly composed of probiotic potential species actually. So they have isolated the bacteria, and they used sausage to get the beneficial bacteria from the babies actually. So another way to understand the functional potential is the gastric bypass surgery, that is a bariatric surgery actually. This is one of the major surgery used for obesity. They do actually. Millions of people are waiting for the surgery in USA actually, but it's not cost effective. And they observed that in animal, like before surgery and after surgery, there is a shift in microbiome diversity and abundance actually. So after surgery, they reduce the weight, but apart from reducing weight, their microbiome diversity also reduced actually. Based on this, they conducted an animal experiment, like the obese mice was treated by gastric bypass surgery, and it became lean mouse, the normal mouse. And they have taken fecal sample from the bariatric surgery conducted mice, and the fecal sample was transferred to another obese mouse without surgery, like without surgery. After consuming, like a transfer FMT procedure, fecal microbiota transfer, without surgery, the animal weight started reducing. So the same kind of thing can be applied in human, but now it is under clinical trial. And in future, like microbiome, like a fecal transplant based weight reduction, so possible. So some mice, they can also degrade toxins. There's some bacteria present in wood rat. They consume usually the poisonous plants. Actually, we cannot consume because that the poison degrading bacteria is present in human. So some the milk companies, like they wanted to show high nitrate like a high protein content in the milk actually. For that, they have adulterated the milk with the melamine actually. So when melamine is adulterated, the adulterated milk consumed babies were facing some issues with the kidney related problem actually. So later they identified this actually it happened in 2018 with this many victims. Actually 54,000 babies were hospitalized and 6 infants died with the kidney stones. And they found that the problem is not with directly with the melamine they mixed actually, but the melamine was metabolized by a gut bacteria present in the babies that is called the Klebsiella terrigenea. This Klebsiella terrigenea metabolize melamine, and the melamine will be converted into cyanuric acid. This is toxic to kidney actually. So they also checked this animal experiment actually. An animal fecal sample with mixed with the melamine can directly produce a cyanuric acid within 24 hours. So they found that why all children were not affected, even though this bacteria, melamine is consumed, the babies were hospitalized. Only few babies were died. All the babies died were reported to be from rural area, where in rural area the babies play with the soil. Actually, this Klebsiella terrigenea was colonized from soil. They got from soil to their gut bacteria. The urban children were not having this bacteria, so they didn't get much affected. They were not affected by this particular adulteration. So my work mainly, I'm working on isolation and genome-based description of novel bacterial species actually by metagenomic based and whole genome sequencing based actually. So people with health and disease, I'm analyzing metagenome based functional diversity. So my main work is taxonogenomics actually. This is if you want to know the entire functional genes, we have to know the complete sequence of the genome actually. So with the draft genome, we will be missing some genes actually. So with conventional method, with the 16S rRNA and the other biochemical method and microbiological method, we can describe the taxonomy. And the reason we are describing novel species and taxonomy is whole genome based actually, along with the conventional therapy like microbiological, biochemical, and phylogenetic analysis. We have also incorporated whole genome data with for identifying and characterizing the whole genome sequence. These are the novel species I have described. These are isolated from human guts. So why we have to do the whole genome sequencing? If you are not doing a whole genome, entire genome of bacteria, we will miss some functional genes actually. So for example, this is the entire, till now, how many bacterial genomes are present from NCBI were taken actually. From that, we have analyzed the carbohydrate active enzymes. So how many are there? Whether they varies among different phyla actually. For example, you can see the clear variation of the carbohydrate active enzymes in bacteria is Firmicutes, Actinobacteria, Proteobacteria, and Fusobacteria. So, for example, Fusobacteria, you can find that the carbohydrate esterase are very high in Fusobacteria. So, what is the significance of that? This is one example I have given carbohydrate active enzymes. The same way, many functional genes and cake pathway genes can be analyzed. And but due to time constraint, I'm not showing all the results here. So this is another analysis report that how cake pathway genes also play a major role. If you take the entire microbiome diversity of all the bacterial genomes of human gut, the functions varies actually among you can find here that the top one more functional genes in gut microbiomes are associated with the carbohydrate metabolism, amino acid metabolism, and xenobiotic biodegradation and metabolism. This one is interesting, xenobiotic biodegradation, but we don't know what kind of ingredients are present in many food actually, like other than like carbohydrate, fat, and protein. So many other chemicals and other things for environmental contamination will be there. This we have to. I'm mainly focusing on this one to understand what kind of xenobiotic degradation is happening by gut microbiome. So aging also play a major role in microbiome diversity. That for example, in elderly people, their diversity will reduce actually because of that they face some digestive issue. So for aging related one, so with the like modern diet with rich in salt, sugary, and fatty processed food may damage the gut actually. So to manage a healthy gut, we are considering food like fruits, vegetables, and seeds, beans, nuts could be combined with exercise to help protect a healthy microbiome. If you are maintaining healthy microbiome with the balanced one, the functional diversity of your microbiome also will be known. So diversity of intestinal microbiome and food habits also, many people they have reported that probiotics and prebiotics. Already I have told that fermented foods have a more beneficial in increasing the diversity. So in summary, I would like to summarize that host microbes are very essential because they help human hosts through their functional diversity. So imbalance in host microbes are associated to many different diseases or clinical symptoms. So identification of symbiotic and pathogenic bacteria species and to characterize their functional potential is very essential. Reducing antibiotic intake and take only if it is necessary will be associated with it. Like we can avoid antimicrobial resistance if you take antibiotic judiciously. And use of probiotic, prebiotic, and symbiotics and postbiotics when required to maintain the balanced microbiome. So this is the talk today, and take care of your microbiome, and your microbiome will take care of your functional diversity. So if any questions, I can reach me through this mail ID. Thank you. I'm waiting for the questions.
Thank you, sir. Thank you very much for the impressive talk. So I have a couple of questions actually.
Yes.
So which E. coli species is more prevalence in the gut selection? Because E. coli has many species. Which E. coli species is more in the gut microbiome?
There are nearly 30 different E. coli species are reported in actually. That E. coli is the species actually. There are different strains actually. Many number of E. coli strains are there.
Okay, I got it.
Yeah. For example, E. coli just
because usually for a various engineering,
one is a highly pathogenic one. Many are there actually.
Because usually for engineering applications,
yes,
people are using K12 strain. So is it, if we introduce after they're getting the in vitro results, is it possible to introduce directly K12 strain into the gut microbiota?
Right. Any modification you can do in gut microbiota, but the thing is by engineering, if you do now, it should be more specific actually. So the gut microbes function as a synergistic way. For example, if you consume antibiotic, like along with if you are targeting antibiotic with only one E. coli, so it is very difficult because the antibiotic we know that it binds on only some 3 to 4 target region. Along with killing E. coli, it will kill a similar E. coli specific like other proteobacterial species also will be killed. But protein engineering or genetic engineering, if any modification done, is it possible to target only that particular species E. coli? For example, I have pointed out that the Clostridium difficile is highly pathogenic, not responsive to antibiotic. So in that way, like targeting specific ways the challenging task. Now either protein engineering or genetic engineering, the recent way the promising results are coming through the page-based therapies are more successful than antibiotics because they are more target oriented. Specifically, they are targeting.
Oh, thank you, sir. Over to Jamuna, madam.
Yes.
Thank you, sir, for the informative talk on the role of the gut microbiota in human health and disease. And the analogies which you gave in the initial stages of your presentation were very interesting. And now we have a lot of knowledge on the various factors that play a role in the diversity as well as abundance of the gut microbiota. Now it's very clear that diet plays a major role in the composition and diversity. And the other concepts that we got to know about were related to the use of gut microbiota metabolites as biomarkers to identify various diseases, and its ability to transform most of the products that we consume. And we also got to know about the application of symbiotics as well as postbiotics. And postbiotics is an evolving concept, and I think the studies have been recently started on that.
Yes.
And so now I would like to ask you the few questions. So the current practice is to administer antibiotics as well as probiotics. So is it really helpful in restoring the gut dysbiosis?
Antibiotics with the probiotics, ma'am?
Yeah, so that is the current practice that the doctors are carrying out. So
yes,
is it?
Actually, I'm not a medical person because but thing is, It is good. Many doctors, even I have myself personally have experience. Some of them they suggest only antibiotic, consume antibiotic, and after curing, after some interval, then you consume probiotic. This is one group of they suggest like that. The other group, it depends on their infection status and condition, and they recommend along with antibiotic, they recommend probiotics and even some prebiotic, like probiotic and prebiotic mixed capsules are there. They are also suggesting actually, but still research is going on actually. But many anyway, we have to balance the microbiota after antibiotic therapy. But they reported that once antibiotic is taken and it is imbalanced, it's very difficult to recover. Even though with the probiotic, it is very difficult to recover the complete loss of the bacteria. Only at some particular level only we can recover and imbalance. Actually, some bacteria loss means loss. Actually, it will take minimum like 6 months. It take to get the bacteria back. Actually, but they reported that some bacteria means the permanently lost with antibiotic. That is not possible to recover. Like because that were colonized during childhood. But it's a better, in my personal opinion, I'm telling it is better practice like to consume antibiotic with a probiotic also. It depends. The doctors will decide whether that is probiotics required or not.
Okay, sir. Thank you. So, and the other question is like, is it better to administer a single species or a consortium of bacteria?
Yeah, it's very good question.
Or any
Actually,
kind of
even yeah. Thank you, mom. Actually, this is for example, like we are consuming single bacteria means lactobacillus tablet, and if we are having any like consortium is better than a single bacteria, but it depends on the function actually. For example, like even commercially in India, they use like either bacillus, that is lactobacillus, or like if you have dysentery, they use bifidobacteria.
bacteria.
Yeah,
Yes,
so
sir.
bifidobacteria contain around some 6 bacterial species. That is like consortium. So now many capsules are coming as a cocktail, like with the mixed bacterial species, giving more potential. Because even our gut bacteria alone, even in human gut bacteria in vivo system also, it's working in a synergistic way. Like if you remove one bacteria, the one bacteria will produce some metabolites. That bacteria metabolites will be used by other fungal or viral species. That viral and fungal species is essential for for digesting some other product. So they depend on each other actually. So if we remove only one bacteria, there is some other bacteria or fungal will be affected. So if it is like particular lactobacillus is very specific to kill some particular pathogen, means we can use only single bacteria. But in general, if you use the mixed one, that consortium of bacterial species will be more beneficial, I think.
And lots of studies have been done on the gut bacteria. But even though the microbiome as well as the virome is prevalent, why is it like more focused on the bacteria? Is it because of the technologies that are focused only on sequencing the bacteria?
Yeah,
Or what could be the other reason?
exactly you pointed out, ma'am, the deficit actually. Yeah, exactly. One is a technology issue, like extracting the metagenomic DNA and doing either shotgun metagenomic sequencing or amplicon sequencing by NGS is easy. But when you go for a viral diversity, like suppose even shotgun metagenomics also, we can identify some DNA virus diversity. If you want to do entire viral diversity, we have to go for transcriptomics actually. So collecting environmental samples and isolating RNAs and doing a transcriptomic analysis, and then finding abundance and diversity based on that cDNA based characterization and the NGS. It will take now they have developed a technology actually, transcriptome and whole exome based studies actually. So many virome diversity papers are coming. That's what one study I have quoted actually, like for the highest number of viral species are present in human gut, they reported actually.
So
Now
which
many
kind of
people
viruses are they?
Actually, the majority of them are bacteria. I have not mentioned anything about the taxonomy and all. I didn't go in detail.
Okay, sir. So I think that's all we have for
Okay,
now.
ma'am. Thank you very much for the nice
Okay,
questions.
thank you, ma'am.
Thank you, sir. So although
Thank
it's
you, sir.
virtual, we would like to
Thank
honor
you.
both of you. First, I would
Thank
like to honor the speaker with the e-certificate and the memento.
Thank you.
Next, I would like to honor our chairperson with the e-certificate and the memento.
Thank you, sir.
Thank you. Thank you all.
Thank you, sir. Thank you, madam. Thank you all for joining. Thank you once again.
Sir, good morning to one and all. Good morning, sir.
Hello, good morning, sir. Am I audible?
Yes, sir. Yes, sir. Yes, sir.
Great.
It's audible, sir.
Okay.
Shall
Thank you, sir.
Shall we start, sir?
Yeah, we can.
So, good morning to one and all. The speaker for the session is Dr. Suresh Ellaraj. Speaker for the session is Dr. Suresh Ellaraj. I welcome the speaker and persons for this session. I would like to introduce the chairperson, Dr. Kumar Sadish Ravi. Sir is an additional professor and head in the Department of Anatomy from Gorakhpur. Sir has completed his MBBS in Muzaffarpur and completed his MD in Jipmar. Sir is Vice President in Anatomical Society of India and Editor-in-Chief in National Journal of Clinical Anatomy. Sir is also editor of Snell's Clinical Anatomy. Next chairperson is Dr. K.C. Shanti Madam, Professor Head from Department of Anatomy, Vinayaga Mission Kripananda Warrior Medical College and Hospital, Salem. Madam has completed her UG in Park Medical College, completed MD in Stanley Medical College. Madam has completed has 15 publications. Now we request the chairpersons to take over the session.
Thank you so much, sir. So, welcome one and all. I'm glad to invite Dr. Suresh Silvaraj for the guest lecture. And before I invite him to deliver the guest lecture, let me have the honor to introduce Dr. Suresh Silvaraj. He is working as assistant professor at Health and Social Science Cluster in Singapore Institute of Technology, and that is located in Singapore itself. He has completed his MBBS degree in Coimbatore from Coimbatore Medical College, and he has done his MD Anatomy from the topmost institute of the country, USA AIMS, New Delhi. He is a member of Institute of Adult Learning, that is Adult Education Professionalization, Singapore, a specialist adult educator. Apart from that, he is a reviewer of international journals. He has received 2 grants for the interactive anatomy learning, virtually engaging students with QR-coded 3D anatomy specimen. Apart from that, he has also received another grant for using immersive learning for developing surface anatomy palpation competency among physiotherapy and occupational therapy students. Next slide, please. Along with the traditional teaching, he is using modern digital tools such as augmented, virtual, and mixed reality, that is AR, VR, and MR tools for anatomy teaching sessions. His current research topics include virtual human anatomy project, developing 3D images from the series of CT scans for developing virtual, augmented, and mixed reality tools for anatomical educations. Creation of VR, AR, and MR models for the plastinated specimens. So with this brief introduction, now I would like to invite Dr. Suresh Shelvaraj to deliver the talk on the topic Modern Technologies Tool for Anatomy Teaching and Learning. Welcome, Dr. Suresh.
Thank you so much for the wonderful introduction, sir. Can you hear me?
Yeah, my pleasure, sir.
Thank you, sir. Thank you so
You
much.
are very much audible.
Okay, sir. Okay, shall I share my screen, sir?
Yeah, please.
Okay. Okay, good morning, one and all. It's my pleasure to being a part of the Basic Medical Sciences Conference conducted by the Arupadavid Medical College. I would like to thank the Arupadavid Medical College and the organizing committee members and the organizing secretary and organizing chairperson for giving me this opportunity to present my the basic bit ideas to all in this conference. Thank you so much. I would like to extend my thanks who has contacted me and organized these things. Okay. With this, I would like to go for my presentation, which is the modern technologies and tools for anatomy teaching and learning. Before going to that, I just want to give by self a kind of advertisement. This is our university that is called Singapore Institute of Technology, which runs various courses. Along with that, we run the allied health sciences. We are the only university in Singapore conducting the allied health science programs, which include therapy, diagnostic radiography, speech and language therapy, dietetics, and all. Okay. So the total size of our student cohort who study anatomy and physiology will be around 460. So all the year 1 students will undergo the anatomy modules. So whatever we developed or we are going to discuss, we are implementing the teaching and learning for those year 1 students, not only the year 1 students, and also we are implementing these 2 other clinical year students as well. We all know that our anatomy is study of internal organs. It is started almost around 200-300 years back with the dissection of dead bodies. Then later, the technology has developed. The dead bodies were preserved with the formalin, which is with the help of embalming. And in India, and not only in India, all around the world, we are using the cadavers as a prime source of anatomy teaching and learning. So in Singapore Institute of Technology, we are also following the basic one. So we are teaching anatomy with the cadavers. So this is the image of our year 4 students, those who are doing the elective dissection module. So they learn anatomy with the by doing the dissection on their own. And this is the image which showing our year 1 students who are attending the anatomy practical. They will be learning the anatomy with the prosected specimens, and which is under the supervision of all our anatomy and anatomy faculties. So apart from the cadavers, we have a variety of tools to teach the anatomy, that is including the plastic models. So in the self-learning, the students will use this plastic model to revise the anatomy, whichever they learned during the anatomy practical. And apart from the plastic models, we have the number of plastinated specimens. The plastinated specimens are the well-dissected specimens, which are commercially available one. And that will give the clear-cut anatomy and the relationship to the particular structures. So our students will be using the plastinated specimens during their self-studies time, and also they will be using this one during their tutorials. And before the exam, if they want to revise something, they will be using it. Okay, so apart from that, we have some electronic tools also to learn the anatomy. But the problem is the plastinated specimens will give some kind of advantage. They're well dissected and clear anatomical structures and relations, and there is a possibility of peer discussion among the students, and the structures are well clear and preserved. If we go with the dissected specimens, some structures may be damaged during the dissection, or some may not be clearly identifiable. The relations may be altered, but all the things are preserved in plastinated specimens. And the students' acceptance rate using the plastinated specimen teaching is more because it is not under the formalin. So there is no problem with the formalin smell and handling with the bare hands, and they no need to visit on particular time to the anatomy dissection hall. Here the plastinated specimen kept in the anatomy museum, they can use at any time during the official hours. So the students' acceptance rate is slightly higher comparing to the cadavers. But the problem of using the plastinated specimen is, again, the students has to visit the lab, the anatomy museum and the lab, and they need to get a permission from the lab, the manager, to visit the lab. And especially on weekends, it is difficult for them to use the lab. And during the COVID time, the students are unable to come over the campus, because in Singapore they maintain the restrictions very much. So there are no students allowed inside the lab during the COVID restriction. And these are expensive models. If you get the commercial one, which are expensive, and the number of specimens available are limited. We have 2 sets of whole body with the limbs and the organs, but still our cohort size is 460. So all the students, if they want to use it, they need to wait for the turn. So it will be difficult for them to access it. And the problem is damaging. So if the students are using repeatedly, all the 460 students, there is a possibility of damage. Still, in some of our, we can fix it, but still some of the superficial vessels and nerves are cut off because of the repeated handling, and sometimes the students may not use it properly. The maul handling also damages the structures as well as the small tendons and all. That's what I have mentioned, that restricted usage during the lab closure, during the public holidays and the weekends, and also the time factor. The students has to come to the university to access the specimens. And in the first few weeks of year 1, the students having some inhibition in handling the cadaveric specimens. And because the cohort size is big, so they have to finish their self-study within the stipulated time. So they may not use the specimens for as long as they want, because they have to hand over to the next group of students. So they have the limitation in the timing also. So because of these limitations for using the plastinated and the plastic models, and also the limitation in accessing the anatomy dissection hall to access the cadaveric specimens, we thought of creating some new tools. So that's why we came up with the idea of creating the new digital tools that will be helpful for the students. So we decided to create tools which is self-accessible and also remotely accessible, and which have the more impact comparing to the other tools available. So we decided to create the 3D models using the plastinated specimen, and also we created some 3D models using the CT scan slices, and if we want to give the real life experience of lab, so we created the 360 videos. So whatever the technology we discuss, we follow in our SIT. Most of the technologies are well accepted by the students, and even after crossing the year 1 also, they are still using in year 2, year 3 during their clinical rotations. Okay, I'll start with the first one. Means apart from the plastinated models, so we have the anatomage, and also we have the VR lab to study the anatomy also. So we are the only institute in Singapore having the 2 anatomage tables. So the anatomage also well used by our students, especially learning the cross-sectional anatomy and all, because we have the diagnostic radiographers. So it is well used by the diagnostic radiographer, and also the physiotherapy students, they want to study the musculoskeletal the anatomy and all, they will be using it very well. So aim of our study to expedite and integrate the modern technology to engage the students actively in their self-learning. That is the prime aim of our study. So to motivate them to study on their own. So by means of creating the 3D models using the different plastinated specimens and mobile applications. So we created the 3D models of the plastinated specimen by means of the 3 levels. The first level is capturing the images. The second level is processing and refining. And third level is supposed to be the labeling. And finally, we have given to the students self-learning. So the capturing, actually we use the high-end cameras to capture, and we tried with the photogrammetry software to stitch the images, but there are some details are missing while using the photogrammetry software. So we were trying to find out some other solutions. So we came up with a simple solution with that the mobile phones which are using the sensor called LiDAR sensor. So that will identify the image which is located in the space using the gyroscope. So we used the iPhone's LiDAR sensor using the Polycam app to capture the images. So once we switch on the camera through the Polycam app, we go around the model, and the app will automatically snap the photos in an overlapping manner. And there is no further need of manual adjustment and the framing or stitching using the photogrammetry tools. So this will the product will be the 3D model. So it will be taking maximum 30 to 40 minutes to capture the entire bigger specimen, the life-size specimen. So after that, there are some algorithms which are present in the app itself, which will find the markers on each photo to match with each other. So it's basically the AI one. Nowadays, the LiDAR sensor and Polycam app is usable in even in the not only in the iPhone, also we can use it in the Android phones itself. So we created the 3D models using the Polycam app. Later, the 3D models had some the background kind of artifacts and some unwanted background images as well. So if you want to remove those, we use the free sourcing app. We can use it to remove the background, and also we polish the images. Once we come up with the 3D model, we need to label that. So for labeling, we use the Sketchfab. The Sketchfab is kind of the commercial app. So we place all our 3D models in Sketchfab, and the Sketchfab itself giving the opportunity to label the structures. So we label the structures, and we get a commercial license by paying the premium account. So it is still there. It is accessible only with the links and the QR codes. So only the students of SIT can use it. Otherwise, whoever having the link or QR codes, they can access it. So this is our 3D model, which we created with the Polycam app and also the Blender. So this is the for life-size specimen. This much clarity will come up with the end product. You can see the numbers. Those numbers are the labels. This is the screen record of accessing the 3D models. So from top to bottom, if you use this technique to capture almost all the vital structures, and also it will maintain the relations. The clarity also good comparing to the using the 3D scanners. So our prime aim is only the back and the upper limb. So we focused on this area. And with the labels, we can access it, and we can so we can make the labels, and also we can give the descriptions also in this. So this is 360 rotatable. I'll show you the image which I have created, which I kept in the 3D model. So these are all the number of models we have created. It will take some time to load because we are connected with the Zoom. There may be some lag. Yeah. Yes. So it is rotatable now. So we can rotate in any direction. And the clarity also, it's having the capture in the proper lighting, will get a good clarity. And the clarity also depends upon the camera which we are using in mobile phone. But still, nowadays the recent mobile phones having the good pixel rate camera. So I don't think there is a problem with the pixels and the clarity wise. So this is the final end product of what we created. So the students are using these models instead of coming to the plastinated lab. They're using these models to find out this. And we give the unlabeled structures also for them to study on their own. And we have given some self-learning quizzes also. So the students will be answering the self-learning quizzes using the 3D models as well. So by this way, these 3D models of plastinated specimens are helpful for the students to do their self-learning and also attending their assessment and revising the contents of anatomy before their exam. Come back to the slides. Okay, so this is the process of this project. So this is the project which is funded by the SIT internal grant. Actually, we use this one, the Polycam app, to capture the images. Then we transfer it into the web app. Then we stitch together. Later, we labeled them and it's placed in the Sketchfab. Then we given the QR code for each specimen. So the QR code is circulated to the students for their study, for the pre and post exposure study, for the comparison. So the before and after exposing the 3D models, the students have given their the quiz. We compared the pre and post exposure quiz results as well. So this is the number of the students those who are using the 3D models. We have given only to the particular group of students, not to the entire cohort as the preliminary study. Then we evaluated the usage of 3D plastinated specimens. So we 219 students were participated in the study, which is in the the academic year of 2022 last year. The participants are given the access to the QR coded anatomical photographs. Later, students were given the post-exposure questionnaire and also the qualitative feedback. Okay. So these are all the results. In the pre-exposure, before exposing to the 3D models, almost 83.1% of the students answered that they have some difficulty in accessing the plastinated models because in year one, the students are from the primary school, the secondary schools, or they need some time to adapt to the university environment. So during that time, they didn't get a time to visit the anatomy lab to use the plastinated specimen. Instead of using that, majority of the students rely on the lecture notes or anatomical website and the written materials as a main source of learning tools. Then 100% of the participants agreed that online 3D interactive tools of plastinated specimens, allowing them for accessing the 3D models in a multidimensional manner. Later, in the post-exposure study, almost 70% of the participants used the 3D models incorporated with the QR coded images, and 52.5% of the students finding that it helped them to deepen their anatomical knowledge, and 42.5% of the students were neutral, but the remaining 5% students tell that this is not helping them much, and 75% of the students responded that they would like to continue the 3D models even during their clinical years. So as a whole, the QR coded 3D images given the positive response in enhancing the learning anatomy experience of allied health science students. Not only the pre and post exposure, and also the qualitative study we used. In that qualitative study, almost 57% of the students rated our model between 7 to 9 in the Likert scale, and 35% of the students rated between 4 to 6. Around 9% of the students rated between 1 to 3. So that means majority of the students are impressed with the 3D models. So that's about the 3D models. Now we have our other lab. It's called Virtual Human Anatomy Lab. So in VR lab, we created the 3D models to give the immersive learning experience to the students, because the modern generation students or the X generation students, they are having the tendency to explore the study the topics as a game or something, the funful one. So they're very much happy to access the 3D models in the virtual environment. So whatever the QR coded specimens, the 3D plastinated specimens, actually these are enabled with the VR one, so they can use they can study that one with the VR environment also. But we wanted to create the artistic model using the CT scans. So we have landed up in this project. This is also the granted project by the Singapore Higher Education Department. So we use the CT scans of the real patients from our clinical partners from the various hospitals in Singapore, and we use the software to segment to create the 3D models of the CT scan. We use the variety of software, including the OCRX, depending upon the operating system we use. Then the layers are constructed together with the 3D model using the application. After that, the VR model introduced into we converted the 3D model into the VR environment. So the section added into this. Along with that, we use the cursor in X and Y direction to see the cross-sectional images also of the CT, mentioned the different cross-sectional images that will be helpful. CT scan and the MRI and all. So this is the final end product of one organ we created, that is the heart we created as a part of the project. This is the single organ we created one. So our students were, we have around 10 headsets. So this is our VR lab. This is the kind of temporary lab. We will be converting this one into the highly sophisticated one in the new campus. So the students were given almost at a time, almost around 6 students can access this VR tools. This is a various group of students attending it in the sitting posture. and the study will be conducted in the supervised environment to avoid the students to getting dizziness or vertigo because of the first time users. They may experience some side effects. To avoid that, it is conducted in the supervised manner. Once the students are exposed to the VR environment for a particular time, for example, the minimum 30 to 45 minutes, after that, the students are allowed to explore the 3D models in VR environment on their own. So the students need to be completed the induction session using the VR. After the induction session, the students are allowed to use on their own in the VR lab. So this is our the screen recording under the VR environment, how the students can access it. So this is the heart, and which is labeled with the hot spots. Student means if you trigger the hot spots, it will show you the labeling of the each heart. So the students can go around the heart, or if you hold it, they can rotate around the heart. And within this environment, we have variety of tools to access the 3D model. So we have the cardiac cycle one, and also we have the CT scans, and also we have circulatory system. So if they click it, each will appear on their own. Yeah, if you want to rotate around the heart, we can in the standing position. Otherwise, if you want to rotate the heart in the sitting position, if you want to use the VR, we can rotate using this key. Then we can say visualize the posterior or superior or inferior aspect, whichever way you want. And apart from that, yeah, we can correlate this one with the actual specimen which we have in our museum. So this is the actual specimen which is available in our museum. So they can compare the 3D reconstructed model with the actual specimen. So which also be placed in the VR environment. So the students can compare that one. And this is the video which showing the cardiac cycle. So once you click the cardiac cycle, which will show you the contracting heart with the different the phases of cardiac cycle, how which chamber will be working on it. So that is also incorporated with that. So the contracting heart also rotatable. And this is the cross-sectional, so the sagittal coronal section, which is showing the interior of the heart for the blood flow. And apart from that, we have the CT scan sections. Once you click the CT scan, just a moment. Yeah, once you click the CT scan, then we can move the cursor to see the different orientation in the chest. So this is the axial, coronal, and the sagittal. All the three planes are here. Depending upon which cursor you are moving, which show you the CT scan images of the chest. It's going towards up and coming down. At the same time, the cursor will move in all the X, Y, Z, all the three axes. So it will be helpful for our radiography students. So this is an example of cardiovascular system alone. Likewise, we created with the digestive system and other system as well. So within the same single application, the students can learn the 3D anatomy and the physiological concepts, and also the imaging techniques, everything. So we have the videos we incorporated. So the recorded videos related to the cardiovascular system. So once you click it, the video will run to explain the concepts of anatomy and all. So everything will be happening within the virtual environment. The students were still wearing their the VR glasses. So this is showing the cardiovascular system. Actually, this is not our model. This is the commercial one we purchased and we placed in the VR environment. That's it. Okay, so that is our VR study. So once we given this one to the students, we find the means we collected the survey anonymously. Almost the 40 out of that, 3% of the students never used the VR. So 57% of the students used it, and 57 thought it was somewhat easy to use, but 21% of the students they have some difficulty in using the first or second attempt. And 50% of the students use that this is the learning experience was expected, but the 36% of the students were expected. Means this the experience was more than they expected, but 14% of the students stated that this is less than what they expected. The majority of the students find that this is useful for their anatomy study. They will be they feel quite well. That means they never feel any kind of dizziness or vertigo. So overall, 57% of the students enjoyed the learning experience of using the virtual human anatomy. So 86% of the students would like to learn about other system also, but only 14% students they said they may or may not use for the further systems. So this is our second one. So we still want working on further to progress for the making this one AR enabled content. Using they no need to use the headset. Instead of headset, they can use the handphones to study the contents in the AR environment. So we are working on it. Then our third content is the creation of 360 videos. The 360 videos which give the students to get a sense of being in their lab. So we created the 360 video of the entire anatomy lab. So which is actually the entire anatomy lab is captured in using the 360 camera. It's called Insta360. So all the 360 capturings are transferred to the software with using the AI to merge all the 360 images together. So we use the commercial platforms. This is our one of the practical session. We use this 3D model. Actually, this is the entire anatomy hall. You can come from exit also. This is our entry point of the anatomy hall. So this will give you the 360 view of the entire anatomy hall. So the students would like to visit whichever the cadaver they want. They can visit this, and they can go around the cadaver and they can visualize the structures. And if they want to zoom in, they can zoom in to find out the relations and structures like this. This is our musculoskeletal system practical. So we given the projected specimen of upper limb. So like this way, we can create the 360 environment for the students, so they can explore or they can access the anatomy lab without coming to the campus, and also they can access the specimens. So by this way, the students are getting the experience of being in the lab, and also if they miss something during their practical sessions, they can go through that, and it will give their immersive learning experience. So the students also happy with it. We have not conducted the study until now. We just created the prototype, and we got just given for the just prototype modeling for the one group of students to get their opinion. Most of the students felt that it will be useful for the revision during the practical and before the practical exams as well. And apart from that, we use the commercially available softwares also for teaching. There are a lot of commercially available softwares, but we use the visible body for the students to learn. We have the course, so the license will be given to the students so they can access the 3D anatomy application to access the virtual dissection tools, so that will be useful for them. And also there are some quizzes also incorporated for their self-practice. Most students feels that visible body also one of the useful tool for their anatomy revision. And actually we have enough number of license to cover until the students from year 4. So all the students of our SIT from in allied health science, they have access to the anatomy atlas, so they can access it even during their clinical years. And apart from that, we did means we are conducting not only the year 1, and for year 4 students also, we are doing the anatomy module that is called elective dissection. So many of the year 4 students almost completed their the clinical rotations during the year 2 and year 3, and also the first phase of year 4. So in the second phase, we give this is the elective course. So the elective in the elective course, we given the limited number of opportunities. Only 30 final year students, 20 from physiotherapy and 10 from diagnostic radiography, given this opportunity to take part in the elective dissection. So we allotted 10. So they did their own dissection, but they correlated the anatomy which they're learning with the clinical cases they have seen during their clinical rotation. The clinical rotation also simultaneously going along with the elective courses. So whichever the case they're studying in clinical rotation, they will come and compare with the elective dissection. So almost around we did a pre and post test, pre and post exposure. Pre-exposure, the class average was 58 in the anatomy, because once they go to the final year, many students forget the anatomy and the basics. So before the exposure to the elective dissection course, the class average in the pre-exposure quiz was 58%, but post-exposure was 89%. So total students scored 60% is the pre-module is 43%, and the post-module is almost around 96%. So almost 88% of the students strongly agreed that module helped them to correlate the clinical condition with the anatomy, and 96% of the students agreed for professional, and not only the anatomy learning, and also these cadavers are helpful for them to learn the professional and ethical values, and 84% of the students agreed that session will be helpful for their clinical practice in future. So this is also one of the way we do the vertical integration, not only in year 1 and also doing in year 4 for the giving the anatomical as well as the clinical knowledge. So in this year 4, they mainly learn most of the clinical anatomy. They will correlate the clinical contents with the anatomy by doing the dissection. So this is the perception of one of our radiographers student. So yes, with that, I conclude that the help of modern mobile applications and the gadgets, we can successfully create the 3D anatomical models. The 3D anatomical models of plastinated specimens, and also the CT scans, and also the 360 videos are helpful for the medical, allied health science students, and also other health science students, those who are learning anatomy for remote learning. With this, I conclude my presentation. Thank you so much for the patience. If you have any questions, please feel free to ask me in the system.
Thank you. Your section on modern technological tools for anatomy teaching and learning.
Thank you.
They explained very well regarding how the 3D models can be created and how can it be technology utilized to give 3D image for the students for better learning. And your virtual lab concept is also fine. Now the forum is open for discussion. I request Kumar sir, if you have any queries, you can ask sir. And I request participants to post questions in the chat box.
Wonderful presentation, Dr. Suresh. I must congratulate. And I was thoroughly impressed to see your labs, right? It's so rich infrastructure you have.
Thank
right?
you, sir.
Your VR lab and everything means what I've observed that before this pandemic, when we see, right, we were not using all these technologies, right?
Yes.
We were not fond of using technologies, especially when we talk about the teaching of anatomy, right? In that, we were solely basically dependent on the cadaveric dissection and all. But this, when we talk about this COVID-19 pandemic, it has changed a lot, whether it is the teaching methodologies or use of technologies, right? So this pandemic has seen a turmoil, right? And the problem
Actually,
is
in our SIT, we started the VHAB project before the COVID, meaning we started our project in 2020, not even 2017, sir. 2017 we started. Our prototype was ready by 2019. So we were prepared for that. I don't know, accidentally the COVID came, so we had a chance to explore it more.
yeah, that's what we can see, that it must not be, means, as we know that Rome was not built in a day. So
Yeah.
the infrastructure what we have seen, it must not be of 1 or 2 years. It means you had the vision and you people have planned accordingly, right? But when we see in the recent days, the anatomy teachers, you say the across globe, right? They have started using these technologies. Either you say AR, VR, MR, you say the anatomies was not common in India. When I was working in San Lucia, we were using this 3D virtual technologies, right? But in India, this anatomies only came, and then we have SECTRA, and also even now this National Medical Commission has given the guidelines. They have
To use
recommended
the anatomy.
to have this anatomies or the SECTRA. So I really appreciate the way you presented, and we came to know that what all the things you are using in teaching this anatomy, right? So thanks a lot. If the questions are there from the delegates, we will welcome.
Thank you, sir. Participants, kindly post your queries in the chat box.
So if there are no questions,
I think one question.
Dr. Sangeeta
There is
has
one
asked.
question for you, sir. Does this particular VR lab facility for assessment?
Actually, we are not using this VR lab facility for assessment until now, but we have a plans to implement. We in short while we are going to the new campus, so we have the sophisticated VR AR lab, the bigger lab. So once we move there, we will be conducting the assessment in the VR lab. Yes, of course. But that current lab is enabling only 10 students at a time. But our future VR lab will be almost around 50 to 75 students. The maximum capacity will be 75 students. So once we have that, then we will be assessing VR also.
Thank you for your response, sir. Thank you, Dr. Sangeeta, for posting the question. Any other questions are welcome.
Good morning, sir. Dr.
Good morning, sir.
Good morning.
Congratulations, sir. Good morning, Ravi, sir. Morning,
Thank you,
Shanti, ma'am.
Morning,
It was
sir.
a wonderful presentation, sir. We do also have the VR lab. Very soon we'll communicate to you. We sought your help to establish further, sir.
Happy to collaborate with you all, sir. Thank you, sir.
Thank you. Thank you. Nice presentation, sir.
Thanks a lot. Thank you, sir.
So hence actually not a single one size will fits for all. So learning anatomy is really a tough task, but it is very interesting topic. We all agree, I believe. So augmented teaching learning methodologies with an integrated teaching tools, multiple tools definitely gives a good output, and the students' learning will be better. And it is really interesting to learn from you how to create a 3D models and how we can incorporate it in the institution. Even though the Google and the YouTube videos are available, if it is from our institution, from our own patients, definitely it will have an impact on our students, I believe. Any other queries, sir? Sir, I have only one query, sir. In this
Yes,
360 videos, you showed that lab there. It is taking the students to their lab, dissection lab. Is there any facility that you can add demonstrations, like when any discussion topics can be taught there, or spotters can be taught there, and it can be displayed when they visit virtually the lab?
Okay, ma'am. Actually, this is 360. It's kind of a photograph, ma'am. So we cannot add any videos in that. But we have another content means we created the 3D 360 video for the surface anatomy palpation. So in that both student and the means we do the surface anatomy with the real students. So in that both the students can be seen in the 360 video. Yes, video. It is possible, ma'am. So the demonstration also possible. But the thing is, the bone or maybe the organ supposed to be exactly in front of the instructor. Meaning both are supposed to be like that. But in our anatomy lab, the what is that called? The specimens are in the horizontal manner, but the instructor like this. So the 360 camera only focus in front and back, both sides. If you go like this, the distal part maybe it's having the blurring effect. So need to be vertical. If it is vertical, definitely it will be helpful, ma'am.
Okay, okay, it is better. Thank you, sir. How far? What is the outcome of this anatomy, sir? 3D dissector. When the students started using it, do they are comfortable than the cadaveric specimens?
Definitely, ma'am. They're more comfortable because they can access remotely. They can access on their own face. But if they come to the cadaveric one, the duration is limited. So within the limited duration, they have to complete all the practical. They have to complete all the structures. For example, if we go for the muscles of upper limb, there are a lot of muscles. They cannot finish all the attachments, the blood supply, nerve supply on particular timing. But once they go to the 3D models, it will be helpful for them to revise and get the concepts as well.
I would like to add over here, Shanti ma'am.
Yes, sir.
At AIIMS case, we had 3 anatomies and 2 sectra. Right now, even at AIIMS Gorakhpur, we have anatomies. So definitely, this it means cadaveric dissection cannot be replaced, but this anatomy or the sectra, what we say, right, it is definitely useful because not only the dissection part we see in the anatomies, but even the histology, histopathological cases, or the you say the CT scan, MRI, all these we can incorporate, right? And then we can relate. Even the surgeons, they can plan their surgeries, they can see their CT scan, MRI, and then accordingly they can plan. So definitely it is useful. But as I said that there is no substitute, means cadaveric dissection should not be replaced, but these technologies definitely can be used, right?
Definitely, technologies will be augmenting the learning process, sir.
Yes, yes, exactly.
For our elective dissection course, the students will be using the anatomists to do the virtual dissection before going for the actual dissection. For example, if they're dissecting the upper limb tomorrow, they will be doing the virtual dissection of upper limb with the anatomists in the previous day. So they will get some prior idea what they have to see once they go deeper to the particular structure. So it will be helpful for them to avoid the damage of the particular structure. But like Satish sir said, it is the cadavers are the prime. No other tools will replace the.
Right.
Yes, sir. And one thing in your virtual lab slides, I have seen that it is easy for the student to compare the cross-sectional anatomy also, because cross-sectional anatomy, they cannot have a visualization, but this CT scans 3D models definitely will make them to have a better understanding. Okay, I think there is no other questions in the chat box. Participants, anyone else want to ask any queries? Kindly get clarified.
If anyone would like to collaborate with the Singapore Institute of Technology, I am open for the collaboration and happy to collaborate with you all. Thank you so much.
That's great. That's great.
Thank you, sir. I'm from Salem Vinayaka Missions Kripananda Warrior Medical College. We'll have in touch with you, sir, following the footpath of AVMC.
Thank you, ma'am.
Okay, then.
Thank you.
So we can end the session, ma'am, if there are no further questions. Right.
Yes, sir.
So thank you so much, Dr. Suresh sir and Dr. Shanti ma'am. We had a great session. Thanks a lot. And thanks to the organizers also. We are grateful for inviting us to chair the session. Thank you so much, sir.
Thank you, sir.
Thank you all.
I would like to congratulate the speaker, and as well as I thank the organizing committee for giving us an opportunity. Thank you all.
Thank you, Dr. Suresh sir. You have enlightened the interactive anatomy using various models. Thank you so much.
Thank you, sir.
Thank you, Dr. Ravi sir and Dr. Shanti madam for chairing the session. So now we would like to honor our speaker, Dr. Suresh Selvaraj, with a certificate and memento as token of appreciation.
Thank you very much, sir.
Thank you, sir. Welcome. So we would like to honor our speaker, Dr. Suresh Kumar Sadish Ravi, with certificate and memento. We would like to honor our chairperson, Dr. Shanti Madam, with certificate and memento as token of appreciation. Thank you so much, sir. Now we'll end the session and go to the next session. Thank you, sir. So, dear delegates, we have 10 minutes break. We'll resume after 10 minutes. Thank you.
Welcome to Vinayaka Missions Group, where education meets healthcare excellence. Our academic landscape spans across 13 colleges and 11 schools, situated in 4 vibrant campuses. We are proud to serve the Union Territory of Puducherry and extend our reach to Salem and Chengalpet districts in Tamil Nadu. Pioneers in private medical education. We established the first private medical college in Puducherry back in 2000. Today, we operate 2 medical colleges in Puducherry, where we provide healthcare services to an astonishing 4.5 lakh outpatients and over 50,000 inpatients annually. We manage 9 rural health centers, where we attend to approximately 1,000 patients every day, ensuring quality healthcare for all. And now, as we embark on our next level in healthcare, we are proud to introduce the Vinayaka Karkinos Oncology Institute, or VCOI, in collaboration with Karkinos Healthcare. Karkinos Healthcare is a purpose-driven, technology-led oncology platform, revolutionizing cancer care in India, with a primary focus on early detection, precise diagnosis, patient care, and groundbreaking research. Through setting up of the Vinayaka Karkinos Oncology Institute, the aim is to bring comprehensive cancer care closer to the communities under the distributed cancer care network. Our mission: early detection programs through community screening programs, identifying high-risk patients and guiding them throughout their entire care journey. The facility will be equipped with state-of-the-art diagnosis and treatment options. And as part of our commitment to the people of Puducherry, Vinayaka Karkinos Oncology Institute aims to screen nearly 1 lakh population within the next few months. This collaboration between Karkinos Healthcare and Vinayaka Missions Group, including Vinayaka Missions Research Foundation, in setting up of the Vinayaka Karkinos Oncology Institute, VCOI in Puducherry and Salem, shall be a testament where education, research, and affordable and accessible cancer care come together for a brighter, healthier future.
So good morning, sir. We'll give introduction and then you can share the slides, sir.
Okay, should I stop now?
Yes, sir. Thank you. Good morning, everyone. Welcome for the fourth talk for today's day 2 of our BMSeCON. Today's topic is acute threat of aluminum exposure, a multipotent risk factor, by our eminent speaker, Dr. Prasoon Nayak. For this talk, we have two chairpersons, Dr. Sharada Ningtojam, Professor and Head, Department of Physiology, RIMS, Imphal, Manipur, and Dr. Vivek Kumar Sharma, Professor and Head, Department of Physiology, AIMS, Rajkot. Now I invite Mrs. Hemalata to invite and introduce our chairperson, Dr. Sharada Ningtojam.
Very good morning. Myself, Hemlata, Assistant Professor, Department of Physiology. It's my great pleasure to introduce chairperson for today's session, Dr. Sharada Ninjotham, Professor and Head, Department of Physiology, Regional Institute of Medical Science, Imphal, Manipur. She's completed her MBBS and MD Physiology from RIMS 2002. She's an excellent teacher with 21 years of teaching experience, more than 40 national and international publications in her credit. She is also a member in Ethics Committee Board at Manipal University. She is a member of Academic Standing Committee, Medical Education Unit, and Head in Curricular Committee in the RIMS. She is installed in an in charge in Skill Lab RIMS. Welcome you, madam.
We feel privileged to invite our next chairperson, Dr. Vivek Kumar Sharma. Sir has done his MBBS, MD Physiology, and has many additional degrees to his credit: MD Alternative Medicine, MSc Yoga Therapy, and PG Diploma in Yoga Therapy, and PG Diploma in Health and Lifestyle Management. Sir is currently working as Professor and Head, Department of Physiology, AIMS, Rajkot. He is an active member of Medical Council of India, Indian Federation of Sports Medicine, Association of Biomedical Scientists, Association of Physiologists and Pharmacologists of India, and Research Society for the Study of Diabetes. He has published more than 50 publications in reputed national and international journals, and he has many awards to his credit. Some of them have been mentioned here. Gold Medal in Alternative Medicine and Health Excellence Award in January 2017 by Indian Board of Alternative Medicine, Kolkata, India, and Professor R.C. Shukla Oration Award in November 2018 for Best Cardiovascular Research by APICON 2018, and Best National Teacher Award in Physiology awarded during International Conference by Basic and Medical Sciences and Appellate Physiology in 2020. And he has been invited as a keynote speaker on Mind-Body Medicine in North California, USA 2019. He has received many prizes to its presentation. Some of them have been to his credit, like effect of 6 months of structural and physical activity of perceived stress, executive functions, and reaction time in adolescence. And his main areas of interest are lifestyle modification disorders, exercise physiology, and neurophysiology. We welcome you, sir. Now I invite Dr. Vivek Kumar Sharma to introduce our speaker, Dr. Prasoon Nayak.
Namaskar, Dr. Prasoon Nayak, and to everyone, all the audience over here. Am I audible, Dr.
Yes,
Gopinath?
Yes, sir, you are audible, sir.
So let me please introduce a very good friend of mine, Dr. Prasoon Priya Nayak, who is a professor. Dr. Prasoon Priya has completed his MSc and PhD in medical physiology with first class from University of Calcutta in 91 and 99. Currently, he is working as a professor of physiology as told in AIMS Jodhpur. He has got 58 publications to his credit, and he is an executive editor of Indian Journal of Physiology and Allied Sciences. He is a life member of many learned bodies like IBIA, IBRO, ISCA, SOP, PSI, and APPI, and he is a member of editorial board of for international journals like Frontiers of Physiology, and reviewer of reputed journals like Frontiers of Neurosciences, Human and Experimental Toxicology, Apart from that, he has got many awards and many credits. And to name a few, he got recently the Professor S.B. Deshpande Award of APPI for the best work in neurophysiology. And after that, he has got awarded for a Professor B.B. Sarkar International Oration Award. And he has got many other awards like Young Scientist Fellowship, 16th International Union of Biochemistry and Molecular Biology, BMG, and apart from all these things, the things which you can see is that he is a very learned person and a very good speaker also. And I'm sure that we all are going to learn and we are going to enjoy his talk also. Thank you very much, Dr. Prasoon. Welcome.
Should I start?
Yes, sir. Yes, sir. Yes, please, sir.
Thank you very much, sir. It's really a pleasure to get introduced by Professor Sharma. I know him very well and he is very, I really like him. Thank you very much, sir. It's really a big surprise and big pleasure for me you are kind enough to to introduce me. And I also convey my sincere thanks to the organizing committee for giving me this opportunity to talk my presentation in such a huge gathering. It's a huge one and it's a August gathering. And first time I'm meeting Dr. Madam. Namaskar, Madam. So,
good morning.
Maybe we are listening lot of presentations. This one yeah, my slides are visible, I believe. Yeah.
Yes, they are visible, sir. Please carry on.
So, good morning, everyone. And it's a very pleasure and happy occasion to talk to you. And I really congratulate the organizing committee for arranging such a huge gathering and a huge presentation. It's a nice presentation since morning, and yesterday also it was very good, quite good lot of presentations. Today, I'm going to talk about the aluminum toxicity. I am trying to emphasize the acute threat of aluminum exposure, a multipotent risk factor. Aluminum is a multipotent risk factor, and all of us, we know about it. As I can see, like yesterday, the second lecture, Dr. Sucheta, she has mentioned about aluminum and its possible role as an endocrine disruptor in PCOD. And I think they have recently carried out a study in Bangalore where they have found that the aluminum level in PCOD patient from Bangalore, it's quite high. Then Dr. Sarkar, in his keynote address, he has mentioned about the inflammation, importance of inflammation, generalized inflammation in the neurodegeneration. Actually, aluminum is a known factor, known neurotoxin throughout. It's a long, it's actually even in pre-scientific era, these salts are being used as a stoppage of bleeding, astringent. The aluminum is present in everywhere. In the earth crust, it is the highest metal which is present in earth crust, and in element wise it is third one, like oxygen, silicon, and then aluminum. Aluminum is not a heavy metal. Today morning also we had a discussion about COPD, and there also they have cadmium they were discussing. I think Dr. Pooja was discussing about cadmium, and then during the discussion process they brought in that aluminum's role. Aluminum is everywhere almost. We cannot avoid exposure to aluminum, and it is nothing new that aluminum is having a neurotoxic effect in long-term exposure. It's years of exposure can lead to neurodegenerative diseases, and it has been linked to almost all types of neurodegenerative diseases, starting from highly acclaimed Alzheimer's disease, Parkinson's disease, Guam's dementia, so many dementia-related neurodegenerative any diseases. There is a chance that aluminum might have some role there. But as I have highlighted, the highly reactive metal, it's not heavy metal as I have already mentioned. Still, its toxicity is so much that it is considered as a heavy metal in terms of toxicity. And because of its high reactivity, it is never found alone in the nature. It is always combined with some element it will combine. And because of this high reactivity with other elements, the biological system is not cannot metal, and there are some other physicochemical properties of aluminum which make it that the biological system, the evolution has bypassed this element, never used this element in its life. Nowhere in the life process, nowhere it is being used, and it is considered as biologically inert. And this concept, biological inertness, and there are plenty of physicochemical properties which is helping it to be used in our daily life, made it so useful to our life, to our daily uses, that we are literally living in age of aluminum. Even though it was named long back, maybe a quarter century ago, still the age of aluminum persists. Concept is that because it is minimally biologically available or biologically inert, so it should not be toxic. But what we understood, that because of some opportunistic chances for some other metal, aluminum enter into our body and slowly accumulate for lifelong. It's years after years it started accumulating, and in long term it's producing its effect, the toxicity. With the growing graying, that means as our life expectancy is increasing, because of our health services, we are doing very good in health services. Medicine is so improved nowadays with so many facilities. So our life expectancy is increased, and so the aluminum availability for the life process is also increased. And with aging, when the blood-brain barrier is slowly losing its integrity to some extent, aluminum can enter into the nervous system and can cause toxicity. But the high economic impact of aluminum, commercially this metal is so valuable. You can, there is an institute, International Aluminum Institute. They have 2020, they have reported that there is around 76% recyclability of aluminum. 76%, aluminum is being recycled continuously. Global level, I'm saying, some nations even doing it 94%, 96%. Some are doing relatively less developed countries. They are doing around 38 to 40% of recyclation of aluminium. Still, till now, we have utilized, we have extracted billions of tons of aluminium, and we have put it in the environment so that we made our environment so bad, so infiltrated with aluminium. Coming to the redox inactive, all the neurodegenerative changes, it is once said that if there is aluminium, there is oxidative stress. And I have raised this question in 2009-10, that if aluminium can produce oxidative stress, it must be redox active. But chemically, we know aluminium is redox inactive. Then how it can produce oxidative stress in our body? Then we found in 2014-15, we have established that aluminium itself is not an oxidant, but it promotes oxidation. If there is any oxidative threat, it will encourage that oxidative threat and give more and more. So aluminium itself may not be redox active, but it promotes redox activity. This is very, very peculiar. Not being active, promoting others to be active in terms of oxidative stress. If we see the research, PubMed search, what is mostly accepted search in our medical field, PubMed search on aluminium, it shows that it is around 1945 it started, and then last 2-3 years, as you can see, there is a declining stage on aluminium study. If we see the aluminium toxicity study, again, we can see a declining. So slowly we are accepting that, okay, aluminium is toxic and we have nothing to do. As a scientist community, research community, we are accepting its toxicity. As you can see, even in India, the toxicity study related to aluminium toxicity studies are declining. This made a problem that how we can fight with aluminium. Is it that we don't need to fight with aluminium? Is it so biologically inert? It is only with the long term. Let us see how things are in our surroundings. You can see toothpaste, antiperspirant, deodorant, crystal alarm, talcum powder, sun protection lotion, all of them, they are having aluminium. Even if you see some drugs, all these, in India, it is not so strict to mention about the drug content, but international standard, it is aluminium is mentioned so many places. You see including cosmetics, you can see, I'm not highlighting this cosmetic part, but what I want to highlight is that presence of aluminium in all these drug packs. You can see all of them, they have some or other compound of aluminium in our personal hygiene, including cosmetics. Our drinks, our food, our packed food, our fast food, I think this is a very common picture. The fast foods packed in aluminium, served with hot packed processed food, packed, served with pickles, served with some types of soury sambar, rasam, something like that, chutney, achars, these things. So we are actually taking lot of aluminium. In tea and coffee, they are aluminium. Packed bread, flour, cake, cheese, sauce, fruit juice, drinks, packed, sealed packed drinks or canned juice, canned drinks, all of them, they are either naturally having tea and coffee, they will be naturally having sometimes it is being added as preservatives. Flowers, in shop we buy like the whitest flower we try to go. Its whitening agent is the aluminum. Sometimes aluminum is added as a shelf-life enhancer, so that it can be stored for longer time. Why it is so? Why we are so fond of aluminum? Because of its malleable character, moisture retainer, lightweight, cheaper, and obviously recyclable. So we are not, sometimes we say we are not doing harm with the environment. We are reusing that aluminum so that unlike plastic, but we are causing harm to us. Spices, seasoned foods, some specific areas where grown rice, cereals, some vegetables, herbs, pound cake, pastries, biscuits, because of to increase their shelf life, to make them like that khasta, kurkure type of things. Like we want it very crispy. That crispiness is coming from aluminum. Packaged salt, we want it's a free flow salt. I think everyone might be remembering some days that advertisement, free flow salt. It's not getting any moisture. Those pre-packaged salt is because of aluminum. Rock salt, colored rock salt, aluminum. Some nutritional supplements, they are being added with aluminum so that it can be as an anti-caking agent. Some color, some presentation is being given by aluminum salt. Anti-caking agent is commonly being used. Not to forget about the medicines like antacids, phosphate binders, buffered aspirin, then astringents. As an astringent, so use aluminum. Vaccines and allergen injection, aluminum is being used as a adjuvant. All the vaccines, they are commonly with aluminum. And then dental crowns and dentures, aluminum. Dental cements, this aluminum is being used. So many cases we give them as medicaments, aluminum is being used. Industries, for construction industry like cement, automotive industry, fuel, they use aluminum. Aviation industry, you know, the lightweight metal corrosions are there. Electrical industry, sometimes the electrical wires are being used. Aluminum is being used in the explosive and fireworks. Fuel additive, solid fuel rocket propellant, aluminum being used. And this aluminum, part of it is coming to the environment, and we are like, remember this air fresheners, those are aluminum salt. Those salts, they are giving us to take aluminum, they are forcing us to take aluminum through the nasal routes. If we see the dynamics, how the aluminum is it's being entering in our body from water, from earth crust, through plants, through meats, through fishes. It's entering into our circulation. Not to forget about the medications, over-the-counter drugs, IV injections, dialysis, parenteral nutrition. Once they are in the circulation, deodorants, cosmetics, they are also adding. Once they are in the circulation, mostly it is being used, being excreted by kidney, not being absorbed easily. Its assimilation is very poor. However, whatever assimilation is done, those are going mostly in CNS, bones, liver, and kidney. You remember that all the intramuscular injections, vaccinations, it's myofasciitis. Its cause is aluminum, because the drug what is been given, that drug has been absorbed, but aluminum remain within the muscles. They cause slowly leaching out into the muscle related areas, and there is intramuscular muscle myofasciitis. Then through nose, it can goes into the occipital cortex, into the frontal cortex, into the hippocampus directly, and we can have memory problem, brain disorder, brain neurodegenerative problems. Internalization is very poor, as I have mentioned. Luckily it is only 0.1 or even less percentage of aluminum. But the way we are being exposed to aluminum is too high. 0.1% become a considerable nowadays. And unfortunately, we are taking this deliberately. We are adding aluminum into our life. However, overall, the aluminum entry into the body is restricted, and it depends on the routes, like doses and accompanying substance, like green tea. The acidity, citric acid, it helps aluminum to get into the plasma easily. Then within plasma, it remain bound, mostly bound with transferrin. So you can identify there is a problem with aluminum. Kidney is very efficient to excrete it, but we don't have any system to excrete aluminum because our body does not identify aluminum that as it has entered. We don't have any opportunity, any scope. We don't give any scope for aluminum to enter. At the same time, we don't give any scope to aluminum to leave. So if by any mistake aluminum entering into it, very less 0.1%, but we have very less chance to let it go out. I am not talking about the body exposure. As I have mentioned, assimilation, the amount of aluminum which is entering into the cells, sorry, excuse me. That part is very less, but once it entered into the cell, once it is inside the cell, there is no option we can take it out. That's become a big problem. As we grow, we are from childhood onwards, like we start vaccination, we start nutritional supplements. Then as we grow in adolescent, we started the food which is not fast food. So throughout every options, then middle age we started acidity, so antacid, over the counter antacids and other drugs. So we start accumulating aluminum. By the time we age, we are already have a sufficient load of aluminum causing them to us. What problem it is doing? We do many things. It has problem with memory loss. It can cause irritability, mental confusion. It has proven effect on anemia, muscle fatigue, dyslipidemia can be there. Not very specifically mentioned. Joint stiffness, male infertility, female endocrine disruption, osteomalacia, allergy, bowel problem. Even BIS is IBS is also sometimes, Crohn's disease is also sometimes associated with the aluminum. And very recently, breast cancer is also, like 2005 onwards, breast cancer is identified that it has role, aluminum has some role. As we know, as we grow, see that it is a toxic, neurotoxicity, hepatotoxicity, bone toxicity, lungs limited, but toxicity is there. Skin toxicity, sometimes we can see. Reproductive endocrine toxicity, how it is working. It is sometimes so that it is related to diabetes because of the damage, autonomic damage it can cause, or neural damage it can cause, so that the responses, neural control is being withdrawn. But as endocrine disruptor, as role in breast cancer, as role in male infertility, how aluminum can be linked with that? Later it is found that aluminum is metalloestrogen. Let us see what are the early symptoms of aluminum toxicity. Nobody can say all these symptoms has any role to any particular disease. This is a so common, so generalized symptom that we cannot say that because of these things there is aluminum toxicity. That's a big problem. So we don't have any specific symptom which can be related to aluminum toxicity. If we see later symptoms, and these symptoms, many, it's a list of compounding list of symptoms, and many of these symptoms are only can be said it is because of aluminum toxicity if it is identified, if we see that aluminum is there in that particular patient, then only. Otherwise there may be 101 causes for these diseases. So we don't have any specific symptom to identify and tell that this is aluminum. Remove aluminum and prevent these problems. Experimentally, it is a long study. People are working in this, that neurotoxicity is proven. We have also done plenty of study. We have done since 2000, not 2000 even. I think I started working 1992. I'm working in the field of aluminum toxicity, and my area is neurotoxicity. We have seen that like almost everything in brain has some role with aluminum, can be linked, not proven specifically, but can be linked with aluminum toxicity, and it finally come to the learning, memory, behavioral disorders. Very recently, we have reported to some extent, just a few cases, that aluminum has some cardiotoxicity, autonomic toxicity, dysautonomia can be linked with. But all these are long-term studies. So the question is there any gender difference in aluminum-induced neurotoxicity? Because if we see that it is going into breast cancer, it is going into male infertility, it is going as an endocrine disruptor in female, as Madam Dr. Sucheta Madam has also mentioned that PCOD, there is polycystic. So female reproductive as well as male reproductive infertility, male infertility, this problem is suggesting that link with estrogen. And aluminum is a xenoestrogen, it acts as a metalloestrogen. How it is working is not clear. Then, is there any acute effect of aluminum? And not in terms of lethal doses. There are many studies, 15 days, 20 days, 3 weeks, 4 weeks studies, where aluminum is given in excessively high doses. Who is going to take that excessively high dose of aluminum? Unnecessarily, just to see the toxicity, it is of no use. But my question is there any acute effect of aluminum with considerable low dose, very low dose and very low level of exposure? Can we identify that this may be because of aluminum? Is there any systemic toxicity response against aluminum exposure? Can there be an identifiable effect of aluminum which can be noticed early? If we can identify that, okay, this likely a condition, likely to be a stage going towards aluminum toxicity, then we can ask the patient, we can ask the subject public that you avoid this, you do this. So with this, we have found that there is some gender difference. In female geographies, we found that even in 96 hours, very low dose aluminum exposure, we could see that aluminum effect, it is changing the behavioral pattern in female and male. But the male-female difference is there, gender difference, slight gender difference is indicated. Similarly, if you see here very clearly, if you see minutely, there will be some reddish, pinkish lines in the histology graphs. Those lines are aluminum staining, special staining, aluminum staining. We have found that within these 96 hours, brain cells, they could accumulate aluminum and they could retain them. In female, you could see that it could be easily cleaned off, but in male, you could see it is retaining. So there is likely, at least in acute effect, there is some study, some difference in gender. Then we have started with if there is a gender difference. Female, pre-pubertal female and the pubertal female, they are supposed to be gender different. Pre-pubertal female, there is no estrogen, not it's like male, and the pubertal, just young adult, they will be having a estrogen flash. It's like a huge estrogen supply. So we wanted to find out if there is any effect, and we have seen that plasma triglyceride and liver triglyceride is altered in young adult, but not much effect in pre-pubertal. So metalloestrogen, estrogen related effects are there. Similarly, we have seen in plasma total cholesterol and liver total cholesterol also that young adults, they are more susceptible than the pre-pubertal animals. It is this study is done in peripubertal female rats. Then in young adult male female, we have seen that female, you could see that serum triglyceride level is high, but for male it is not. However, there are variations, lot of variations are there. We could see hypocholesterolemic effect, hypertriglyceridemia with hypocholesterolemic effect, and in some cases there are male-female differences. And then we could see the atherogenic indices, even though there is not specific hypercholesterolemia, but atherogenic indices are disturbed irrespective of gender. If it happened, just to remind you, it happened with 5 milligram of aluminium and 10 milligram of aluminium for only 15 days. Very low dose, only for 15 days, but systemic exposure, not oral or any other. It is systemic IP exposure. So even if low dose of aluminium, there are some acute effect, and we proposed these changes that how aluminium could lead to dyslipidemia, which may be atherogenicity and can be linked with the neurodegenerative diseases. So it is that we have proposed in this picture that oxidative stress, metalloestrogenic activity, dyslipidemic activity, and the neurodegenerative diseases, all these are linked together. Then recently we wanted to see what happens, even if this low dose exposure, what happens with the RBCs. We observe that even though in chronic there is no microcytic anemia is reported already, but in acute effect also there is microcytic anemia. We could found there is microcytic anemia with hemoglobin level. As you can see, microcytosis. If both 5 and 10 milligram dose is showing microcytosis. We could see that mean corpuscular hemoglobin is not much altered, with MCHC is increased. So there is a chance of hemolysis also. What we have seen, what we have noticed, that it is to some extent dose dependent effect. In case of male, MCV is decreased, but not the RBC count. With 10 milligram dose, RBC count is decreased, and overall effect is the decrease in hematocrit. Similarly, very similar pattern we have found in female. So as such, there is no specific gender difference. So we can think that because of those areas where there is a chance of estrogen, even in hemoglobin count, is RBC count, there is a role of estrogen and testosterone also, there is a chance that aluminium might be having a gender specific effect. And there are some effects where aluminium is gender non-specific effect it is showing. Then we wanted to see the leukocyte profile. What happened? We saw that total leukocyte count is decreased, and it is more prominent dose dependent effect. As you can see, male, female, not much different, but statistically little difference is there, but not visually, as we can see, not much difference between the male and female. Neutrophil count, we could see decrease in neutrophil count. Other things like other leukocytes, they were not much altered, but importantly, there is an increase in mononuclear polymorphonuclear cell ratio, which is an indication of a generalized inflammatory stage. So even in 2 weeks time, aluminium could generate a stage of systemic generalized inflammatory stage. That means this can be a lead for us when, if you think of specific like hypertriglyceridemia, hypocholesterolemia, as well as hemolysis, anemia, microcytic anemia, and systemic generalized systemic inflammatory stage, could be an indication that, okay, is there aluminum? If all these are ticking, then we can think of aluminum. We don't have any explanation, but this is our observation. Thrombocytosis. We observed thrombocytosis, increased thrombocyte count with almost dose dependent. Not statistically, we couldn't see it's dose dependent, but visually you could see there is a dose dependent thrombocytosis in presence of aluminum, systemic aluminum exposure. Low dose, acute. Hepatotoxicity, very, very limited. But specifically, if we can count cells, we could see there is dose-dependent cytoplasmic vacuolation, which is not going into the hepatotoxicity level, but cellular degeneration has been initiated. We could see that nephrotoxicity, serum urea is increased. Serum urea is showing some degree of, so even in low dose of aluminum, acute effect could be seen. Next, we wanted to see whether there is an autonomic tone, change in autonomic tone, even in this 15-day study. Is it possible? We could see, statistically, we couldn't see, get great result, we couldn't get, but there are some changes. However, we believe that if we can increase the number of observations, we are working on this, then we could have a better output of this result. To some extent, autonomic toxicity could be seen, some indication is there. As you can see, total power is decreased, but here total power is decreased, but frequency and not much change. So power and the both frequency domain is not changed much. So to summarize, what we could suggest that acute low dose aluminum exposure systemic exposure can alter the lipid profile, atherogenic indices, can cause to microcytic anemia, leukopenia, thrombocytosis, dysautonomia, to some extent hepatotoxicity, nephrotoxicity. And if we take all these points together, maybe we can think of a unique form of identifying that, okay, these population, these patient, they are subjected to later stage, subjected to aluminum induced long-term chronic effect. And as you see, those later symptoms, what I have already discussed, it's a well-known fact that it has a breast cancer role, it has PCOD some role, then reproductive function, male and female reproductive function role, then neurotoxicity, osteomalacia, all these, they are hardly reversible. We cannot do anything once we know. But if we can identify, we can have a doubt, we can save those patients to go into that, so that we can prevent these long-term effect to some extent. What we can do as such, my take-home message from this presentation, identify the avoidable sources of aluminium and keep those away from us or keep us away from them, whatever it is. Either way, I would like to acknowledge Miss Tripti Sharma, Miss Priyanka Tanwar, Mrs. B. Sirisa Kalyani, and Dr. Pramod Kumar, Dr. Hiraram, Dr. Surajit Bose, Dr. Sival Srivastav, Dr. Hare Krishna, Dr. Kamlakant, Dr. Mithu, Dr. Abhishek, Dr. Poonam for supporting in this work. And obviously, the infrastructure and big support from my institute, All India Institute of Medical Sciences, Jodhpur. Thank you very much for your patient listening. And if any doubt, if any query, I'm happy to answer. And I thank Dr. Gopinath, organizing committee member, and Dr. Susmita for giving me this opportunity, and the college, institute, and the mission. Thank you very much. Thank you. Thanks a lot.
Thank you so much, sir. Thank you for this wonderful talk, which has enlightened us and also brought in a lot of awareness regarding our food habits. Thank you. So over to chairpersons.
Dr. Prasan Priya, can you hear me?
Yeah, yes, ma'am. I can hear.
That's a wonderful presentation, and it's something which is very relevant to the present times. As we are all born in India from the Vedic times, we have not had written records of aluminium being used in any way. It was always gold or bronze or brass or copper.
That's true. That's true.
And we can link it with the mental equilibrium and the clarity of those days with what we are facing now. I think this is a very, very big challenge. And you have rightly pointed out, and it's very interesting that a lot of things which, this is in fact very alarmingly distressing we are actually invading the world of aluminium. Like everywhere, it's aluminium, aluminium, aluminium everywhere.
Exactly, madam. It is everywhere.
And we don't use those wrappers. Like in South India, we have those plantain leaves and other substitutes. In those days where we have lots of, especially in my state here, we still use a lot of banana leaves, which is considered, a substitute that obviously can be used as a substitute, but then, people always go for the refined one, which is very, very, very harmful for all of us. Now, besides that, I want to know which part of the brain is affected most, and is amyloid protein in any way related with the aluminium toxicity? That's my first question. Second question is there any relationship between aluminium toxicity and the iron metabolism? Because you have specifically pointed out microcytic, hypochromic, probably to do with the iron. And then the variant of aluminium, which is most harmful to us, because there are certain instances where we cannot do without aluminium. For example, the aeroplanes which we use, we definitely require aluminium. The type of variant which is less harmful, let's say. And that's it. And and the last one, which the substitute part, any other things which can add more than that? Can you just clarify these? I really am interesting. I'm really interested in that topic, sir. Thank you.
Thank you, madam. It's a very good. Thank you for your appreciation, and it's nice. It's nice. First one, what you have asked, it's like which part of the brain, as I have studied like in Alzheimer patients. I didn't study Alzheimer patient. I did experimental studies only. But there are plenty of reports showing Alzheimer's brains. They have autopsied Alzheimer brains studied, and almost all the brain areas, aluminum is there. And most effective was, most part they are interested was hippocampus, frontal cortex. And in experimental animal, I have seen it is present in frontal cortex, it is present in thalamic area, it is present in parietal cortex, it is present in temporal cortex, is it present in cerebellum. In fact, I remember I have published one paper where I have shown that the Purkinje cells, the actions of Purkinje cells, I could show them there directly the aluminum deposition in that. But what you have mentioned about, it's a very interesting one. Aluminum and amyloid deposition is not directly linked. They say there are some suggestions that aluminum goes into that hyperphosphorylation of the tau protein. Actually, I didn't touch the molecular part because I felt awareness, as you have identified, that awareness is more important about it. Instead of showing the molecular intricacies, we go into science and we go into details of that molecular intricacies. It's nice to listen, nice. But as you go into the life, real life situation, aluminum is so much embedded in our life, we cannot really replace it. Amyloid deposition is not directly linked, but in some cases they have shown that if it is associated with some other proteins, some some small proteins or some other toxicities, if it is associated, then there is a chance of amyloid deposition. But aluminum as such is not deposited there. And if we think of replacement, the physicochemical property, the chemical and physical property aluminum is having, this lighter metal is having, it's really, really tough to replace it. There are areas, like as you have mentioned, like a rocket fuel, solid rocket fuel, or even if it is for fuel combustion or for this construction industries, we cannot replace aluminum. Mining is required, it is required. But at the same time, there are areas where we can, as you have mentioned, we can easily avoid the aluminum foil. Or if we inculcate that, okay, we will be looking only for paper or only for those sal leaves, as you have mentioned, that banana leaves, sal leaves, and in South India also some big leaves are there, or even for these lotus leaves are used to fold and for cooking also. So those things we can inculcate out. We can just stop our taking those preservatives. Preservatives is a huge amount of preservatives is being given in the pickles, achars, all these storage pickles type. Homemade, they are not being used. Aluminum is not being used, but commercialized. Then things making it crispy. Unnecessarily, we are making it crispy. We go for a atta, wheat flour, and we look for the whitest one. But whitening agent is being given. I think you remember, maybe I'm crossing a time limit. Sorry that, but I feel like talking this, whenever time, you just let me know, madam. And, in our childhood, we used to take any nutritional supplement. The last part of it, it is never, we used to ask our parents, give me that, it is something like chocolates. Nowadays, if you notice, those last part also can remain in powder form, because we are adding anti-caking agent. Then the deodorant, antiperspirant, after the cleaning of armpit, shaving of armpit, girls, they use this deodorant, and these are aluminum. And this aluminum is going, and particularly the upper lateral quadrant is being seen that aluminum is being accumulated there in the breast, and the tumor formation is more in that area. The fluid coming out from those tumor cells or those abscesses, their aluminum source is there, aluminum accumulation is there. We are giving plenty of vaccination. I don't know, I don't have any replacement for adjuvant, aluminum as an adjuvant, but we are giving aluminum at the birth itself. Thank you for interesting questions.
Any other questions from the panel?
Vivek sir, you want to have any questions?
Sir, Vivek sir,
So you are
please unmute yourself.
You're muted, sir.
Sir, Vivek sir, please unmute yourself.
Dr.
Yes, sir.
Prasoon, it was very interesting talk. Already unmuted, but I don't know. Am I audible or not?
Yes, sir. Yes, sir. Yes, sir. Yes, sir. You are audible.
Okay. So, sir, I would like to say that we have to change our lunch packs. That's of the situation. That aluminium is never found on free terms, and it has penetrated deep into our society and to our culture also.
Exactly.
Somehow
sir.
we have to remodify ourselves, and we have to see that we are capable of doing earthenwares and other things again, and we have to get out of it. I'm sure that half of the people who are listening to this talk must be having their food served within the lunch packs, that is having the aluminium foils. Somehow we all have to think about it.
Yes, sir.
And it's a very interesting talk about it. Thank you very much, sir.
Thank you, sir. Thank you, sir.
Thank you very much. We would like to express our gratitude and a token of appreciation. We are providing our e-certificate and a memento for our speaker, Dr. Prasan Priya Noy. Thank you, sir. And for spending their valuable time and as a token of appreciation to our chairpersons, Dr. Sharada Ningtoju. Thank you. E-certificate and a memento. To our professor, our teacher, Dr. Vivek Sharma, sir. Thank you, sir. Thank you for chairing the session. Dear delegates, with this, we'd like to conclude this session and we'll continue with the next topic. Thank you.
Thank you so much for giving us this opportunity to moderate, to chair the session. Thank you, Madam Manju, then Dr. Gopinath, and my student Frank. Thank you so much, Frank, for all the for the invitation. And sorry, I was a little late by 2 or 3 minutes because I had some technical problems. Thank you so much.
Thank you. Thank you, everyone. Thank you.
Thank you.
Thank you. Thank you, the speaker especially. Thank you so much.
Very good afternoon. We'll start with the next session. Rohini ma'am, good afternoon ma'am. Dr. Shweta, are you there? Dr. Shweta, can you just switch on your video, Dr. Shweta? Dr. Kulkarni Shweta? Dr. Shweta, are you there? One minute, ma'am. Rohini, ma'am, 1 minute.
Sure, ma'am. No problem.
Dr. Shweta, can you hear me now? No, you're not heard here. Madam? Dr. Shweta? Dr. Shweta? I can see your name, but I can't hear you. Yeah, now.
Ma'am, please rejoin.
Dr. Shweta, just leave the meeting and just rejoin. That's what our technical team is. Yeah. Please take, she has logged out, madam. She'll be logging in again. Please do help her in putting her as a panelist.
Yes, ma'am.
Yeah. Good afternoon, Dr. Rohini, ma'am.
Good afternoon, ma'am. Am I
How are
audible,
you?
ma'am?
Yeah, you're audible, ma'am. How are you? Fine. Thank you,
Fine,
ma'am.
ma'am. Thank you.
Yeah.
Glad to join for this conference, ma'am. Thank you.
Me too. Thank you for accepting our invitation, ma'am.
Thank you.
Hello. The tech team, please note, Madam has entered as Prarthana. Just see the name Prarthana in the attendees and please make her the panelist.
Done, ma'am.
Okay, thank you. It's not right here. Yeah, she's there. Okay. She's not entered. Dr. Shweta? Dr. Shweta, can you hear me?
Dr. Manju,
Yeah. Yeah, can you switch on your video? She's having a net issue. It's not there. Yeah, she's inside. She's there now.
hello.
Yes, ma'am, you're audible. Okay. Okay. Dr. Shweta, you're audible.
I'm there.
Yeah. We'll start the program. Rohini ma'am, just sorry, ma'am, for a small delay.
No, not at all, ma'am.
Okay.
Yeah,
We'll start it. Actually, Dr. Shweta ma'am, she's
Yeah.
having a university exam. She's conducting the exam. That's why from there she's chairing the session. So I would like to start the next talk, and that is it's being chaired by Dr. Kulkarni Shweta. She's an MD in biochemistry and PGD GCLP, the postgraduate diploma in good clinical laboratory practices. She's a professor and the head of Department of Biochemistry in Mahatma Gandhi Medical College, Puducherry. She's having a 14 years of teaching experience, and she has secured a gold medal in postgraduate diploma in good clinical laboratory practice from Sri Balaji Vidyapeeth University, Pondicherry. She has published a book titled Competency-Based Practical Biochemistry Textbook. She's a qualified internal auditor for clinical laboratories, and she has experience of work as the quality manager for many accredited labs. She has published original articles, case reports, and a lot number of review articles in national and international journals. She has guided many MSc medical biochemistry students and has also guided ICMR STS projects, and she's a recognized PhD guide at various universities. So with this, Dr. Shweta, you can just start introducing our speaker and you can take over from here. Thank you.
Thank you, ma'am. Thank you, Dr. Manju. Am I audible, Dr. Manju?
Yes, you are audible, ma'am. You can just start. You can start introducing, ma'am, the speaker.
Yeah. Today's eminent speaker is Dr. Rohini Karnakaran, madam. Madam is associate professor and deputy dean academics and international affairs, faculty of medicine, AIMS University. Madam has done PhD in biochemistry, and she has done various fellowships. Among important one is the UK honorary doctorate in education articles, research papers, and 63 conference proceedings. Madam has 7 patents, 10 books, and 10 chapters. And she has been associated as a member of several internationally reputed educational societies and communities. So, Madam is an eminent speaker on AI today. So, let us have academic on AI today. Thank you, Madam, for giving, Madam.
Thank you, ma'am. Thank you for inviting me for this session.
Madam, I have
Can I share?
completed. Rohini, Madam, can start now. Thank you, ma'am. Thank you for patiently waiting for us.
Thank you, ma'am. I truly understand the difficulty of the examination system and how busy you must be. Thank you for joining and being a speaker for this, chairperson for this session. Good afternoon. So thank you for inviting me for the session. So the commonest thing is, the question is, most of us debate as to, will AI take over the world? Because AI is a term known by even children here around the world. And the commonest thing that we question is, will AI take over the world? But the question that we must have, or we must ask, is how AI will take over the world. So that is a challenge that we have here. As we know, AI is everywhere. And I'm presenting on digitalization and artificial intelligence, the future of clinical biochemistry, where AI is involved in biochemistry and in clinical chemistry. Thank you for the invitation. So as we know, AI is everywhere. AI is in our mobiles. Every day we wake up with AI, the alarm, the smartwatches, the nano sensors, everything is integrated in today's world. So AI is a part of every aspect of our life. And I'm here to tell you more about AI and laboratory diagnosis. And when we're talking about technology, AI is all about robotics. AI is about the computing that is available for our system. AI is also in the environment, the big data. As I mentioned, the smartphones, the big data, everything is connected and everything is around with AI. But our focus is mainly on the healthcare. When we say healthcare, we have the pharmacology, pharmaceutical companies, the biomedical companies, where their market values are improved with a lot of AI into the system. And we have patients. When we're talking about healthcare, patients are the main one. So for patients, we may have access to personalized therapy. We will have a lot of time and a lot of cost efficiency and a good quality results which could be obtained. So that could be a major thing for AI. And also with respect to research, we can improve the research facilities. We can improve that and analyze through the big data in no time, in a very short period of time, which can really help the patients for the diagnosis. So AI is everywhere, and we have AI in every part of our system, and we use AI in everyday life. So when we're talking about AI and AI about healthcare, what is basically this AI? AI refers to the ability of machines to act in a seemingly intelligent way. So we expect machines to understand us, do what we want. So artificial intelligence has gained its reputation because of the intelligent ways of making decisions in response to new inputs, which is explicitly being set and programmed. So when you take a general computer, it's just a set of instructions, whereas AI is designed to use data-driven models to make the predictions. So AI drives the world around. So AI is today being a very explosive one, which is all around the world and everyone knows about AI. But was AI the recent one? Actually, AI was originally from John McCarthy in the year 1956. It's a very old thing, which is now in boom. So basically, AI is about the theory and development of computer system where you perform the task which requires human intelligence. So that is why it is artificial intelligence. So the AI has been growing over and over through a significant improvement with computers, hardwares and softwares, and it has been rapidly going in various other fields. And today's our topic is on the healthcare, and healthcare digitalization has been of a great impact, and AI has offered a great opportunity to improve patients' outcome, the reduced cost and personalized care, which is the most key important thing when it comes to AI in healthcare. So the integration of AI, the digitalization and the integration of AI has significantly impacted in the field of clinical biochemistry, more because of the advancement of the tools and improved diagnostics, the treatments, and overall healthcare processes. So digitalization has allowed the gathering and storing of enormous amount of patient data. Patient data is our wealth. So for a clinical biochemist, the enormous amount of patient data has been digitalized and been saved and have been used for later purposes. So AI-driven data analytics have found to examine and evaluate the most recent trends and correlations, which has been helping the people and the data that can be used. So they can be quite challenging for the clinicians, and AI-powered data analytics has been a boon and a great help for the clinicians. So basically, what does AI do? The goals of AI is to make sure it is done in no time, which is quick. The precision is very good. It reduces the problem-solving conditions, and it is quite problem-solving. And the knowledge is represented because it's artificial intelligence, the planning, the natural language processing, and finally the motion and manipulation. So these are the basic goals of an AI. So AI is amorphous. Amorphous, it exists in any form, exists in many forms. So the level of complexity among the AI could be classified as, it is mentioned as classification and prediction. When classification is used for diagnosis purposes and prediction is used to forecast or predict the treatment and the diagnosis for the patients. So AI is in many forms and it can be used in any forms in healthcare setting. So when we talk about biochemistry, artificial intelligence is involved in various cases with healthcare and biochemistry. So basic is diagnosis. We used biochemistry, being a biochemist, we use diagnosis, which is early detection of diseases. When we talk Alzheimer's is something which has no cure. However, there is still research going on, but it could be diagnosed earlier, which can help in prevention, which can help in the treatment and management. So early detection of diseases is of great support with AI. And predicting using the key biomarkers on the impact of various diseases. The life expectancy of the patient could be improved. The progression of the severity of the illness could be maintained. So these are the predictions which can be done using AI. Symptoms, identifying the symptoms from the clinical records, which is all about the data, the impact of comorbidity, the big data, the lot of clinical trials and the data that is being stored in the AI in the system. Treatment, effective medication, which is based on the symptoms and prior treatments that can be done through AI. New drug discovery, being biochemist, we do bioinformatics, the genomics, the proteomic studies. You can do a number of studies which involves new drug discoveries by drug formulations. And imaging is one system which is involving AI and is being successfully going on for years now, where imaging uses AI and it is able to achieve and used in the diagnosis of diseases. So AI is used in a lot of important aspects, which is in the laboratory for diagnosis and various other treatment and managing the conditions. So there are novel applications, including those involved in the laboratories, which involves the results, their diagnosis, and most importantly, today's world, we need to be aware of the risk prediction. So risk prediction may help to transform the role of biochemistry laboratory using AI, because AI has changed the world, and that can help in a lot of identifying, diagnosis, in treatment, in management, and most importantly, the risk prediction, which is a very crucial part of our clinical biochemistry. So starting with AI for clinical biochemistry, when we say AI, it is basically technology. We're talking about automation. We're talking about digitalization and decision-making in an intelligent behavior, but we are not doing it. It's the artificial one, which is the computers, the systems, the data, which is doing it. So fundamentally, AI is categorized into non-adaptive and adaptive mode, non-adaptive being the rule-based mode and adaptive meaning the machine learning. So non-adaptive is basically auto-verification of your laboratory results, which we do every day. So those are non-adaptive artificial intelligence, where auto-verification of laboratory results take place. However, adaptive AI is automated classification of conditions, example like leukocytes from digital image of peripheral blood smear, which is by a trained machine learning algorithm. So we need an algorithm and it is an adaptive AI, and an auto-verification is a non-adaptive AI, which we are using regularly for our laboratory results. So AI is a very important role in clinical biochemistry. And since it's revolutionizing in clinical and medical biochemistry, there is a large set of complex data that is being stored. And we have this AI which generates a lot of data, which is from the various sources, like, as I mentioned, the genomic sequencing, the genomics, the proteomics, and the metabolomics. So researchers have been studying and they have been identifying and studying a lot of data, which have stated that reading a data, interpreting a data could be time consuming, and the most important is they could be prone to human error. However, AI is revolutionized and has reduced the time. With no time, you can get the data which could be error-free. So AI can process and analyze the data sets as much fast as we can. And the researchers can identify the patterns, the correlations, they can understand and digitalize. The digitalized version of the results could help to transform the patient condition by understanding the results. So as a result, the laboratory medical profession would be facing a big transformation with AI, because the digitalization of the big data, the AI and the machine learning has turned the world around with the human healthcare setting. So when we talk about AI, we know it is adaptive, non-adaptive. It can also be further classified as general, narrow, and other different various of levels of classification depending on the healthcare setting. So AI in general can be classified as general, which is for all purpose, as we know from the term, and it can be task independent, whereas narrow is focused and task targeted. So we are looking at the narrow AI, because AI which is experienced are looking into narrow applications, where narrow AI is basically rules based and which includes machine learning. So this encompasses expert systems where machine learning algorithm means a fixed automated algorithm is also set in, which is to avoid errors and have a quick results in no time. So the expert systems of AI employs the rules of that are explicitly programmed by the human expert, and there can be auto verification, there can be algorithm reading, there can be results, there can be predictions, all this being done. So when we're talking about the narrow artificial intelligence, as we all know, narrow artificial intelligence encompasses expert systems, the rule system, and the machine learning. And they are also called as supervised ML. So the supervision refers to the ground truth, which is basically the narrow artificial intelligence, which has expert systems, supervised machine learning, and unsupervised machine learning. When we say expert system, it is rule-based. It includes, as we say, about the results that we get every day in the automated instruments, which is auto verification, the results flagging, the specimen routing, identifying the results, identifying the specimen if there is any error, and reflex testing protocols. All these are expert systems. Supervised machine learning is algorithmic machine learning, where regression analysis, identifying the calibration curves, prenatal screening, mass spectroscopy, and neural networks, which is the most important thing when it comes to gel electrophoresis involving serum electrophoresis and so on. Unsupervised is in genomics, where you can run the samples, you can do a lot of docking and studies which can involve unsupervised machine learning, where the algorithm is set, and you also have text mining, which can involve. So all these are the narrow artificial intelligence, which plays a very important role. So machine learning based software does not require human experts to explicitly program directions to get into the question to answer. However, supervised machine learning, it does not, for supervised machine learning, it does not require human experts to explicitly define the ground truth categories. So AI applications relevant for clinical chemistry laboratory are framed in the context of phases of testing. So when we say phases of testing, they involve the analytical, pre-analytical, the analytical, and the post-analytical. So most of the machine learning based technology that is used in clinical laboratory is supervised, as a supervised ML. That is, we have supervised, unsupervised machine learning. So supervision lives with the ground truth, which is represented by the data set. So example is, this is one of the thing which is for the machine learning, which is involved for the analysis. So example, you can see the data that has been used in the supervised machine learning algorithm with the input data and the output data. So how it appears. So this could be an example where a collection of digitally scanned immunofixation electrophoretic gels are labeled by human expert as non-monoclonal protein or monoclonal protein. So using that data set, the associated labels through the process of training, ML can identify the patterns within the data set and correspond to the presence or absence of the monoclonal protein, which will enable us to identify the condition, which is a very crucial thing in analysis. So in laboratory analysis. So as I mentioned, basically ML involves three cases: the context of phases of testing, which is pre-analytic phase, the analytical phase, and the post-analytical phase. So anytime in a laboratory, we have a test order, the patient's specimen is received, it is identified, the specimen is collected in the OT, which is the first one, and then the specimen is transported where the analysis takes place. Specimen is received by the respective laboratory technician. It is being processed and analyzed. So once this is done, we have the results that is reviewed and the results are reported. Now, this is a general phase of our regular reporting system. So we have the three different phases in this, which is the pre-analytical, where we identify the wrong blood in tube detection. It is a pre-analytical phase where the specimen is collected, identified, and transported. We also have the result detection, and the specimen quality is identified during the pre-analytical phase. In the analytical step, it is a specimen analysis, where here we have a very important thing, which is about the risk prediction, the diagnosis and risk prediction, the clinical interpretation support, image analysis, result prediction, and the quality control is in the analytical step of your the phases of the laboratory testing. And when you take the post-analysis, it is a risk prediction, the diagnosis and risk prediction, the clinical interpretation support, image analysis, auto verification of the results, and the reference intervals. Only then it finally concludes with the reporting of results. So the overall process takes place in three phases, which is pre-analytical step, identifying the quality, identifying the wrong blood in the tube, detecting if there is a wrong blood in the tube. All the result detection is in the pre-analytical phase, with risk prediction starting in analytical, continuing with post-analytical, and you have the results prediction and the quality control, followed by auto verification and reference analysis. So with so much of laboratory diagnosis that is going on, so we have a different lot of framework that is set for disease detection system. So being a machine learning algorithm being set up, we have the medical data set where we have the disease symptoms of the patients and the pre-processing take place. And then the disease symptoms, we feature the vector, identify through the apply the machine learning technique, the deep model technique, and using the prediction model, the disease is predicted. Example, as I mentioned, the alzheimer diseases and conditions, it could be predicted at the early time where it could be managed quite effectively for a patient, and identifying disease is the most important part of machine learning and deep learning model. So once it is prediction model is set in, then you have the medical test data, which is also verified, used in for the prediction model, which will enable you to do the disease prediction. So you have the medical data set, the disease symptoms, and the test data. So both the pre-processing of the test data as well as the medical data set is done. The disease symptoms associated with the test data feature vector, both these are used. With machine learning, the prediction model is involved, which predicts the disease for any person, which enables us to identify the disease. So this is basically the disease detection system in the laboratory, a framework for identifying and predicting the disease earlier using AI and machine learning. So when there is so much of machine learning and data, we have a holistic artificial intelligence in medicine, which is a framework which processes data. It's not that the sampling, the auto verification of the results, but when we say a big data, it is the healthcare institution, the whole university, the hospital that is involved. So based on the healthcare institution and hospital, we have a lot of data from the patients, and we have a lot of big data, the lot of data that is stored in the patient files. So we have the patient files. So using AI, we can easily retrieve any patient file using AI. So that will enable you to identify what is the condition of the patient. I'd feature the extractor, what is the tabulated data, what is the time series, what are the condition of the patient, and what is the state, the notes, and what is the current position of the patient, the pre-trained transformer, with the final medical images with using AI. So a doctor can easily look at all these using the healthcare institution AI that is set up, which using the resources they can sit at home, look at the patient data, look at all the images, the results, and the natural language and time series, and they can easily go in for inputting the final results. So the doctors can treat patients at a very clear, short span of time with no error in the artificial intelligence of results, which enables us for the treatment. So artificial intelligence is involved in mapping in laboratory medicine. So when we say about patient's input, the patient data and the files and how it goes on. So when we talk about the laboratory procedures, we can have the set of data that is inputted. So we have the algorithm that is set. We have logistic regression. We have support vector machine. We have artificial neural network, which is very important. And all these are involved in the machine learning algorithm, which is already set. So once we input the data, the raw data with all the labels stating the condition, then automatically we get what is the probability, the prediction probability of the sampling. So this is very effective, and in no time we can predict the conditions. And this artificial engineering and mapping of laboratory medicine has been very significant and have been quite popular in healthcare setting. So talking about laboratory sciences and those with biochemistry, we have a management of artificial intelligence and various other conditions in biochemistry. So the commonest one which we start with is the smart watches. We have even smart lenses. We have a lot of things with smart. So basically, the smart watches and phones or apps are used nowadays, and there is a lot of apps that has been developed for the treatment, for the management, for the identification and prediction. So management of diabetes with artificial intelligence is become a more prominent, a commonest one in today's condition. So smartphones, which has the conditions, which shows the levels of sugars. We have tattoos, which can predict the conditions. So smartphones and apps can help in recording the e-health records, and the data can be stored, and through telemedicine, online communities, and social media, it can be employed. So we have wearable devices, which could be a smartwatch, a smart phone which can be used, which can be smart lenses that can be used. All these are involved for the artificial intelligence for the management of diabetes, which can help the patient at a critical situation. And as we already know, basically during a physical activity, during eating, during insulin administration, the levels of glucose can all be predicted, and using the artificial intelligence with your smartphones and systems, this personalized insulin can be identified and it can you can identify and it can alarm and activate and predict the glucose levels that is increasing, which can help the patient for the prediction of increase in glucose and for the treatment purposes. And one of the latest innovations in AI is about the breast cancer detection using the artificial intelligence in breast cancer detection. Circadia Health, an innovative wearable technology, which is IT-BRO, has been developed for monthly breast scanning. So this will be a crucial one, and it is a very effective one. And they find to be, researchers have shown that this could be a very significant one for early identification of breast as well as other malignancies that could arise. So artificial intelligence has moved into diabetes has been one commonest one, and it has moved into breast cancer detection using the significant artificial intelligence IT-BRO, which can be scanned, which can be used for scanning, not just for breast and for most other malignancies involving this. And prediction of the long non-coding RNAs can be done with biochemistry on deep learning technology. So predictive techniques of long non-coding RNAs can be recently created with the limits of biosequencing technology. So here again, we use machine learning algorithm to identify the model. So these properties, they all can be retrieved using the coding potential calculator. And they can be trained in a support vector machine, and you can create the model and you can predict the RNAs, the long non-coding RNAs and the mRNAs for deep learning for identifying the conditions of the patients. And the most commonest one and the best prediction model is a protein prediction model, which has been used to study the protein secondary structure of the sequence. So there are more than 500 distinct proteins with unique sequences, and using the technology, the alignment of the sequences is a best prediction model of protein secondary structure for a protein sequence for any unknown 3D structure. So artificial intelligence is very significant in identifying the protein secondary best prediction for the protein secondary structure. So we can do that, and a lot of studies and a lot of data can be retrieved for protein studies. And drug discovery, moving into bioinformatics and using of drugs, identifying drug design, and in other pharmacological studies and chemical synthesis, in drug designing, drug repurposing, and drug screening. AI is involved because it's involved in predicting the 3D structures where we spoke about the protein. It's involved in predicting the drug-protein interactions that take place and about the de novo drug design, determining the drug activity so that you can identify. And you can also design the multi-target drug molecules and polypharmacology. And in other chemical conditions, it can be used in prediction of reaction yield. It can be used in reaction mechanisms. And in drugs, you can predict new therapeutic drugs, identifying the therapeutic target and predicting. And it can also be used to study the toxicity level, the physiochemical properties, classification of the target cells, and the bioactivity properties, which can be done through artificial intelligence in drug discovery. And machine learning is very significant, and we know about the algorithm. However, machine learning is very important because recent studies have shown that they play a very important role in enzyme engineering. So machine learning for enzyme engineering has been quite challenging. However, researchers have shown that they are predictors that aim at elucidating the structure and functional relationship of enzymes, both in predicting the structure for a known sequence and predicting the catalytic activity or the substrate specificity for a known sequence or a structure. So both the properties of solubility and stability of the proteins can be studied, and it's critical for successful protein engineering and especially in enzyme engineering. And the diagnosis and risk prediction, as I mentioned, the risk assessment, the risk prediction of AI is very crucial, and AI applications have an opportunity to improve our existing methods of diagnosis and risk prediction based on the data that we have, the clinical laboratory data. So the significant process has occurred in the recent years where AI has a various identification in the disease states with the laboratory data alone. It could be predicted with the laboratory data that is available, the big data, the large data that is in the healthcare setting. So early AI applications in various sectors could help us on the areas focused on risk-based models for common clinical conditions. So diagnosis and risk prediction is a crucial thing when it comes to AI. So when we talk about any condition, example, multiple regression, which is based on the models, example for biomarkers with limited clinical data, they are developed in predicting the risk of various conditions, example like prostate cancer and biopsy in patients, and non-conclusive total prostate specific antigen results and so on. So risk scores are derived from these models, from the results that we are identifying. and can be used to further stratify the patient's risk and inform the decision to proceed to prostate biopsy, reducing the risk of unnecessary biopsy in some of the patients. So this could help with the risk prediction in cases and in patients to avoid surgeries and predict results in unique results and perfect results. So there are several approaches. Example, as diabetists, we use smartwatches and other AIs in the common everyday day-to-day life. There are similar approaches incorporating specialized biomarkers, which has been leveraged to develop models to predict the decline in the kidney function, especially in patients with type 2 diabetes. So the risk scores have been applied in acute care setting to predict a wide variety of clinical outcomes in patients with type 2 diabetes by having specialized biomarkers to identify and predict the decline in the kidney function tests for patients with type 2 diabetes mellitus. So AI has shown a promising one in diagnosis, especially in the clinical laboratory, only with the laboratory data, the data that we have from the patient. So that data can help us get the profiles. Even the highly complex data could help us to identify and evaluate the cases of multiple sclerosis or multiple myeloma and further other conditions using all the profiles and MS data for steroid profile and plasma amino acid profiles and so on. So the diagnosis can be using only the laboratory data alone. So machine learning models developed from these have shown to differentiate the abnormal as well as the normal because algorithms are all set and structured for machine learning. So the classification models can help to reduce the subjectivity in interpretation and helps to give an expertise view and specialist expertise on the laboratory results that we are looking into. So another interesting fact is about the clinical conditions with overlap with the results. An example, the commonest one with iron deficiency, anemia, and thalassemia, which shows both present and hypochromic or hyperchromic myocytic anemia. In all these conditions, is very AI is being very significantly being used in the diagnostic application because it helps to clearly identify the differences and the conditions by identifying the hypochromic or microcytic anemia, which will enable us to define and differentiate the profiles, which is a very crucial one and when used in diagnostics with using AI. So this is a significant challenge in conditions with thalassemia trait, which is quite common in areas with thalassemia trait. And it is very prominent and the results could be within time. And you know the differences of the anemic conditions, which could be identified and it can help in diagnostic applications. And AI leveraging has been predominantly with the laboratory data, which I have been mentioning, and it can help to predict the treatment appropriateness and responsiveness. So that is why AI is so significant in the healthcare setting, and especially to the laboratory in a biochemistry or any laboratory, it is very important to predict the therapeutic response in patients who are receiving treatments. Example, in a inflammatory bowel disease, the treatment and all these could be predicted using this AI. So monitoring patients using drugs is a crucial one, which is a narrow therapeutic index, and AI is very significant and supportive in helping and the treatment appropriateness and the responsiveness in patients. So using urine strips have been a subject to interpretation variability and can lead to a number of errors by doing manual testing. However, beyond specimen processing workflows and artificial intelligence and machine learning technologies have really shown a streamlining in today's world, requiring manual interpretation, which has been avoided, which has been removed with a very advanced algorithm, which could be used for enabling and identifying, avoiding errors and giving us the fast results. And one of the most important thing is about the gel electrophoresis. The gel electrophoresis is a commonly used methods for all the paraproteins, for all the lymphoproliferative disorders and other monoclonal gammopathies, and especially multiple myeloma is one prominent one along with all this. So the diagnosis of this involving identification of the paraproteins is done using gel electrophoresis. So this gel electrophoresis interpretation, as we do in a laboratory in an everyday life, it's going to be time consuming, taxic and intensive for us to prepare the gel, load it on, use the specimens, and it could be so many thousands of specimens that needs to be done. But recent studies and literatures have shown that AI and ML has based on the detection of paraproteins have found to be very effective and very effective in the results and diagnosis. So AI ML based detection of paraproteins in the digitally scanned electrophoretic gels have been proven to be very successful And although this is not commercially used, it's been specifically used, the initial results are quite promising, and they may serve as a tool in assisting the laboratories in detecting and streamlining the tests, which is of growing demand, because this is one which will take a longer time, quite intensive and time consuming. But AI and ML is currently in work for detection of paraproteins within a short period of time with good results. So risk assessment, as AI is a large amounts of data, we all know, we have a lot of data from the environment, from the human studies, from the various patterns of protein predictions, and they predict the risk of exposure to chemicals and other agents. So AI can examine all these that is in the environment in the human studies. So adverse outcome pathways of analysis in conceptual framework that represents the current understanding of the relationship between a direct molecular initiating event and an unfavorable result at a biological level can help in the risk assessment for human health. So AI has overcome 5 major challenges, which is one, in a laboratory, we get the results done. The patient's sample is collected, it is transported, it is analyzed, and the reports going in a manual way takes a time consuming one and is a laborious one for the lab technologists, and it might lead to inaccurate and inconsistent results. However, AI-powered diagnosis overcomes the inaccurate and inconsistent results through manual work. And large volumes of data has to be stored in a hard disk, which is by AI, which can basically reduce the paperwork, which can be done in a digitalized way, and digitalization is AI powered. So, and also the results could be given with no time of waiting, and time consuming tasks has been overcome by AI-powered diagnostic conditions. And limited expertise, all the algorithms have been reviewed and set. So there is limited expertise that is required when it comes to manual versus the AI-powered diagnostic labs. And the cost and resource constraints could be one, which is also reduced with AI, because the initial algorithm and everything being set, it would be of great support for the AI-powered diagnostic laboratories and for the laboratory technicians. To summarize, there are various applications of AI. It decreases the pre-analytical errors. It has auto-verification of results. It has been very supportive in improving the turnaround time. With no time, we get the results in hand. We have personalized medicine care, better documentation, big data being stored, image analysis for all the ECGs, and a lot of other imaging support, increasing efficiency of the results, and so on, are the applications of AI. Whenever there is applications, and there are definitely challenges, the bias in diagnostic process definitely could machine learning lead to machine decision making, legal issues, the manpower, and the cost. There are major challenges which could be because of AI. So the opportunities could be automated urine analysis system, which you use neural network, which can analyze the whole sequences and the data, clinical decision support system for the diagnosis of the patients, predicting the progression of diseases, identifying the disease and predicting the disease earlier. So these are application challenges and opportunities of AI that is involved in today's world. And take the key points that is involved is investigating of artificial intelligence applications has proliferated, has various other fields in the environment, in the human health, in everyday day-to-day life. And comparing them based on the applications, it could be as pre-analytical, analytical, and post-analytical when you take a healthcare setting. So the novel applications, including the use of laboratory results, the diagnosis, and risk prediction, is a very important application and novel application when it comes to AI. So although there are few artificial intelligence applications which have been fully used in clinical chemistry, we need to look at the regulatory and ethical considerations, which is the most important thing, which must be addressed as a medical laboratory technologist to look into, to incorporate the artificial intelligence technologies in our routine practice. So the way forward, we have seen AI revolutionizing. The revolution in diagnostics and clinical decision making has already started, and we are using AI-enabled analysis for biochemical parameters, and we're using development and application of precision medicine, which is already way forward. And this seems to be a promising method in-house, in hospital, as well as a remote patient monitoring. With the big data that is available, the integrated set of sensing tools and data fusion algorithms, which is all powered by AI, we can detect the wide range of biochemical markers of age-related conditions, the disorders, continuous real-time and personalized manner. And the more smooth integration of digital technology and artificial intelligence, it is expected to be the future and is already the future of biochemistry, resulting in more precise diagnosis, individualized treatment regimes, and best overall healthcare results. So this is a way forward and the future of clinical biochemistry, where we are moving forward and still improvising and having a lot of biochemical parameter analysis that is done for the development and application of precision medicine. So, of course, we do have future challenges also, the ethical implications of AI in healthcare, which involves the algorithm bias, the responsible data use, need careful considerations when it comes to ethical implications because it's a large amount of patient data. So, protecting patient data, the data privacy, and the security is a most critical thing when it comes to digital tools. So, that is a future challenge which we are currently even facing and still continues to be the challenge even in future. So these are my few references that I selected and reviewed through. Thank you very much for listening. Thank you, ma'am. Dr. Shweta? Dr. Shweta?
Hello, ma'am. Am I audible?
Yes. Yes, ma'am. You're audible. Please. You're audible, ma'am. Please.
Yeah. I'd like to thank
Dr. Shweta? Yeah,
Yes,
please.
madam. Audible? Audible?
Yeah, now audible. Please go ahead.
Yeah, Dr. Rohini, madam, thank you for giving such a wonderful talk.
Thank you, ma'am. You have any questions, ma'am?
Okay, ma'am. There are myths regarding AI, no? If AI comes to lab, it will replace the manpower. So what is the opinion of madam? I just want to ask.
That is what, ma'am. Actually, the whole thing is about, there's so many saying that AI will replace human intelligence. Artificial intelligence will replace human intelligence. But we as humans who are experts will decide where the AI stands. So that is how we will take over the AI. We will know where diagnostics level, at what level do we use AI. So although we have prediction, no time and all these, it's finally the doctors, the technologists who is going to play a role. So we will stand and put a saturating level for AI. And of course, we will know in a laboratory what should be using AI and how we can prevent. And data privacy, we still have ethical challenges involved in AI. So definitely as a biochemist, the future biochemist who's coming up, we will know where the AI should stand when it comes to laboratory diagnosis.
Okay, ma'am. Thank you. Thank you so much.
I have a question, ma'am. I really have a small question. It's not a question, ma'am. I really want to know whether anything, though it has ethical things and all, you have any, put into practice or any of this AI is used in your labs, ma'am, there? We are using apps, ma'am. We are Apps actually
for?
currently devising app, using us, identifying for glucose and basic things. Our students are doing industrial training and other things, which is involving AI, ma'am. So
Okay.
those are basic things which we are involving, not into a full-fledged laboratory diagnosis.
Because we can't do, I suppose, no, ma'am. Because we need ethical.
Yes, ma'am.
This thing to
We have to
introduce
have
it.
ethical.
Are there any papers, ma'am? Because we wouldn't have done, because once before we put it, though we speak, it works well, like what you told, how to differentiate between thalassemia and microcytic anemia. It's very minor changes. Similar things. So we want, are there any studies which has compared this AI results with a manual results, so that we can say that yes, it's equally good like nothing, I suppose, ma'am. When I have gone
Yes, ma'am.
through.
Imaging has already taken.
That
AI has
I have
taken over
seen.
all the imaging part.
Images I have
Same
seen.
with goes with some of the results, like all our auto verification results. The morning we give a blood sample, by afternoon we get all the results done. So all of them are digitalized and but however, for patients with various other comorbidities, we will have to recheck because there could be other things which has to be identified. So in patient data analysis, we will have to relook at it, ma'am, in case by case conditions.
Okay, and really, I would really thank ma'am. We have heard that AA is come. AA can be put into clinical biochemistry lab. Only radiology we were really used to that, and some pathological diagnostic things I have heard, but I never knew. So I'm openly accepting that in all these fields, each pre-analytical, analytical, then risk prediction, what not. Even I was surprised to hear that it has entered into our molecular biology techniques like mRNA identification of maybe next will be SNP identification also. So it was very eye opener, and a lot of information you have shared, ma'am. And I think if it comes as we all go for accreditations one after the other, Dr. Shweta will also agree. Half of the time we are behind accreditations only, NABL, NABH, quality indicator. If AI comes into our labs, I think 90% of our human work is over. It will give perfectly errors, percentage of errors, calculation of CV percentage, reporting errors, then whatnot. All the quality indicators can be covered by, I think, artificial intelligence. I was really hope it comes, but again, like what Dr. Shweta is
The
telling,
question of where we will go is
not where we go. There should be some places to make a perfect diagnosis. I still think that there should be, like what you told, I support completely that where AI should go, humans have to decide. That is the only thing, not to completely replace it. That's what I would like to end. But it was so informative speech, ma'am, as usual. Rohini ma'am, thank you so much. And now coming to the end, anything? Okay, now I would like
just saw an question, validation process for an AI system. So when an algorithm is set, it's basically the data input. So validation process, again, it involves the quality control as we do for a laboratory, as it goes for same for AI also. The algorithms, everything has to be reviewed for the neural network has to be reviewed with quality control.
There's one more question, ma'am. In what ways does there is one more question from the participant. In what ways does AI contribute to the discovery of INC RNA and microRNA biomarkers in diseases? How does it
prediction of diseases using RNA analysis, the protein enzyme engineering. We can also use electrophoretic techniques for identification of diseases. And RNAs, the studies are being going very successful, and a lot of studies are being done on RNA prediction. Like as I said, the long non-coding RNAs and other things are also being done. And it's still in research mode. It is not, not a lot of research have come out, but it's still in research mode, and a lot of things are being done using RNA studies for prediction of diseases. More challenging, and AI has become more attractive because of predicting the diseases earlier. It's not about once a disease has come, auto verification is not of a great advantage, but predicting the earlier diseases in earlier conditions and preventing, at old age-related diseases is more prominent here.
And I think you have answered the query, ma'am. I think, see, if quantitative, it works well. If we use some semi-quantitative things like strip test and all, I don't know how much AI has come inside. I think there little subjectivity will be there.
Definitely, ma'am. They are even urine strip analysis. The studies are being done instead of urine strip analysis. When we have large volumes of samples every day, it's going to be time consuming. So, but how AI is working on it is working on the sample. Again, you have to do the sampling part. Again, they have an algorithms that is set, and they're currently working on those research which is going on now. So, which could reduce the time for us as lab technicians.
Definitely. Laboratory physicians. Yes. Thank you so much. I'd see only those two questions. Thank you so much, as usual, ma'am, Rohini ma'am, for coming and helping me out, as well as enlightening all the delegates who have definitely would have been listening to, because we all were interested in what does the latest things of AI in clinical biochemistry lab. We have as a general in medicine, AI means, as you told, radiology, diagnostic. Mainly we hear in pathology and all, but in molecular biology as well as it's good to hear, ma'am. Thank you so much, ma'am, for joining us. And anyhow, it's though being a virtual platform, we would like to honor you in the virtually by a certificate as well as a memento.
Thank you.
Next, I would like to thank you. Next, I would like to honor our chairperson, Shweta, Dr. Kulkarni Shweta also. I once again thank both of you. In spite of your busy schedules, you have taken time to help us and share your knowledge with all of us. Thank you very much, ma'am.
Thank you. Thank you, ma'am. Thank you very much. And
Dr.
thank you,
Shweta,
Dr.
thank you.
Shweta.
Bye, ma'am.
Thanks.
Welcome to Vinayaka Missions Group, where education meets healthcare excellence. Our academic landscape spans across 13 colleges and 11 schools, situated in 4 vibrant campuses. We are proud to serve the Union Territory of Puducherry and extend our reach to Salem and Chengalpet districts in Tamil Nadu. Pioneers in private medical education, we established the first private medical college in Puducherry back in 2000. Today, we operate 2 medical colleges in Puducherry, where we provide healthcare services to an astonishing 4.5 lakh outpatients and over 50,000 inpatients annually. We manage 9 rural health centers, where we attend to approximately 1,000 patients every day, ensuring quality healthcare for all. And now, as we embark on our next level in healthcare, we are proud to introduce the Vinayaka Karkinos Oncology Institute, or VCOI, in collaboration with Karkinos Healthcare. Karkinos Healthcare is a purpose-driven, technology-led oncology platform, revolutionizing cancer care in India, with a primary focus on early detection, precise diagnosis, patient care, and groundbreaking research. Through setting up of the Vinayaka Karkinos Oncology Institute, the aim is to bring comprehensive cancer care closer to the communities under the distributed cancer care network. Our mission: early detection programs through community screening programs, identifying high-risk patients and guiding them throughout their entire care journey. The facility will be equipped with state-of-the-art diagnosis and treatment options. And as part of our commitment to the people of Puducherry, Vinayaka Karkinos Oncology Institute aims to screen nearly 1 lakh population within the next few months. This collaboration between Karkinos Healthcare and Vinayaka Missions Group, including Vinayaka Missions Research Foundation, in setting up of the Vinayaka Karkinos Oncology Institute, VCOI, in Puducherry and Salem, shall be a testament where education, research, and affordable and accessible cancer care come together for a brighter, healthier future. Welcome to Vinayaka Missions Group, where education meets healthcare excellence. Our academic landscape spans across 13 colleges and 11 schools, situated in 4 vibrant campuses. We are proud to serve the Union Territory of Puducherry and extend our reach to Salem and Chengalpet districts in Tamil Nadu. Pioneers in private medical education. We established the first private medical college in Puducherry back in 2000. Today, we operate 2 medical colleges in Puducherry, where we provide healthcare services to an astonishing 4.5 lakh outpatients and over 50,000 inpatients annually. We manage 9 rural health centers, where we attend to approximately 1,000 patients every day, ensuring quality healthcare for all. And now, as we embark on our next level in healthcare, we are proud to introduce the Vinayaka Karkinos Oncology Institute, or VCOI, in collaboration with Karkinos Healthcare. Karkinos Healthcare is a purpose-driven, technology-led oncology platform, revolutionizing cancer care in India, with a primary focus on early detection, precise diagnosis, patient care, and groundbreaking research. Through setting up of the Vinayaka Karkinos Oncology Institute, the aim is to bring comprehensive cancer care closer to the communities under the distributed cancer care network. Our mission: early detection programs through community screening programs, identifying high-risk patients and guiding them throughout their entire care journey. The facility will be equipped with state-of-the-art diagnosis and treatment options. And as part of our commitment to the people of Puducherry, Vinayaka Karkinos Oncology Institute aims to screen nearly 1 lakh population within the next few months. This collaboration between Karkinos Healthcare and Vinayaka Missions Group, including Vinayaka Missions Research Foundation, in setting up of the Vinayaka Karkinos Oncology Institute, VCOI, in Puducherry and Salem, shall be a testament where education, research, and affordable and accessible cancer care come together for a brighter, healthier future. Welcome to Vinayaka Missions Group, where education meets healthcare excellence. Our academic landscape spans across 13 colleges and 11 schools, situated in 4 vibrant campuses. We are proud to serve the Union Territory of Puducherry and extend our reach to Salem and Chengalpet districts in Tamil Nadu. Pioneers in private medical education. We established the first private medical college in Puducherry back in 2000. Today, we operate 2 medical colleges in Puducherry, where we provide healthcare services to an astonishing 4.5 lakh outpatients and over 50,000 inpatients annually. We manage 9 rural health centers, where we attend to approximately 1,000 patients every day, ensuring quality healthcare for all. And now, as we embark on our next level in healthcare, we are proud to introduce the Vinayaka Karkinos Oncology Institute, or VCOI, in collaboration with Karkinos Healthcare. Karkinos Healthcare is a purpose-driven, technology-led oncology platform, revolutionizing cancer care in India, with a primary focus on early detection, precise diagnosis, patient care, and groundbreaking research. Through setting up of the Vinayaka Karkinos Oncology Institute, the aim is to bring comprehensive cancer care closer to the communities under the distributed cancer care network. Our mission: early detection programs through community screening programs, identifying high-risk patients and guiding them throughout their entire care journey. The facility will be equipped with state-of-the-art diagnosis and treatment options. And as part of our commitment to the people of Puducherry, Vinayaka Karkinos Oncology Institute aims to screen nearly 1 lakh population within the next few months. This collaboration between Karkinos Healthcare and Vinayaka Missions Group, including Vinayaka Missions Research Foundation, in setting up of the Vinayaka Karkinos Oncology Institute, VCOI, in Puducherry and Salem, shall be a testament where education, research, and affordable and accessible cancer care come together for a brighter, healthier future. Welcome to Vinayaka Missions Group, where education meets healthcare excellence. Our academic landscape spans across 13 colleges and 11 schools, situated in 4 vibrant campuses. We are proud to serve the Union Territory of Puducherry and extend our reach to Salem and Chengalpet districts in Tamil Nadu. Pioneers in private medical education. We established the first private medical college in Puducherry back in 2000. Today, we operate 2 medical colleges in Puducherry, where we provide healthcare services to an astonishing 4.5 lakh outpatients and over 50,000 inpatients annually. We manage 9 rural health centers, where we attend to approximately 1,000 patients every day, ensuring quality healthcare for all. And now, as we embark on our next level in healthcare, we are proud to introduce the Vinayaka Karkinos Oncology Institute, or VCOI, in collaboration with Karkinos Healthcare. Karkinos Healthcare is a purpose-driven, technology-led oncology platform, revolutionizing cancer care in India, with a primary focus on early detection, precise diagnosis, patient care, and groundbreaking research. Through setting up of the Vinayaka Karkinos Oncology Institute, the aim is to bring comprehensive cancer care closer to the communities under the distributed cancer care network. Our mission: early detection programs through community screening programs, identifying high-risk patients and guiding them throughout their entire care journey. The facility will be equipped with state-of-the-art diagnosis and treatment options. And as part of our commitment to the people of Puducherry, Vinayaka Karkinos Oncology Institute aims to screen nearly 1 lakh population within the next few months. This collaboration between Karkinos Healthcare and Vinayaka Missions Group, including Vinayaka Missions Research Foundation, in setting up of the Vinayaka Karkinos Oncology Institute, VCOI, in Puducherry and Salem, shall be a testament where education, research, and affordable and accessible cancer care come together for a brighter, healthier future. Welcome to Vinayaka Missions Group, where education meets healthcare excellence. Our academic landscape spans across 13 colleges and 11 schools, situated in 4 vibrant campuses. We are proud to serve the Union Territory of Puducherry and extend our reach to Salem and Chengalpet districts in Tamil Nadu. Pioneers in private medical education. We established the first private medical college in Puducherry back in 2000. Today, we operate 2 medical colleges in Puducherry, where we provide healthcare services to an astonishing 4.5 lakh outpatients and over 50,000 inpatients annually. We manage 9 rural health centers, where we attend to approximately 1,000 patients every day, ensuring quality healthcare for all. And now, as we embark on our next level in healthcare, we are proud to introduce the Vinayaka Karkinos Oncology Institute, or VCOI, in collaboration with Karkinos Healthcare. Karkinos Healthcare is a purpose-driven, technology-led oncology platform, revolutionizing cancer care in India, with a primary focus on early detection, precise diagnosis, patient care, and groundbreaking research. Through setting up of the Vinayaka Karkinos Oncology Institute, the aim is to bring comprehensive cancer care closer to the communities under the distributed cancer care network. Our mission: early detection programs through community screening programs, identifying high-risk patients and guiding them throughout their entire care journey. The facility will be equipped with state-of-the-art diagnosis and treatment options. And as part of our commitment to the people of Puducherry, Vinayaka Karkinos Oncology Institute aims to screen nearly 1 lakh population within the next few months. This collaboration between Karkinos Healthcare and Vinayaka Missions Group, including Vinayaka Missions Research Foundation, in setting up of the Vinayaka Karkinos Oncology Institute, VCOI, in Puducherry and Salem, shall be a testament where education, research, and affordable and accessible cancer care come together for a brighter, healthier future. Welcome to Vinayaka Missions Group, where education meets healthcare excellence. Our academic landscape spans across 13 colleges and 11 schools, situated in 4 vibrant campuses. We are proud to serve the Union Territory of Puducherry and extend our reach to Salem and Chengalpet districts in Tamil Nadu. Pioneers in private medical education, we established the first private medical college in Puducherry back in 2000. Today, we operate 2 medical colleges in Puducherry, where we provide healthcare services to an astonishing 4.5 lakh outpatients and over 50,000 inpatients annually. We manage 9 rural health centers, where we attend to approximately 1,000 patients every day, ensuring quality healthcare for all. And now, as we embark on our next level in healthcare, we are proud to introduce the Vinayaka Karkinos Oncology Institute, or VCOI, in collaboration with Karkinos Healthcare. Karkinos Healthcare is a purpose-driven, technology-led oncology platform, revolutionizing cancer care in India, with a primary focus on early detection, precise diagnosis, patient care, and groundbreaking research. Through setting up of the Vinayaka Karkinos Oncology Institute, the aim is to bring comprehensive cancer care closer to the communities under the distributed cancer care network. Our mission: early detection programs through community screening programs, identifying high-risk patients and guiding them throughout their entire care journey. The facility will be equipped with state-of-the-art diagnosis and treatment options. And as part of our commitment to the people of Puducherry, Vinayaka Karkinos Oncology Institute aims to screen nearly 1 lakh population within the next few months. This collaboration between Karkinos Healthcare and Vinayaka Missions Group, including Vinayaka Missions Research Foundation, in setting up of the Vinayaka Karkinos Oncology Institute, VCOI, in Puducherry and Salem, shall be a testament where education, research, and affordable and accessible cancer care come together for a brighter, healthier future. Welcome to Vinayaka Missions Group, where education meets healthcare excellence. Our academic landscape spans across 13 colleges and 11 schools, situated in 4 vibrant campuses. We are proud to serve the Union Territory of Puducherry and extend our reach to Salem and Chengalpet districts in Tamil Nadu. Pioneers in private medical education. We established the first private medical college in Puducherry back in 2000. Today, we operate 2 medical colleges in Puducherry, where we provide healthcare services to an astonishing 4.5 lakh outpatients and over 50,000 inpatients annually. We manage 9 rural health centers, where we attend to approximately 1,000 patients every day, ensuring quality healthcare for all. And now, as we embark on our next level in healthcare, we are proud to introduce the Vinayaka Karkinos Oncology Institute, or VCOI, in collaboration with Karkinos Healthcare. Karkinos Healthcare is a purpose-driven, technology-led oncology platform, revolutionizing cancer care in India, with a primary focus on early detection, precise diagnosis, patient care, and groundbreaking research. Through setting up of the Vinayaka Karkinos Oncology Institute, the aim is to bring comprehensive cancer care closer to the communities under the distributed cancer care network. Our mission: early detection programs through community screening programs, identifying high-risk patients and guiding them throughout their entire care journey. The facility will be equipped with state-of-the-art diagnosis and treatment options. And as part of our commitment to the people of Puducherry, Vinayaka Karkinos Oncology Institute aims to screen nearly 1 lakh population within the next few months. This collaboration between Karkinos Healthcare and Vinayaka Missions Group, including Vinayaka Missions Research Foundation, in setting up of the Vinayaka Karkinos Oncology Institute, VCOI, in Puducherry and Salem, shall be a testament where education, research, and affordable and accessible cancer care come together for a brighter, healthier future.
Good afternoon to everyone. I welcome you all to the BMSeCON 2023 YRS PG session. The session starts now. For the oral presentation, total presentation time is 9 minutes. So in which 7 minutes is your presentation time and 2 minutes is for discussion. So please stick to the time. Thank you. For the first presentation, before starting the presentation, I would like to introduce the judges. Judges Dr. Kashish Singh, Assistant Professor, Department of Anatomy, MMCMSR, Ambala, Haryana, and Dr. Chandrasekhar, Professor, Physiology, COE, Balaji Medical College, and Dr. Arul Vijayavani, Associate Professor and Head, Department of Biochemistry, JIPMER. I welcome you all, the judges. So I thank you for your wonderful time judging the session. So for the first presentation, I would like to call Parneet Sandhu. Parneet Sandhu, you can share your slides now.
Yes, sir. Good afternoon, sir. Am I audible?
Yeah, you're audible.
Is it okay, sir?
Yeah, okay. You can start now.
Okay. Sorry. Good afternoon. I'm Dr. Praneeth from GMC at Chandigarh. The topic of the present study is histogenesis of human fetal cerebellum during the second trimester of gestation. Cerebellum is one of the most important structures in the posterior cranial fossa, and it plays an important role in the motor learning, coordination, precision, and some cognitive functions as well. It is one of the first structures in the brain that starts to differentiate, but it is the last one to finish maturation. Cerebellar cortex is divided into molecular layer, middle Purkinje cell layer, and inner granular layer. External granular layer consists of indifferent cells, and it may convert into nerve cells or glial cells. With respect to the external features of the cerebellum, the fissures of the cerebellum, vermis, and the hemispheres, they appear by the second trimester of pregnancy. The aims and objectives of this study is to observe the normal development of the cerebellum and to study the pattern of development of the fetal cerebellum. We regularly perform fetal autopsies in our department between the age group of 12 to 28 weeks as per standardized protocol, and detailed antenatal history was taken in all the cases. 20 fetuses between the age group of 12 to 28 weeks were included in this study. Macerated fetuses and fetuses with neural tube defects were excluded. After fixing the fetus, we dissected the cerebellum, and then it was subjected to routine histological procedure. We used hematoxin and eosin and periodic acid shift stains. The following observations were made. This is an image of the dissected cerebellum from a 20-week-old fetus. We can see the cerebellar hemispheres, vermis, and fissures can also be seen. This is a histological appearance of the cerebellum under H&E stain and 4x magnification. The first age group under study was 12 to 16 weeks, in which under 10x magnification and H&E stain, we saw the external granular layer. Now, figure number 4, we can see under 12x magnification, the external, the magnified size of external granular layer with indifferent cells. The second age group was 16 to 20 weeks of gestation, in which we observed very few Purkinje cells, and they were rounded in, I'll just zoom, they were rounded in appearance. Under past staining and 20x magnification, basket and stellate cells were beginning to differentiate. The next age group was 20 to 24 weeks, in which there was increase in population of Purkinje cells in figure number 7 under 20x magnification, and under PAS we could see the different layers of the cerebellum. The last age group was 24 to 28 weeks of gestation, in which we saw Purkinje cells were increased in number, and a discrete layer of the Purkinje cell layers was seen under H&E magnification. Under PAS staining, we can see a Purkinje cell layer with the dendrites as well. So this present study covered the histological observations during the second trimester of gestation. According to previous data in the literature, in the age group of 12 to 16 weeks, cerebellar surface was covered by external granular layer, and in 16 to 20 weeks, stellate pellet and basket cells were observed. In the present study also, we made similar observations. Then there was another study in which around 22 to 24 weeks of gestation, there was clear demarcation between different layers of the cerebellum. However, in our study, we saw this change between 20 to 24 weeks along with the oval to round shaped Purkinje cells. We did not see the typical appearance of the Purkinje cell, which is flower shaped in the study, because according to the literature, it starts to appear after 30 to 32 weeks. This is a table summarizing various studies in the literature as well as our study. To conclude, since cerebellum is the first structure in the brain which starts to differentiate and last one to maturation, so this long period of development makes it a very vulnerable structure for various developmental defects or congenital defects. And most of the data that is available in the literature is regarding the morphometry or external features of the cerebellum. There are very few studies available which are based on the histological or microscopic levels. There are various congenital cerebellum abnormalities: hypoplasia, dysplasia of cerebellum, Dandy-Walker syndrome. And the data that is available in the literature is based mostly on the ultrasound and MRI observations. However, these investigations, they have limitations. So therefore, I would like to say that performing an autopsy and then studying the specimens at microscopic level provides us with a better picture of the conditions, and this information can be shared with the neurological department, and cerebral anatomy can be explored in future. These are the references. Thank you.
Thank you, Pani. The judges can ask their questions.
Yeah, you can share. 1 minute. Just need you the.
Yeah, respected judges can ask the questions.
Sorry, ma'am, I can't hear. Hello? Can you please repeat the question?
Yes. You may go ahead.
Dr. Kaushik Singh. Hello. Madam, we cannot able to hear you.
Hello, sir. Can you please repeat the question?
1 second, I'll call the judge.
Okay,
Yes,
sorry.
please. Pani, please wait.
Yes, sir.
She has some technical problem. Yeah.
No problem.
Yeah. Okay. The other judges can also ask questions if you have any queries.
Hello,
Yeah,
can you hear me now?
Yes, ma'am, you can ask the question.
Dr. Parneet?
Yes, ma'am. Good afternoon.
Yeah,
ma'am.
good afternoon. I just want to know, you have selected the fetuses from the age group of 12 to
28 weeks. Yes, ma'am.
20. Can you just tell me what was the time period when you received these fetuses, or what they're the, I mean, how did you select the fetuses?
Ma'am, we perform fetal autopsies in routine. So every second day, third day, we keep receiving fetuses from the gynae department from our college only. But I started working on this study in May.
Actually, you should just
excluded the I only included the normal fetuses as per the ultrasound report, and then we performed the autopsies.
Yeah, that I know. I know GMCH. That's my alma mater only.
Okay.
So that is why I'm asking, was there any special criteria to select the fetuses? Because it should be either a continuous selection, otherwise the bias will come. How do you select?
Continuous selection. I started working on this study in May.
Okay, that was when you started working. You just selected the normal fetuses.
Normal fetuses, yes, ma'am.
Grossly, which were normal.
Yes, ma'am.
Okay. And the presentation is good. The slides were good, but one thing I would like to just see in your histology pictures.
Yes, ma'am.
When you were pointing out, you should have some more pointers. Pictures are good. Histology training is good. All the layers are visible, but you should because it's a new thing. Not everybody is used to all these slides of histogenesis of cerebellum, that to fetal cerebellum, no?
Yes, ma'am. I'll keep that in mind.
You should have some more pointers to point out the layers correctly and accurately so that everybody can see properly. Okay?
Okay, ma'am. Okay, ma'am.
I think that's all.
Thank you, Parneet Sandhu. Next presentation is by Dr. Ankita Kishore Bhai Davi.
Hello. Yes, sir. I'm audible.
Yeah, you're audible now. You can share your slides.
Okay, sir. Hello. Good afternoon, everyone.
You can start.
Myself, Dr. Ankita Dapi from the BMSeCON Medical College, Ahmedabad. I am a second-year resident. Today, I am presenting a topic on the cytogenetic analysis of couples with recurrent pregnancy loss in Gujarat region. It is a retrospective study. Let's start with the introduction. Recurrent pregnancy loss is classically defined as the occurrence of 3 or more consecutive abortions. However, the American Society of Reproductive Medicine has recently defined recurrent pregnancy loss as 2 or more abortions. It is a clinically recognized pregnancy, involuntary ending before 20 weeks of gestation. It affects approximately 10 to 15% of couples of all clinically recognized pregnancies. There are several etiological factors considered for recurrent abortion, including chromosomal anomalies like genomic instability, single gene mutation, epigenetic changes, X chromosome inactivation, and sperm chromosomal rearrangement. The carrier parent with a balanced translocation may face the problem of infertility and give birth to a malformed baby. They are responsible for 2 to 5% of recurrent abortion. It has also been reported that 50 to 60% spontaneously aborted product of conception have chromosomal anomalies. These are the types of the chromosomal anomalies, mainly being structural and numerical. Structural being translocation, inversion, deletion, polymorphism, etc. Numerical being aneuploidy, hyperploidy, hypoploidy, polyploidy. And these chromosomal abnormalities in the couple are considered to be strong etiology for recurrent abortion. Thus, the parental karyotyping is recommended to assess the genetic cause of RPL. AVMC objective of this study is to find the prevalence of chromosomal abnormalities in couples with RPL and to encourage knowledge about genetic counseling, to compare this study with the other studies and to contribute in national database preparation. A material method which I used to prepare this study, it is a retrospective study which carried out on the patient record of past 5 years from the May 2018 to April 2023 at Genetic Laboratory, BMSeCON Medical College, Ahmedabad. Cytogenetic analysis of couples with RPL was observed. patients were referred from various hospitals to this our genetic laboratory. Their complete case history was recorded, and chromosomal analysis of all patients done according to standardized laboratory technique, and karyotype was prepared following the guideline of International System for Human Cytogenic Nomenclature. For the result, I have observed 35 couples, in which 8 couples having different structural chromosomal abnormalities. No numerical chromosomal abnormality was observed. Here is the pie chart of these patients. Here we can see 50% having the polymorphic variants, 12% cases of balanced reciprocal translocation, 13% being mosaic pattern with balanced reciprocal translocation, and 25 being the pericentric inversion. Here this table 1 showing the age-wise distribution of the individuals, and from this observation, the affected female partner mean age group is 27.8 year, and affected male partner having mean age group was 30.5 year. In this table 2, here there is a distribution based on the number of abortion and in the number of couples. So the affected couples having mean number of abortion is 3.13. Here is the table 3 showing the distribution of pregnancy loss based on this trimester. So commonly most common are the first trimester abortions, and in the third trimester history of empty was present. And it is observed that with couples with a chromosomal abnormalities had history of abortion all 3 trimester, but most common was the first. The mean number of first trimester abortions is the 1.88, 15 being out of 25. Here this table shows the distribution of chromosomal abnormalities in the female partner. There is a number of abortion in the second column, then the aberration type having the which chromosomal abnormalities, then consequences of the marriage relation with it or not, then associated condition in 2 patients, one with mosaic pattern having hypothyroidism history, and one with polymorphic variation having a history of asthma. Here in the second table, here is showing the data of the male partner. So from this above observations, in 2 cases there was associated history disease. There was chromosomal abnormalities present in female partner. In case of conspiracy of the marriage, there was in 1 male partner chromosomal abnormality was present. Here showing the images of the normal karyotype. First is showing of the female, and second is showing the male 46XY. Now in this first there is in male partner there is large satellite region on the short arm of chromosome 22. Here in this case in the male partner having inversion of Y chromosome, pericentric inversion of the Y chromosome. Here in the third male partner having increased heterochromatin of the chromosome 9. Here in the sixth in male partner having increase in the length of centromeric heterochromatin on the chromosome 21. These all are related with the bad obstetric history. Here now of the female partner karyotypes. In this case, there is pericentric inversion of the chromosome 9. Here that is reported that 1 to 3% of the couple with history of recurrent spontaneous miscarriages having inversion of the chromosome 9. Then there is in this case, there is balanced reciprocal translocation between the chromosome 13 and chromosome 17. Then there is in this case, female partner having the increased length of centromeric heterochromatin on the chromosome 21. And here in this case, there is increased heterochromatin of the chromosome 9. And this is the one case in which there is a mosaic pattern of balanced reciprocal translocation was seen, being the mosaic pattern for the 46XS and 45XS translocated. There is translocation between the chromosome number 18P and 20Q, and there is this couples having one partner with balanced reciprocal translocation having increased risk of miscarriages. It's about the discussion. So here in the discussion,
Ankita, you can conclude already.
Okay. Relevance is seen, and in the conclusion, RPL is the constant reproductive problem. This study will help in the providing genetic counseling and to adopt other ways of pregnancy. And this study will guide the clinician for consideration to rule out the genetic causes. Thank you. And here are the references which I have used for this.
Thank you, Dr. So just can ask the questions.
Hello.
Hello, good afternoon, ma'am.
Dr. Ankita. It's a good topic. But one thing I would like to ask, how much do you think is the, relevance of this topic clinically? What did you feel? It's a retrospective study, but
Yes,
if
ma'am,
you
it is
have to
retrospective
counsel,
study.
if you have to counsel the patients and you have to counsel the parents, how relevant or how useful you think it actually will be?
Okay, ma'am. I will show them to this data that if there is no other problem after screening in the in case of the infertility, then I will show them this data to help to educate them that the recurrent karyotyping was important in case of recurrent pregnancy loss or in case of infertility.
So you think you will be able to counsel the patients according to this data?
I will try them.
You have to simplify it so that the clinician can use and even you can use.
Okay, ma'am.
And
Yes, ma'am.
try to just summarize the thing. You were just more reading the slides. Try to explain it in a simpler manner. Okay.
Okay, ma'am.
Otherwise, it's a good study. Okay. Thank you.
ma'am.
Thank you, Dr. Ankita.
Thank you, sir.
Next presentation is by Dr. Nicolasius. Dr. Nicholas, you can share your screens.
Sir, is it visible? Sir, is it audible and visible? Sir, can you hear me? Sir, can you hear me?
Yeah, we can hear you. Carry on.
Okay. Sir, I am Dr. Niklesh in the Department of Anatomy, Jipper, Puducherry. My topic of presentation is clinical significance associated with anatomical variations of paranasal air sinuses, a computer tomographic study. So coming to the introduction. So in the introduction, why we are choosing paranasal air sinuses? This is most common to exhibit lot of anatomical variations. And these variations are asymptomatic and incidentally detected by computer tomography. So the clinical variations and the landmarks are most important for the radiologist as well as surgeons. So why we are choosing CT instead of MRI? CT is the gold standard investigation of choice in delineating the abnormal anatomy and the variations for the surgeon, treatment planning, and for the surgical intervention. Coming to the aims and objectives. So my aim is to identify the anatomical variations of paranasal air sinus and to understand the clinical significance associated with these variations. Coming to the materials and methods, it is of 280 CT scans has been archived in the PACS in the radio diagnosis at JIBMER. So axial and coronal sections was taken for all the four sinuses, frontal, maxillary, ethmoid, and sphenoidal air sinus. Images were reconstructed in sagittal plane to analyze the pneumatization pattern of sphenoidal air sinus. So this is a CT image which I'm showing. It is used in JIBMER, and these two are the softwares which are used to view the images. Coming to the results. So it is of 280 scans. We have male of 142 and female of 138. So frontal air sinus, we are looking for aplasia, hypoplasia, and septations. In my case, it was 4 cases in right and left together. We have 560. So it is 4 present in hypoplasia, 22 of hypoplasia, and 46 in septations. Now you can see the image where you can see here the frontal sinus aplasia, and you can see frontal sinus hypoplasia in the second image, and the third image shows the septations in the frontal sinus, both right and left side. In maxillary air sinus, we looked for hypoplasia and septations, whereas aplasia, I didn't get any case. So 13 cases was present as hypoplasia and 35 cases of septations. So you can see maxillary sinus hypoplasia in the image, and you can see septations on the right side image. Coming to the osteometal complex, conchobulosa. What do you mean by conchobulosa? Conchobulosa is nothing but pneumatization. If it is pneumatized, we call it as conchobulosa of the pneumatization of middle turbinate. If the middle turbinate is pneumatized as lamellar, extensive, and bulbous, in this we got more frequency as lamellar as 32.5% and least as bulbous. So you can see this is a lamellar type of conchobulosa, pneumatization of middle turbinate, and this is an extensive type of conchobulosa, and this is bulbous type of conchobulosa. Next, uncinate process, pneumatization, agar-Neissery cell and Haller cell. So Haller cell is nothing but infraorbital cells. So agar-Neissery cell is anterior ethmoidal cell. So we have Neissery frequency percentage of 45.35 and Haller cells of 17.85. So you can see agar-Neissery cell. So it is anterior ethmoidal cell, and this is the Haller cell infraorbital cells. So this is the pneumatization of uncinate process. Paradoxical middle turbinate, pneumatized superior turbinate, and we have extreme turbinate called as supreme turbinate. So in frequency we have 1.42%. No study encountered supreme turbinate till yet. So we have encountered supreme turbinate. So we can see this is paradoxical middle turbinate. Next, we can see the pneumatization of superior turbinate, and this on the right side image shows the supreme turbinate. So pneumatization of anterior clinoid process, pterygoid plate, dorsum sellae, greater wing of sphenoid, because sphenoid sinus is more difficult to encounter during the surgical procedures. So pneumatization of anterior clinoid process is 9%, pterygoid plate is 8.75%, dorsum sellae is 1.96%, whereas greater wing of sphenoid is 3.75%. So you can see anterior clinoid process is pneumatized here, and this is pterygoid plate pneumatization. Then the next, we can see the greater wing of sphenoid pneumatization in the second image. And this is the sagittal reconstruction which we have done for pneumatization of dorsum sellae. You can say dorsum sellae is pneumatized. So 1OD cell, what is 1OD cell and protrusion of ICA? 1OD cell is 18.03%. Protrusion of internal carotid artery is 3.39%. So you can see this is 1OD cell. So what is 1OD cell? 1OD cell is a posterior most ethmoidal air cell which extends to lie on the supralateral to the sphenoidal air sinus, which is closely approximated to the optic nerve internal carotid artery. So this is the 1OD cell on right and left. So this is the extension of protrusion of internal artery into the sphenoidal air sinus. So this one more image, you can see the protrusion of internal carotid artery on the right side. So septations is present or absent. Midline septa is 96 cases, multiple septa is 43 cases, no septa in 4 cases. So you can see the midline septa and then the multiple septations on the sphenoidal air sinus, and there is no septations present in this case. Coming to cellular, post-cellular, and this is the pneumatization pattern. For this pneumatization pattern, I'm going to do the sagittal reconstruction. Out of all, cellular is the most common on 45.71%.
Dr. Nikhilesh, you are running out of time. Kindly please sum up.
Okay.
Thank you.
This is cellular, pre-cellular pneumatization. So you can see this is the cellular pneumatization pattern. This is post-cellular pneumatization pattern. So coming to Delano classification, this is type 1, type 2, type 3, type 4. This is approximately to the optic nerve. So this is you can do type 1, type 2, type 3, and type 4. So next, coming to the discussion, we have discussion present study. We have encountered a lot of differences, agar-Neissery and pneumatization of the alginate process and paradoxical middle turbinate. And this is 1OD cell and anterior clinoid process pneumatization also we have encountered more than the other studies. And de novo classification, type 1 is R predominance. And this post-cellular is the second most predominant, whereas other studies told pre-cellular is the second most predominant. So clinical significance, you can see optic nerve internal process, greater wing, pterygoid plate, median canal, foramen rotundum around sphenoid sinus. So this is the most important one for any approach to this transsphenoidal surgeries. So this is a transplant surgery. So a lot of clinical variants, headache, blindness, intracranial bleeding can be there, CSF leak. So these are the clinical significances we are talking. And there are a lot of studies we have encountered, giant conchobulosa, cavernous sinus, carotid injury, all these things. So conclusion, so knowledge of this important for radiologists and surgeons for treatment. Mandatory preoperative CT scan must be done for any surgeries encountering to the face as well as transpenoidal surgeries. So this study will help the surgeons as well as radiologists to understand better anatomy and as well as the variants and their clinical significance. So always the radiologists and surgeons must be in a sync to understand better knowledge. Thank you. These are my references.
Thank you so much. Very good presentation. Now the paper is open for the discussions. Any questions, please? Okay. Thank you so much. Next, we are going to the next presenter. Ilakya.
Yes, sir. Am I
Now
audible?
the board is for yours. You can present your presentation. Best of luck.
Thank you, sir. Good afternoon, everyone. I am Ilakya. Today I am going to present my topic titled Short-Term Effects of Transcutaneous Auricular Vagal Nerve Stimulation on Heart Rate Variability, Influence of Gender and BMI. Auricular vagal nerve stimulation.
Ilakya, can you share your full screen in a full mode, please?
Yes, sir.
Yeah.
Now it's visible?
No, you have to click your full mode screen. Just click your full mode.
Yes, sir.
In here, the 68%. No, near it is 68% is showing, no? And just click before that one screen, one icon is there. Just click that one. Yeah. Before. 68, that before. No. No, you can see the 89, no? Just on the left side. You click that one. Yeah.
Now it's visible, sir.
It's visible, but you did not share your full mode screen.
Try to click the slideshow.
Slideshow. Yeah, from slideshow. You can click the slideshow. Just click the slideshow.
So now it's visible.
Okay, anyway, your question
is near the size, madam. 89%. Just left to that.
Now it's visible, sir.
It's visible, madam. You can just click
it's visible, but the problem is you're not going to the slideshow.
The slide is visible.
Sir, I'm on my slideshow only. I clicked that.
Click that.
Yeah. That's click that.
Click it.
Yeah, wait.
Now it's fine, sir.
No, it's not come. Again, click it once again. Yes, your slideshow is, I think, not working. Okay, you go ahead, madam. You have size and you go ahead.
Okay, sir. I will start, sir.
Yeah.
My topic is short-term effects of transcutaneous auricular vagal nerve stimulation on heart rate variability, influence of gender and BMI. Introduction. Vagal nerve stimulation is a promising bioelectronic technology approved by FDA, used in numerous clinical conditions such as seizure, depression, headache, as an alternative non-pharmacological and neuroimmunomodulatory intervention. TENS has been applied in many studies for stimulating the auricular branch of vagus nerve non-invasively, and also the molecular mechanism and anatomical neural network behind the autonomic modulation are yet to be explored. It is a simple and robust prognostic tool in assessing the cardiovascular health. TAVAN's technique has been promising results in clinical conditions, but 25 to 35% of people have not received any benefit following TAVANs. The reasons are said to be varying from simple physiological factors such as age, gender, BMI to the complex neuroendocrine immune network mechanism. In this present study, the role of physiological factors like gender, BMI on the of parents and HRV were analyzed in young healthy volunteers. Aim to assess the HRV following TAVANS in males and female. To assess the correlation between BMI.
Madam, you use the slide, madam.
Sir?
Your slide. Go to the slide. You're on the first slide itself.
Okay, sir. Now, sir?
Your slides are not moving, ma'am.
Go to the next slide. Next slide. You're on the first slide itself. Come to the aim slide. Fourth slide.
Let's change the slide.
Now it's moving, sir.
Not moving.
No, madam.
Sir, I think it is for us only. We are not able to see. You can close the PowerPoint and reopen it again, madam. You
Okay,
can try
ma'am.
it actually. It is not
Okay,
moving
ma'am.
for us.
இருக்கு pendrive வந்து இதுல இருக்கு. So now it's visible.
Okay, it's seen, madam. Try the slideshow. Yes.
You can change the slide.
Your slideshow is
It's
not
okay,
working.
sir.
Yeah, you go ahead.
Can I go ahead?
Yeah, you go ahead, madam.
Okay. Aim of the study to assess the heart rate variability following TAVANS in male and female healthy volunteers to assess the correlation between BMI and change in HRV parameters before and after TAVANS procedure. The methodology, it's a part of my program and there are no relevant articles regarding this topic. So we did it as a pilot study. It's a single arm interventional convenient sampling. 22 health young volunteers were involved at Department of Physiology, JIPMER. Inclusion criteria, healthy young individuals, both male and female, yearly proliferative phase of menstrual cycle, age group between 18 to 35 years, BMI of 18 to 22.9 were included. Exclusion area, known case of DM, hypertension, CVD, neurological disorders, asthma, COPD, active peptic ulcer disease, and also those were receiving chronic medications were excluded from the study. After obtaining the approval,
Dr. Ilakya, we are not hearing you. Dr. Ilakya? Dr. Ilakya, we are not hearing you.
sir, can I continue?
Yeah, man, just finish it fast. Yeah, they're running out of time also.
From where I can continue?
After the medicine method.
Methodology, okay. A stimulus with 30 Hz frequency, 200 millisecond and intensity of 0 to or less than or equal to 8 milliamps for given for 15 minutes over Simba concha or cavum concha of the left ear. HRV was done using Biopac MP36 and were analyzed using Cubios 3.5 version before and after TAVANS. As per task 4 guidelines, BMI was calculated using Quicklets index. Statistical analysis, independent student t-test was done to assess the anthropometric parameter. Paired t-test was done to assess the significant changes in the HRV between the pre-stimulation and post-stimulation. Using the partial correlation between BMI and HRV, the parameters were assessed after controlling for age. P-values less than 0.05 was considered statistically significant. Results comparison of anthropometric parameters between male and female healthy volunteers. This table shows age and BMI have no significant difference between the two groups. So it makes the other parameters comparable to this age and BMI. In this table, we compare the HRV for both males and females and also for both the genders. Pre-vagal stimulation, post-vagal stimulation, HRV was assessed, and this shows a increase in HRV parameters, which indicates mainly a parasympathetic dominant. The p-values were more significant in male group. There is an increase in HRV parameters following TAVANS in both gender, but there is a significant increase in parasympathetic indices such as mean RR, SDNN, total power, ST2, and decrease in mean heart rate, sample entropy, approximate entropy significantly in male. This table shows a partial correlation with BMI. It was also the same calculated in both male, female, and also in both genders. This table shows a negative correlation, and the values were also again more significant in male group. This indicates that the changes in HRV parameters such as RMSST, NN50, PNN50, LFHF, and ST1 were having strong negative correlation in males with BMI than females, following an increased vagal tone following TAVANS. Discussion, time domain indices. There is a significant increase in mean RR, SDN, and decrease in mean heart rate in males. In previous study, MD Koch et al. Showed a significant change in SDNN after vagal nerve stimulation in females. The change in SDNN varies with time. The finding of our study are in contrary with the result of MD Koch et al. In their study, they didn't mention about the phase of menstrual cycle, and they might done the study during the phase of proliferative phase, where there will be more parasympathetic activity. Frequency domain indices. Our study showed a significant increase in total power in male healthy volunteers. Our findings were in line with Gengdi. They found that lower
You can
baseline.
conclude it. Actually, you are running out of time. Long time.
Yes, sir. Non-parametric indices, ST2 in both the genders, and decrease in entropy and approximate entropy. Correlation of BMI with the HRV parameters. Our study is the first of its kind to report the correlation of HRV parameters with BMI. Concluding this, BMI and gender influences the short-term effects of TAVANS on time domain indices and nonlinear methods of HRV analysis. The changes in HRV parameters following TAVANS were more significant and also negatively correlated with BMI. This indicates that the persons who are having higher BMI will have lesser effects to TAVANS and vice versa. Limitations, very low sample size. QEG was not done to assess the brain changes. Gonadal hormone assay was not done. Females, strength of our study, females in early proliferative phase was only included. Participants with normal BMI range were only included. Only younger adult groups were recruited. Future directions, all age groups in larger sample sites will be included. Long-term effects of TAVANS need to be explored. QEG can be done to explore the biofeedback technique. Further studies can also show the effectiveness of TAVANS in older population and peoples with disabilities who can't able to do asanas and yoga. It will replace the traditional interventions like yoga and exercise. These are my references, and thank you.
Thank you, Doctor. Judges can shoot the questions.
Madam, I'm very unhappy with your slides, madam. I stuck up with your just aim slide and I didn't proceed at all with other slides and I couldn't see the values at all. That was a big drawback for us as judges. We couldn't judge your values, especially to correlate your scientific statistical data. Second, which I would like to add was when you try to stimulate the vagus nerve when the ear using the Simba concha and the cabum concha, did you try to stimulate the tragus also, which is also involved in vagus nerve stimulation?
Yes, sir. We
Tragus.
didn't stimulate the tragus, sir. In our study, we did Simba-Con call only,
Because
sir.
when you stimulate,
Based
you're
on
supposed to stimulate all the three areas because which represents a classical vagal stimulation to see the HRV variation, madam.
yes, sir.
Okay, and another thing which I wanted to ask was when you try to compare the male and the female patients, and you find the males were more of HRV changes were more in the males. Is that right, madam?
Yes. Yes,
But,
sir. Yes, sir.
But you told BMI, I selected only the smaller BMI. Why you didn't select the higher BMI?
Sir, we actually included only the BMI range of 18 to 23.9, sir, to avoid the difference. In this, the 18 were more effect compared to the persons who are having 23 and all, sir. In our future studies, we will include the persons having increased BMI, sir.
Ilakaya, when
Sir?
You need to select a larger range.
Yes, sir.
Hello, am I audible?
Hello, yes.
Yes, audible.
Yes.
Yeah. See, that was my question. And the frequency domain also didn't correlate at all. You didn't try to correlate the frequency domain with your BMI also.
Sir, we did the correlation, sir, but the values were not that much significant. In that, mainly LF, HF, and total power were negatively correlated with the BMI in males, sir.
Okay, madam. Okay. Now let's see how you proceed with your further studies. Okay.
Okay, sir. Thank you, sir.
Thank you, Dr. Lakina.
Thank you, sir.
Yeah. Next is by Dr. Sweta Krishnan. Sweta Krishnan, you can share your slides.
Yes, sir. Am I audible, sir?
Yeah, yes, you are audible.
Yeah, thank you, sir.
Start now.
Yeah. Sir, is the screen visible?
Yeah, visible.
Yes, sir. Thank you, sir.
Make it full screen.
Yeah. Can I start, sir?
Yeah, you can start.
Good afternoon, everyone. I'm Dr. Shweta Krishnan, second year physiology postgraduate from Osmania Medical College, Hyderabad. My topic today is nerve conduction velocity in males and females, a comparative study. Nerve conduction study is a part of electrodiagnostic procedures that helps in establishing the type and extent of abnormality of the nerves. The determination of nerve conduction velocity is a useful method to diagnose various pathophysiological changes and neuromuscular disorders, and also to describe the status of peripheral innervation. The use of nerve conduction velocity measurement as a diagnostic tool requires a knowledge of range of values encountered in healthy individuals to set a normative data. There are both anatomical and physiological variations to the nerve conduction velocity. Usually, the nerve conduction velocity depends on the fiber diameter of the nerve, degree of myelination, and the internodal distance. Other factors such as age, temperature, height, gender, and limb are also physiological variables which is affecting the nerve conduction velocity. So it is crucial to set a normative data taking these factors into consideration. So our aim in this study was to compare the motor nerve conduction velocity of the median nerve in males and females. The objectives of the study was to measure the proximal and distal latency of the recorded signal deflection, to measure the distance between the two stimulation points, and to calculate the conduction velocity of the median nerve in both upper limbs of the individual. Then to compare the nerve conduction velocity of the median nerve in males and females. The inclusion criteria involved in the study was 17 to 30-year-old individuals were taken, both male and female subjects, and all the subjects were right-handed individuals with a BMI less than 25. The exclusion criteria of the study were left-handed, mixed-handed, and ambidextrous individuals, and individuals with any history of implant fixation, including pacemakers, any old or recent history of fracture, any local deformity, and any history of nerve compression, neurological disorders, addictions, metabolic disorders, genetic disorders, and endocrine disorders were excluded from the study. Subjects with diabetes or hypertension or dyslipidemia and any family history of neurological, cardiovascular, or metabolic disease were also excluded from the study. Coming to the methods and materials, a total of 60 subjects, including 30 males and 30 females, aged between 17 to 30 years of both genders without any comorbidities, were taken as the study population. The NCV equipment used was Natus Nicola TDX system, and the Flinders Handedness Questionnaire was used to assess the true handedness of the individuals taken for the study. After a detailed history taking and getting an informed consent, an electrical stimuli was applied over the median nerve at the distal and the proximal side, the distal being near the wrist and the proximal being near the elbow. The length between these two stimulation points were measured, and the conduction velocity is equal to distance by proximal latency minus distal latency. The calculated conduction velocities of these two group of people were tabulated and compared. The results of the study were the mean age of males and females included in the study were 22.76 years and 21.83 years respectively for males and females. The mean height of the males and females included were 173.07 centimeter and 158.07 centimeters respectively. The mean conduction velocity calculated was again tabulated in terms of dominant limb, the right hand, and the non-dominant limb, that is the left hand. In dominant limb, males had a nerve conduction velocity of 62.77 meter per second, and females had a value of 61.38 meter per second. Nerve conduction velocity of 59.75 meter per second, and females had 62.23 meter per second. The mean conduction velocities plotted as a graph. You can see that the dominant limb in males have a higher value, and in the non-dominant limb, females have got a higher nerve conduction velocity. Coming to the statistical analysis, these values were statistically analyzed using independent sample t-test, and the p-value less than 0.05 was taken as significant. On comparing the values between males and females, in the dominant limb, p-value was 0.1281, which was insignificant. And in the non-dominant limb, p-value is 0.02376, which was a significant value. Coming to the discussion, in the dominant limb, that is the right hand, because the individuals taken were all right-handed individuals, the mean conduction velocity was higher in males. As you can see in this graph, the mean conduction velocity in the dominant limb was higher in males. And this can be explained by the height differences between the both genders taken for the study. The mean height observed was males had 173.07 centimeters and females had a mean height of 158.07 centimeters. And this is a graph showing the mean height of the study subjects in males and females. Coming to the non-dominant limb, the mean conduction velocity was higher in females. In contrast to the dominant limb, it was higher in females. And the difference was statistically significant, which was less than 0.05. So on further analysis, there was an interesting observation that the interarm difference in the nerve conduction velocity was higher in males. We already know that dominance also plays a part in determining the nerve conduction velocity. So in this study, the subjects included were seen to have different interarm differences. In males, the dominant hand, that is the right hand, had a more nerve conduction velocity. In females, it was more in the left-handed left hand. That is the non-dominant arm. So conclusion, the gender has definite effects on nerve conduction velocity, and hence it is important to consider this difference before making a diagnosis based on these electrophysiological studies. Also from this study, we have found that dominance also plays a part, and it is a factor. So further studies can be done based on dominance, and the study also suggests that a different set of normative values should be followed for both the genders. The limitations of the study were limited sample size. These were my references. And I would like to thank my Department of Physiology, Osmania Medical College, Department of Neurology, Osmania General Hospital, my seniors, fellow postgraduates, and juniors. Thank you.
Thank you, Shweta.
Thank you, sir.
I request the judges to ask the questions. Judges can ask the questions.
Hello.
Yes, sir.
Yeah, can you hear me?
Yes, sir, you can.
Yes,
I
sir.
can
Yes,
hear
sir.
you.
Yeah. Madam, it was a very good study, madam.
Thank you, sir.
What I wanted to know was, when you selected the age group between 70 to 30,
Yes,
the
sir.
height, average height was 173.3. While
In
they were
males.
all tall students, is it?
Yes, sir, it is that mean height was 173, sir.
Yeah, mean 73, 173 is it's a very huge height when
Yes,
compared
sir.
to Indian standards.
Yes, sir.
So,
Most of the subjects were students, sir. So
yeah,
mostly they were.
even then you could have taken a little more smaller students and you could have compared. And see, anywhere, whenever you have a height increase, the nerve conduction study is likely to increase, madam. So that is
Yes,
what
sir.
we know very well. The conduction velocity increases when the length of the vision increases. So
Yes,
I
sir.
feel that bias should have avoided. You should have taken a smaller group and a larger group, whereas the results would have been better.
Yes, sir.
Okay, and I
Yes.
wanted to know dominant limb and how is it going to apply yourself clinically when you tell about the difference in the dominant limb and the non-dominant limb. So when
Yes,
it's a
sir.
right-handed person or left-handed person, what difference does it make?
Sir, there are so many studies saying that dominant limb has more motor nerve conduction velocity because the studies say the motor neurons on the dominant limb are tends to be larger than the non-dominant limb, sir. This is one of the
I
reason they
want to
have
know
given
how
in
is it
certain
applied
studies, sir.
clinically. How it is applied
Sir,
clinically, madam?
yes, sir. Yes, sir. So mostly nerve conduction studies are one is a main diagnostic feature for carpal tunnel syndrome, sir. So, for example, the normative data mostly will be done with the dominant limb, sir, mostly the right hand. So, if at all a person is left-handed or the issue is with his non-dominant limb, the same normative value will be used for diagnosing it in the non-dominant limb, sir. So, there might be chances that we could misdiagnose a person with carpal tunnel syndrome could be not having it, so we could miss a diagnosis based on those values.
Now, why do you think it was higher in the females?
Yes, sir, that was an
Your
interesting
conduction
difference we
velocity.
found. Yes, sir, that was the finding we got in our study, sir. More and similar studies, so many studies show that females have more nerve conduction velocity, but there are equal number of studies saying males have because of the limb height differences. Males also
Yeah,
have.
limb
So that's
differences,
a
there'll be a
conflict.
limb difference. No, apart from the hormonal changes,
Yes, sir.
there'll be limb differences. So how can
Yes,
you
sir.
interpret this in the females?
Sir, in females again, based on the menstrual phase which they are in, again there are studies showing there is differences in the motor nerve conduction
Now, did
velocity
you did,
during the.
did you differentiate the menstrual cycle, follicular phase and progesterone phase and the menstrual phase? Did you try
No,
it
sir.
in these phases? No.
No, sir. Just it was comparison between males and females, sir.
Okay,
Studies
okay,
can be done within females among the
okay.
3 menstrual phases, sir.
Okay, madam.
Yes, sir. Thank you, sir.
Thank you, Shweta. Next is by Dr. Sonika. Dr. Sonika, you can share your screen.
Yes, madam, you share your slide.
Yes, sir. Yes, sir. So, yes. Sir.
Full screen.
Good afternoon to everyone. I am myself
Go to the
Dr.
slideshow.
Make it
Sonita
a full
from BMC
screen.
Nalgonda. Yes, sir.
Yeah, okay, you can start.
Okay, sir. Full screen.
Yeah,
Okay,
you
sir.
can start.
Can I start? Okay, sir.
Yeah.
Okay, so the topic for my paper is alteration in glycated hemoglobin levels in non-diabetic adult individuals with abnormal thyroid status. First of all, background. Glycated hemoglobin has been considered as an important marker of long-term glycemic control. The American Diabetes Association has suggested the use of HbA1c as diagnostic tool for pre-diabetes and diabetes. So the normal value of glycated hemoglobin is between 4 to 5.6%. But a value between 5.7 and 6.5% represents pre-diabetes, while a value greater than or equal to 6.5% is considered as diabetes mellitus. HbA1c levels may change due to altered thyroid status. HbA1c levels in hypothyroid and thyrotoxic individuals who do not have diabetes or pre-diabetes based on fasting and 2-hour post-glucose plasma glucose criteria is measured. Then studies done in hypothyroid patients have shown spurious elevation of HbA1c not only in presence of diabetes but also in non-diabetic subjects. Whereas HbA1c levels in hyperthyroid patients were not significantly different from controls. Now aim. We have to evaluate the effect of thyroid states on glycated hemoglobin levels. Methodology. Number of patients. We took total 120 patients, including both males and females, visiting the outpatient department. Under inclusion criteria, we took patients between age group 21 to 70 years. Cases, we took patients diagnosed with hypothyroidism or hyperthyroidism. Controls, in the control group, we took healthy individuals. Then for both cases and control groups, fasting blood sugar levels were less than 110 milligrams per deciliter as we are taking non-diabetic subjects. Age and gender matched controls were selected. Exclusion criteria, patients with age less than 21 years and more than 70 years. We excluded pregnant women. Then patients with diabetes, liver diseases, renal diseases, hematological malignancies, hemoglobinopathy, recent blood transfusion, and patients on thyroxine and carbimazole. These were excluded. Study design. The research study was done in GMC GGH Nalgonda, Telangana, with the help of Department of Medicine, Department of Biochemistry, and Department of Pathology between August 2023 and October 2023. Parameters like fasting blood sugar, hemoglobin, thyroid profile, like T3, T4, TSH, and HbA1c, which is glycated hemoglobin, were collected before and after the treatment and evaluated. All participants' consent was taken through informed consent form. Method of statistical analysis. It was a case control study with 60 cases and 60 controls. All the data was collected and analyzed statistically for calculating significance of parameters using SPSS software. All values were given as mean plus minus standard deviation. Comparison between cases and controls were made using students' t-test, which is unpaired, and p-value less than 0.05 was considered statistically significant. We took unpaired t-test because we had to take sample from two different groups of populations, and t-test is used to compare the two different groups. So materials were blood sample, glucometer, and injections. So in the result, there is comparison of age and sex in the age and the cases and control groups, and the values, and for this values are taken as mean plus minus standard deviation. For cases, we took 34 males and 26 females, whereas for controls, we took 30 males and 30 females. Now this is the comparison of the different parameters in the study and control groups along with the p-values. The parameters are FBS, hemoglobin, T3, T4, TSH, and HbA1c. Here we can see that in TSH, the mean, there is quite a large difference between the mean, 6.98 and 2.62. And the p-value is also less than 0.0001. HbA1 in HbA1c also p-value is 0.0010. So there is a relation between TSH and HbA1c. This is statistically significant. There was statistically significant difference between p-value of less than 0.0001 and levels of serum TSH were significantly higher in study group compared with the control group. Again, there was statistically significant difference with p-values of 0.0010 and levels of HbA1c were significantly higher in study group compared with the control group. So the conclusion is there was a positive and statistically significant correlation between serum TSH and HbA1c levels. HbA1c reduced significantly with treatment in hypothyroid patients without any significant change in glucose levels, unlike in hyperthyroid patients. HbA1c levels were misleadingly high in non-diabetic subjects with hypothyroidism. This is very important. So HbA1c data should be interpreted with caution in non-diabetic patients with hypothyroidism. These are the references. Christie A.L., Manjrekar T., Babu R.P., M.S.R., Hegde A. Elevation of HbA1c in non-diabetic hypothyroid individuals is anemia, the connecting link, a preliminary study. Second is Rana Bhattacharya, Anubhav Thukrel, Partha Pratim Chakraborty, Ajitesh Roy et al. Effects of thyroid status on glycated hemoglobin, Indian Journal of Endocrinology and Metabolism. Anant Paru S, Vaikara S, Sachan A, Suchitra M. Reddy AP et al. Effects of thyroid hormone replacement on glycated hemoglobin levels in non-diabetic subjects with overt hypothyroidism. And Zhao Fai Gao, Hissing Ling Li et al. Thyroid hormone replacement receptor beta and NCA4 regulate terminal erythrocyte differentiation. These were the references. Thank you.
Thank you. Judges can ask the questions.
Madam, your presentation was good. Only thing is, these are all were known facts. Like we know HbA1c will be decreased in when the TSH level also decreased in hypothyroid patients. Now, what was your rationale on selecting this title and this aim of the study?
Sir, actually, this topic is very least explored in Telangana state. So I thought to choose this topic and see it myself for some experience in the topic.
Because we know in hypothyroid patients, definitely HbA1c, A1c in your study, what was the value? Was it increased or was it decreased?
Sir, HbA1c value was increased, but after the treatment, it decreased in hypothyroids. Whereas in hyperthyroid, there was no relation with TSH and HbA1c.
So what do you like to come to the conclusion?
Sir, conclusion is that there is spurious elevation of HbA1c in hypothyroids. So it is very misleading because it happens because the half-life of RBCs is more. So this is happening. So this is misleading, and we have to be very careful with the patients who are hypothyroid while measuring their HbA1c.
See, in hypothyroidism, there is already a low metabolic state. So when there is a low metabolic state,
Yes.
you mean the HbA1c concentration will increase?
Yes, sir. HbA1c is increased in hypothyroids.
I know why. Why I'm not able to understand
Sir,
the metabolic
sir,
activity
because
is low.
yes, sir, because there is high in hypothyroids, the erythropoiesis is decreased erythropoiesis. So there should be decreased HbA1c, but it is increased, which is false because the half-life of RBCs is more.
So on the contrary, in hypothyroid patients, we expect the values to go down, HbA1c.
Sir, in the study, it shows that in hyperthyroids, there was no change after treatment also. Before treatment also, there was no such association with HbA1c, and after
So
treatment
there
also there was
you
no
will find
change.
there is relatively erythropoiesis is higher in hypothyroid state also.
Yes, sir. Yes, sir.
Then you would expect HbA1c to decrease or increase.
Sir, it will remain same, sir. Sir,
No, need not. Yeah, when you find hypothyroid state,
okay,
it's
sir.
increased.
Okay, sir.
And hyperthyroid state, it should be. Okay,
Okay, sir.
madam.
I
Slight
will
study
start now.
correlation.
Okay. Okay, sir.
Let's see how we are going to proceed. Okay.
Okay, sir.
Thank you, Dr. Sonika.
Okay, thank you.
Chandrasekhar sir. Ilakia want to present
Okay,
again.
sir. We'll go to the next speaker, sir.
Actually, Ilakia want to present again. May I allow her, sir, or?
No, sir. No
Okay,
time.
sir.
No time. Already
Okay.
we are running
Thank you,
short
sir.
of time.
Yeah, yes, sir.
There are 3 more speakers.
Yeah, yes, sir. Yeah. Next is by Dr. Hossein I. Dr. Hossein, you can share your slides.
Yes, sir. Yes, sir. Sir, is it visible, sir?
Visible, yeah. You can start now.
Okay, sir. Thank you. Good afternoon, sir. I am Injamamul Hussain. Title of my project is Effect of Neoadjuvant Chemotherapy on Endothelial Function, Stress Level, and Oncovascular Risk Factor among primary breast cancer patients. They are my respected guide and co-guides. So come to the background. Oncovascular risk factors are those biomarkers which are found altered in both malignant transformation and cardiovascular diseases. Serum level of various cytoadequines such as leptin and adiponectin and cell adhesion molecules have been correlated with breast cancer. High prevalence of psychological distress have been reported in breast cancer patients. Risk for cardiovascular disorder and endothelial dysfunction are expected to be higher among the breast cancer patients who receive neoadjuvant chemotherapy. Come to the rationale. Our expression of overexpression of leptin and leptin receptor improve the therapeutic efficacy of neoadjuvant chemotherapy. Low serum adiponectin levels are associated with large tumor size and poorer prognosis of breast cancer. In breast cancer patients, significant increase in serum level of SVCAM and SICAM have been found with increasing stages of breast cancer. Come to the novelty, only few reports are available about the level of cell adhesion molecules and adipokines before and after NSCT in breast cancer patient. There is scanty information available about stress levels and endothelial dysfunction among breast cancer patient after NSCT. Aim of our study was comparison of the serum level of oncovascular risk factors, stress level, and vascular endothelial function before and after NSCT among locally advanced primary breast cancer patients. Primary objective was estimation of serum level of leptin, adiponectin, SICAM and SVCAM level, assessment of endothelial function, evaluation of stress level using standard questionnaires. Secondary objective was assessment of interrelationship among cell adhesion molecules, adipokines and endothelial function stress level in breast cancer patient before and after NSCT. For sample size calculation, we use clinically significant data, that is brachial artery flow mediated dilation, work done by Q1 Lee et al. With 99% confidence interval and 95% power. Our initial calculated sample size was 17. With the expected dropout rate of 25% and adjustment of multiple comparison, we came into a larger sample size, that is 40. Come to the methodology. Postmenopausal breast cancer patient between age group 50 to 65 years are having hemoglobin level greater than 10 gram per deciliter included for our study. Breast cancer patient having BMI less than 18 kg per meter square and having BMI greater than 20 kg per meter square, and patient with liver and kidney diseases and patient with metastasis are excluded from our study. 40 newly diagnosed breast cancer patients planned for NSCT were recruited from the Department of Medical Oncology, JIPMER. Stress levels are assessed using standard questionnaires. This is perceived stress scale, patient health questionnaire 9, and generalized anxiety disorder 7 before and after NSCT. Endothelial function was assessed by brachial artery flow mediated dilation using color Doppler before and after NSCT in Department of Radio Diagnosis, JIPMER. 5 ml of fasting blood sample were collected before and after NSCT for estimation of oncovascular risk factors such as leptin, adiponectin, and various cell adhesion molecules, that is SBCAM and SICAM, using ELISA technique. For statistical analysis, for normal distributed data, PRT test was used. For non-normal distributed data, Wilcoxon sign rank test was used. McNamara test was used for frequency data analysis, and effect size was calculated using Cohen's d formula. Come to the result. In comparison of lipid profile in breast cancer patient, we found significant increase in the total cholesterol, LDL cholesterol, triglyceride, and non-HDL cholesterol level in breast cancer patient after chemotherapy. In comparison of oncovascular risk factor and endothelial function among breast cancer patient before and after chemotherapy, we found significant decrease in serum leptin level with medium effect of 0.5. We found trend of increase in adiponectin to leptin ratio in after chemotherapy group. We found trend of increase in SICAM level in breast cancer patient after chemotherapy. In comparison of brachial artery flow mediated dilation, we found significant decrease in the BFMD percentage with median small effect of 0.3. This is graphical representation of leptin level before and after chemotherapy, BFMD level before and after chemotherapy. In comparison of perceived stress scale, patient health questionnaire 9, and GAT7 score in breast cancer patient, we found significant increase in all the questionnaire in after chemotherapy group with larger effect size. This is a graphical representation of PSS score, PHQ-9 score, and GAT7 score. In frequency data analysis for depression, we found significant increased number of breast cancer patient in mild and above depression group after chemotherapy. That number is 16 to 30. In frequency data analysis of anxiety, we found significant increased number of breast cancer patient in mild and above anxiety group. That number is 7 to 28 in after chemotherapy group. In frequency analysis of stress level, we found increased number of breast cancer patient in moderate and above stress group. That number is 6 to 12. For correlation analysis between various parameters before chemotherapy, we found significant positive correlation between BFMD GAT7, PHQ-9 GAT7, and SICAM and SVCAM. For correlation analysis between various parameters after NSCT, we found significant positive correlation among PSS PHQ-9, PSS GAT7, and PHQ-9 GAT7. Come to the discussion. A decrease in serum leptin level. A decrease in serum leptin level and trend of increase in adiponectin to leptin ratio was found in breast cancer patient after chemotherapy. Tuss et al. Reported decrease in serum leptin level in non-small cell lung cancer after chemotherapy. Decrease in the BFMD level or increased endothelial dysfunction found among the breast cancer patient after chemotherapy. Kiwanli et al. Reported increased endothelial dysfunction in breast cancer patient who received anthracycline based chemotherapy. So in conclusion, can tell increased endothelial dysfunction, stress level, and altered oncovascular risk factor and dyslipidemia were found among the breast cancer patient after chemotherapy. In future study, we can may conclude the follow-up of the breast cancer patient to understand the recovery or persistence of the comorbidities after chemotherapy. Thank you.
Thank you, Hussein. Yeah, that just can answer the question.
Yeah, Mr. Hussein, it was a well-conducted study and you presented well.
Thank you.
I have some queries. This neoadjuvant chemotherapy, now what is the drug which is used there in these patients? Do you have any idea?
In our JIPMER, for first 4 cycle, they used to use that FEB, 5-fluorouracil, epirubicin, and next 3 cycle, they are using only doxorubicin in our JIPMER protocols.
Yes. So after how long you collected the sample after this neoadjuvant chemotherapy? You told before and after, no? After how long this chemotherapy you collected sample here?
Ma'am, I recruited just before their chemotherapy, and I waited for fulfill of their total 7 cycle of chemotherapy. Then second sample I collected.
Okay then. And can you explain the mechanism? Why do you find this endothelial dysfunction and this cell adhesion molecules alteration, pathophysiology of why it is there?
Yes, ma'am. We all know that one of the major hallmark of oncogenesis, that is endothelial cell differentiation, ma'am. Correct. So when there is higher differentiation of endothelial cell, means that cell adhesion molecules, that is SBCAM and SICAM, will increase in the solubility form. And another thing is that our brachial artery flow mediated dilation, this is a vasodilator activity I am checking actually here, activity. So when there is abnormality in endothelial functionality or plasticity, so this vasodilatory activity is decreasing. So this is kind of finding for me.
This drug you told, that doxorubicin, is known to
Yes, ma'am.
decrease the nitric oxide formation. That is the main
Yes.
reason for this endothelial dysfunction.
Okay, okay, ma'am. Okay, ma'am.
Yeah,
Thank you.
thank you, Mr. Rossi.
Okay.
Thank you, Mr. Hussain.
Thank you. Thank you so much.
Next presentation is by Dr. Kotra Navya. Kotra Navya, you can share your slides.
Okay, sir. Sir,
Maybe you can share because we cannot see your slides.
sir, the last presenter has to close their screen, sir.
Dr. Hussain?
Yeah.
Dr. Hussain?
No, this is it. Yeah, this now it's done.
Yeah, you can share. Yes, visible. Make it full screen and you can start the presentation.
Sir, is it visible?
Yes, visible. Yeah, you can start.
Okay, sir. Good afternoon, judges. Myself, Navya, PG Biochemistry, Savita Medical College. My topic is metabolic score for insulin resistance index as a marker for glycemic control in normal, overweight, and obese population. Introduction. The diabetes mellitus is a metabolic disease that is characterized by the elevated blood glucose levels. It is classified into type 1, type 2 gestational diabetes and neonatal diabetes. The poor glycemic control will lead to the multi-system damage and the pathological changes, functional changes, declines in various tissues and organs. So the glycemic control is one of the critical issue in the clinical management of diabetes. The metabolic score for insulin resistance is a novel non-insulin based marker for assessing the risk for insulin resistance. It also predicts the visceral fat content, subcutaneous adipose tissue, fasting insulin levels, ectopic fat accumulation in liver and pancreas. The people with impaired glucose levels and obesity are more prone for the developing atherosclerosis. Hence, the current study was planned to determine the MET-SIR as a glycemic control marker in normal, overweight, and obese population. The aim and objectives are to compare MET-SIR index between normal, overweight, and obese population, and to study the association of MET-SIR with fasting, postprandial blood sugar, and HbA1c, and to identify the cutoff value of MET-SIR as glycemic control marker. The methodology, the study type is cross-sectional study, and the sample size was calculated by the G power, and the minimal sample size was 338 for ANOVA, and further it was increased by 10% to gain the confidence, and the final sample size were estimated as 372. The 372 subjects were categorized as three groups, 124 normal, 124 overweight, 124 obese. The inclusion criteria was people coming from for master health checkup in our hospital, and the exclusion criteria is pregnant women, known CKD patients, and known high triglycerides levels. The study was started after getting the approval from the institutional committee, and also detailed clinical history was collected from the study subjects through a proper questionnaire. The BMI was also calculated. As per the WHO guidelines for BMI in the Asian populations, the normal is 18.5 to 22.9. The overweight is 23 to 24.9, and the obese is more than 25. 5 ml of fasting venous blood sample was collected and assessed in the Vitros 5600 fully automated integrated biochemistry analyzer and the Biorad D10 HbA1c analyzer. And the following parameters are analyzed by the following methodology. The calculated parameters are VLDL, LDL, and MET-SIR. The statistical analysis were performed using SPSS and are expressed as mean plus or minus standard deviation. The MET-SIR, FBS, PPBS, and HbA1c are compared between the normal, overweight, and obese population. The person's correlation analysis was performed by MET-SIR and FBS, PPBS, HbA1c. The ROC analysis was done to identify the cutoff value of METSIR in identifying glycemic control. And the p-value of less than 0.05 was considered as the statistically significant. My results are, this is the comparison of the anthropometric measurements and the biochemistry and variables, where the weight, BMI, and METSIR are found to be statistically significant. This is the post HOC analysis of METSIR among the groups where the normal, overweight, and obese, the overweight, obese, and normal, obese, normal, and overweight. This is the positive correlation of the METSIR with FBS, PPBS, and HbA1c in the normal population. This is the another positive correlation of METSIR with the FBC, PPBS, and HbA1c in overweight population. This is the correlation of METSIR, positive correlation of METSIR in FBS, PPBS, and HbA1c in obese population. This is the ROC curve, where the highest range of the METSIR is found to be 0.842, and the cutoff value of METSIR was 39.63 with the sensitivity of 79%, and the specificity is 75%. My discussion. The type 2 diabetes mellitus is growing serious issue for the healthcare due to its higher prevalence and mortality rate. The proper glycemic control is associated with the lower prevalence with the microvascular and neuropathic events in patients with type 2 diabetes mellitus. And the HbA1c was routinely done to estimate the glycemic control status, but it was limited to the certain levels. In this cross-sectional study, we explored the association between the METSIR and type 2 diabetes mellitus and provided the information on early identification of the using with METSIR index. Bello Chawla has discovered the METSIR as a simple novel non-insulin based marker in the year 2018 for assessing the risk of insulin resistance. In our findings, we have showed the ability of the METSIR as a potential use for indicator for the identification of individual with glucose metabolism disorders. It is also correlated well with the FBS, PPBS, and HbA1c in all the three groups, which is normal, overweight, and obese population. It indicates that METSIR can identify the glycemic control status in the early adulthood and prevent the complications of the diabetes. My conclusion, the METSIR is a potential useful indicator for the early identification of glycemic control. It is a simple and easily calculated marker used for the glycemic control, and the levels of METSIR in early adulthood can be associated with high risk for diabetes. These are my references. Thank you.
Thank you, Kotra. Judges can ask the questions.
Dr. Navya. Yeah, it is a well presented. Congratulations. I have some queries. Okay. Do you find any sex difference between male and female with respect to this METSIR?
In males, it is when compared with females, males we have received high marks.
Males are having high values. You didn't compare this indices with other indices, only this you measured.
Yes, ma'am.
Okay, but you would have compared with other insulin sensitivity indexes also. That would have been
okay, ma'am. In my further study, I will.
So what is your future plan? How can you proceed this with further studies?
Maybe with the lipid profile and all, where I can compare and
you can actually do a follow-up after treatment, how this is improving. That can be an idea. You included only, I mean, you excluded only individuals with the high TG levels, or those who are having those who are on drugs also you would avoid it?
No, ma'am, only high TG levels in pregnant.
Okay then. Okay.
Thank you, Dr. Navya.
Thank you.
The last one is by Dr. Nitya. Yes.
Yeah, Doctor, I have one question.
Yeah. Navya.
The question is whether this METSIR is really a reliable index. This was come in 2018, and now the formula has come out and people have started using. How reliable is this METSIR, madam?
It's reliable, sir, but mostly it's not used still, sir, even though it's found in 2018.
I just want to know, because people started using, but I do not know how reliable it is, because it tries to identify your HDL fat, LDL fat. It makes a calculation on its own, seeing the fat at the pancreas, your liver, abdomen thing and all. So how far it's reliable, we have to wait and see, madam.
Yes, sir. Shall I add it, sir? The thing is, the marker which is used is HOMO-IR, sir. The problem with HOMO-IR is we have to measure this insulin level, which is highly costlier. You have inter-laboratory variations for this, sir. Whereas with respect to this, the index which you med-IR, it is used on simple parameters like lipid profile, glucose, which takes care of all the components of the metabolic syndrome, sir.
Ratios and all they.
Because we have some criteria for metabolic syndrome, sir. It includes every one criteria as a part of that calculation. That is the advantage over the HOMO-IR which we are using, sir.
Okay.
Thank you, Dr.
Thank you, sir.
Nitya, you can share your screen.
Sir, is it visible?
Yeah, you can share the screen. Yeah, make it full screen and you can start.
Yeah. Is it visible, sir?
Yeah, visibly. Make it full screen.
Yeah, okay. Thank you.
Yeah, you can start.
Good evening to all. My presentation is on topic study of comparison between total bilirubin level, total bilirubin and albumin ratio with disease activity in rheumatoid arthritis patient. Introduction. Rheumatoid arthritis is a chronic inflammatory systemic autoimmune disease which is marked by polyarthritis with extra-articular manifestations, and the main pathogenesis of rheumatoid arthritis is based on the oxidative stress. So the reactive oxygen species and ROS oxidized products cause damage to the lipids, protein, and DNA, which triggers the production of autoantibodies. Bilirubin, the final product of heme catabolism, at physiological or moderately elevated concentration, it has potent antioxidant and powerful immunomodulatory ability. At higher concentration, it has cytotoxic effects on multiple tissues, especially it's exerted by the serum-free bilirubin, which can cross the intact blood-brain barrier. Since there is no assay to measure the serum-free bilirubin, we use total bilirubin albumin ratio, which is considered. The aim of the study is to study the relation between total bilirubin albumin ratio, and disease activity in rheumatoid arthritis patient. Materials includes a cross-section study involving 102 rheumatoid arthritis patient and outpatient inpatient from the Department of Orthopedics and Rheumatology in GMKMCH Salem. The patient who attended the master health checkup in outpatient was selected as healthy controls. Inclusion criteria: patient fulfilling the 2010 rheumatoid arthritis criteria of American College of Rheumatology is considered. Exclusion criteria: patient with any elevated liver enzymes, renal failure, viral hepatitis, and with active infections, and also patient who has undergone hepatobiliary and pancreatic surgery, malignant tumor, and other autoimmune diseases were excluded. Method: serum bilirubin albumin C-reactive protein measured with ERBA autoanalyzer, and erythrocyte sedimentation by Westgen method, rheumatoid factor by agglutination method, interleukin-6 measured by electrochemiluminescence immunoassay method, and other clinical and laboratory data including age, sex, medical history, disease duration, and other parameters were collected from the records. The disease activity score 28 ES, so that is DAS score, was calculated. The value of score of less than 3.2 was considered patient in remission or in mildly active disease. Of that, 41 patients were recruited, and the score between 3.2 and 5.1 was considered moderately active, and 29 patients were included, and score of more than 5.1 considered severely active, and 32 patients were included. Statistical analysis: mean and standard deviation was used for normal distributed variable, and median interquartile range for non-normally distributed variables. Numbers as percentage for categorical variables, which is compared by chi-square test. The correlations were done with Spearman rank coefficient correlation and Pearson correlation. P-value of less than 0.5 were found to be statistically significant, 0.05. Results: the total bilirubin of value 10.7 micromole per liter in rheumatoid arthritis patients was significantly lower than the healthy controls of 13.7 micromole per liter. Total bilirubin and albumin ratio of 0.24 micromole per gram in rheumatoid arthritis patient was significantly lower than the healthy controls of 0.3 micromole per gram. So this is a box whisker plot showing the total bilirubin levels in healthy controls and in the rheumatoid arthritis patient. There is a significant difference. And also the total bilirubin and albumin levels in healthy controls and in rheumatoid arthritis patient. There also we can see a significant difference. The other laboratory characteristics here in this table column, we can see total bilirubin and albumin has a significant decrease with the increase in the disease activity. The other parameters like ESR, CRP, interleukin 6 have a significant increase with the increase in disease activity. And there is also positive of rheumatoid factor IgM in the significantly associated with the DAS score. Spearman rank correlation analysis revealed that bilirubin, total bilirubin albumin ratio, they were negatively correlated with the disease activity and also with the inflammatory markers like CRP, ESR, and interleukin 6. This is the correlation graph between total bilirubin. This is activity score 28 in rheumatoid arthritis patient. It shows the negative correlation. And all the total bilirubin albumin ratio with the DAS score shows the significant negative correlation. This is a correlation of here we can see the DAS score has the negative correlation, and the ESR and C-reactive protein all has the negative correlation with the significant negative correlation with the total bilirubin and total bilirubin albumin ratio. And interleukin 6 has the negative significant correlation with total bilirubin, but has no significant correlation between the total bilirubin albumin ratio. This is a graph showing the correlation between total bilirubin and total bilirubin albumin ratio with the ESR, which shows the negative correlation. Discussion. The study demonstrated that serum bilirubin has significantly decreased with increase in disease activity, and it may use as an indicator to assess the extent of disease activity. As bilirubin acts as an effective scavenger of reactive oxidant species, it indicates a possible antioxidation property of bilirubin. And since bilirubin levels remain associated with reduction of disease activity, it suggests it may be a protective factor for RA aggravation. And the patients with the higher bilirubin levels, they are likely to achieve remission, suggesting it can be tried therapeutically. Albumin related parameters like albumin fibrinogen ratio and CRP albumin ratio has emerged as a novel biomarkers for predicting inflammation. So with this precedent, we move forward with the total bilirubin and albumin ratio, and it has an inverse association with the disease activity and can be used as a reliable index for assessing the degree of rheumatoid arthritis. Conclusion. So this study suggested that total bilirubin albumin ratio has inverse association with disease activity, and they can be used for assessing the disease progression. Limitations of the study is a small sample size. The factors affecting the disease progression, such as medications, were not factored in, and also factors affecting serum bilirubin, such as alcohol, smoking, anemia, were also not factored in. The causal factors cannot also be concluded in the study. These are my references. Thank you.
Thank you, Dr. Nitya. The judges can ask the questions.
Yeah, Dr. Nitya. So what is your implication of your study? Like, how it will be clinically helpful here?
Repeat, sorry, ma'am.
What is the clinical implication of your study? How you are going to put this study in your real practice?
Ma'am, for the now we are using for the progression of disease, we are using ESR, CRP, and interleukin 6. All these parameters or followed up regularly for any seeing any disease progressions. So instead of using the high costly parameters like interleukin 6 and CRP, we can switch for the serum bilirubin, ma'am.
So how this bilirubin helps in preventing the disease progression? It's only the antioxidant property, or it has got many other role to play?
Especially mainly because of the antioxidant, it also has an immunomodulatory effect. The serum bilirubin will inhibit the B cells and T cells production, so that the rheumatoid factors also gets decreased and interleukin 6 also get decreased, and also the other cells like neutrophils and also lymphocytes also decreased with the serum bilirubin, ma'am. So this
Madam, I have a
indicates.
question.
Yes, sir.
Madam, you carry on.
So these indicates the disease progression activity also, sir.
Madam, normally rheumatoid arthritis rarely affects the liver. Okay. Another thing is only people who are on long-term treatment, those people will have liver affected in a larger scale. And when you took the rheumatoid patients, did you classify into mild, moderate, severe, or people on medication, on not medication, or irregular medication? Those people are the people who gets the liver affected.
Yes,
Okay,
sir.
so how do you correlate this in your study, madam?
Sir, I have not factored in the medication. I've just scored with the ESR value and
No, you should have to ask what whether they are on a regular medication or they are on irregular medication. People only with irregular medication, you see the liver getting affected.
Yes, sir.
As
Yes,
per
sir.
the study, we know very well that rheumatoid arthritis normally doesn't affect the liver. Only when patients on long-term basis who are on irregular treatment, we see liver getting involved.
Yes, sir. So I have asked when collecting the data from them, but most of the people they are not on the continuous medication, sir.
Okay.
That patient I have recruited, they are not on continuous medications.
Okay. Okay.
Okay, sir.
Thank you, Dr. Nitya.
Thank you, sir.
Yeah, thus the session came to a conclusion. I thank all the presenters for a wonderful presentation. To know the results, please join tomorrow during the valedictory function. Especially, I thank all the three judges for spending their valuable time judging the session. I kindly request the judges to share the screenshot of the score sheets to the respective professors. So, thank you once again.
Thank you.